Good morning. To begin with today's proceedings, may I please invite Director of Wildlife Institute of India, Dr. Ruchi Babala, Dean Faculty of Wildlife Sciences, and Dr. S. Satyakumar, Registrar Wildlife Institute of India, with one individual. Welcome all of you to the 7th International Annual Research Seminar. We will have the annual research seminar next month, beginning of next month. So, I welcome Dr. G.S. Rao, former Dean and Director of the Institute of India. Dr. Rao has kindly agreed to chair this International Annual Research Seminar. We have guests from Indian Institute of Science, Education and Research, Mali, Dr. Mandeep Jain, we have Professor Lucy Dana from 7th University of Agriculture and Science and Technology. So, Dr. Proceed, who is also an alumni of the Institute of India, we have Dr. Raman Pura. So we have three guests in the seminar. I welcome all the research scholars, all the MSc students. So we have a variety of MSc students. We have MSc students from 15 batch, not many, two of them. We have 20 MSc students from 19th batch. We have 18 MSc students from the 28th batch of Wildlife Science. We have 14 MSc students from the first of the So I welcome all the master students, all the research scholars, all the officer training. We have 16 officer training who are listening to join. Welcome all of you. And with these works, we will start the proceedings of the International Session today. I request Dr. Ruchi Babala to give a welcome address.
Good morning. Respected Director of Wildlife Institute of India, Registrar Dr. Satyakumar, Research Coordinator Dr. Devash Kanda, Dr. Rama, former Director and Dean Wildlife Institute of India and the CMR Chair, Dr. Ruchi, Dr. Raman, Dr. Mandeep, my dear faculty colleagues, students, staff. Welcome to the internal annual research seminar. Now, this is one event of the institute which everybody who has been a part of this institute has witnessed and has participated in. And this whole concept of having these annual research seminars actually started with almost the initial research projects that this institute took. And the first research seminar took place in 1988. And that was the first annual research seminar. There was no IARS at that time, it was just ARS. And the ARS started with 7 to 8 presentations. And that time, because the number of students was less, the institute was small, we had very, very specific projects which catered to the issues of management and conservation of species which needed immediate attention. And they were most often large mammals. Some of them could have been, like rhinos and all that, but mostly it was large mammals, elephants, tigers, snow leopard. So these were the species that the initial researchers of Wildlife Institute of India were working on. And then came the second batch of research scholars to this institute, and an additional dimension was added to our research, and that was the human dimensions in wildlife conservation. And in 1989, in the second annual research seminar, two presentations on the human dimensions of wildlife conservation were initiated. Subsequently, this institute started developing its expertise on various aspects, starting from GIS, remote sensing labs, computer application, genetics, biology labs, and so on. And very, very slowly but steadily, we started making our inroads into these fields. And initially when we made presentations on these aspects, it was very novel for us. It was something new and something which was taken very seriously. And we had a lot of experts who comment on, and not so many experts. Some real experts and some not so real experts, but very, very concerned about the application of these techniques into the conservation and management of species. Over the years, the number of students increased, the number of projects increased, and slowly we as an institute started to understand that the problems of conservation facing multi-dimensional, and probably we needed to move away from species-specific research to landscape-related research, ecosystem-related research, habitat-related research, and so on. And so the flavor of the research projects also started changing, and thus the annual research seminar started, increasing in size. The number of projects increased. Each project had so many dimensions because it was realized that we need an integrated approach. And so, not all, some were actually focusing on the science and ecology of certain species or whatever the topic was, but many projects started having different dimensions to them. And therefore, even the single project could have 3 to 4 presentations because a lot of work was being done, as I said, on habitat, on mapping, on genetics, and so on. And therefore, the number of presentations started increasing. Now, let me also tell you that the annual research seminar was also a way of meeting and networking with the conservation community in the country. We as researchers of this institute, we were just young researchers. And the most exposure we had was to our faculty. But during the annual research seminar, we got opportunities to meet the field managers, the senior managers, the top conservationists in the country who had actually shaped the law, the direction in which the conservation in the country was going and is still going. And therefore, for us, it was something which was the most sacrosanct to be able to present during the ARS. And it was something for which each researcher prepared from day one. The day we finished one seminar, we immediately started thinking that, all right, this is my work for the next season, and this is what I'm going to present for the next annual research seminar, because it was so important. And it was also there, where all our committee members would be there, they would provide their inputs into our presentations and try to make them relevant to management. And wherever this gap existed, this was the place where we tried to deliberate upon it, where we discussed about it, and where we made the midterm corrections to our work that was going on. This annual research seminar was also a platform from where the young researchers were selected and groomed to become the future faculty of this institute. Let me tell you that. And the grooming started from there. And therefore, from that time only, this seminar has been taken up as a platform to discuss our research that is going on for all of us, because there is so much work going on. We really don't know a lot about what each one of us is doing. So this platform gives an in-depth insight into what the different research projects of the institute are doing. It is an opportunity to discuss our methodologies, our approaches, and also certain issues which we have in the field, and actually to do those corrections. It is also the time when we are grooming the fresh master students and the fresh researchers to be able to make the presentations from this platform. And believe me, I was only the second batch of researchers when I started making my presentation in the annual research seminar. And my experience was that it was less scary, and Dr. Satyakumar is smiling at me because we were together, and it was less scary to present in an international platform than it was to present at the annual research seminar of the Wildlife Institute of India. Because you could not get away with a single mistake or a single wrong thing that you have done in your methodology, in your analysis, or in your presentation, including even the size of font that were projected here. So everything was corrected. That is how the researchers and some of the faculty here were groomed over the years in this institute. And that is what we would like this seminar continue to do. And as the scope increases, as the number of students increase, we realize that it is not possible to have all the presentations during the annual research seminar where a lot of people from outside are coming. And they may not have the time to be present for all the days. And so we divided it, presumably 17 years ago, into, or maybe 18, because probably one seminar we missed during COVID, into internal research seminar and annual research seminar. So as you know, internal is largely for the short-term projects, which are just initiated, or projects who do not have a lot of data to present, or the beginners basically. And the annual research seminar is where we need to discuss with the different people, the managers, about the course that the research of the institute is taking. Each is equally important, each is equally sacrosanct, and in the seriousness with which the faculty is going to look at your work, is going to provide inputs into your work, is the same. So I hope you are as excited about this research seminar and as nervous in presenting in the seminar as we were maybe 30 years ago, or maybe 30 years ago or more. So I hope it is still there. You are getting jitters when you come up to this dais to present, but when you come up here, you become very confident that this is my work. I have worked hard, and nobody knows it better than I do, and I will be doing my best. And I will be accepting all the criticism that comes my way. And next year I will do better. And I will win the prize. For us it was a huge thing. And for you it should be a huge thing. Because there is no platform in the world which is as thorough as this one. Believe me, I've been to all. Most of my colleagues have been there. And they will still tell you that this is the one which will peel out every day. And there is no other like this. So I think we are all extremely lucky to be here today, to be able to listen and present whatever we are doing. And I wish you all the best. And I wish you all the enthusiasm and good learning for this internal annual research seminar. Thank you. Have a wonderful time.
Thank you, madam. I presume very serious. Last 4-5 days, I have been saying till 11 o'clock, 12 o'clock. The rooms inside, the lights inside the room are on. So I presume all of them are working hard and taking it as seriously as you were taking in your days. Those days when I joined the Wildlife Institute, there were having 30 research scholars. We have a listed research scholar of 534 as of today. So the institute has grown leaps and bounds. And in such a large crowd, managing everything becomes, I won't say extremely difficult, because it's a tough job. So the institute administration is working very hard towards making our life comfortable. And Dr. Satyakumar has been heading the administration for the last 1 year. I welcome, I request Dr. to deliver Please address to all.
Thank you, Dr. Vilas. To remain tonight director, Dr. Dehradun, Vilas Pandey, Dr. Sir, Director Dean, Chief Dr. Ahmed, Dr. Anthony, online faculty colleagues, researchers, students, diploma trainees, and friends. It's a privilege to be here. As Dr. Michi just said, 35 years ago, both Dr. Michi and myself had made presentations in the annual research seminar, which was in a small room in the Australia College campus. And when we moved here, we did annual research seminar in two halls. One was the library floor today, and the other in the first floor of library. And in 2003 onwards, we have been doing the annual research seminar here. Over the time, presentations increased, and we had to have this internal annual research seminar, as well as we have had seminars for only Himalayan projects, and then one Kappa Himalayan projects, and so on and so forth. So we have grown in by leaps and bounds in the last 25 years, from a handful of researchers to over 650, 700 researchers. The idea of the annual research seminar or this internal annual research seminar is that all our projects then were funded by the government mandate. Today we have funding from various other resources. The idea is to review the projects, what progress they have made by the bank members, and so this is one opportunity for all of us to know what project is in this stage and how much the project has progressed in trying to achieve the objectives. And this also gives an opportunity for all the researchers to make a presentation, to get an idea about how to make presentations and make the presentation within that specified time and also define their work well. And this review is a very rigorous exercise, as Dr. already mentioned, and we are doing this regularly. And this will also help us to know how mid-course corrections can be made in our projects, and also for the students who need to make those necessary corrections in their projects. So I wish all of you a very, very happy learning, and I look forward to all interesting interactions in the course of the next 2.5 days. Thank you very much.
Now I request Student Director of India to address you.
Good morning to all. Sitting on the dais, Dr. Dehradun, Faculty of Life Sciences, Dr. Satyakumar, Dr. Vilas Pandey, Research Coordinator, all my faculty colleagues, officer trainings of diploma course, all nursing students, all researchers, and all other officers and employees of Dr. The outset, I welcome Dr. U. Savak for kindly agreeing to chair the 17th IRS and for being with us and guiding all our research scholars in the course of this seminar. I also express my sincere thanks to Dr. Kushinaran, Professor Sherry at University of Agriculture, Science and Technology, Srinagar, Dr. Manjari Jain of ISER Mohali, and Dr. Raman Kumar for agreeing to be with us during this IRS. As already Dr. Badola and Dr. Satyakumar mentioned about the origin of IRS back in 2005. And the purpose this was to give an exposure to our researchers, to make presentation and also to present the projects which are in the initial stages or it is just a complete presentation. Well, this is the 17th annual international research seminar, and 50 presentations will be made during this course, and out of that, 2 is by our faculty, one from WI and one from WSFR. And these seminars will continue for 2.5 days. Now, talking about some of our achievements during last financial year regarding research. So last year we completed 22 projects in WII and 17 in SECON. And the ongoing projects in Wildlife Institute is 84 and that in SECON is 29. So this is the volume of projects which are ongoing, and subsequently last 6 months we must have added many such projects. Last year we published 320 articles and research publication in our index, peer-reviewed index journals. The highest impact factor we got was around 56, and 20 of our publications got impact factor of more than 5. About 300 were from 1 to 5 category. Our publications contributed to many SDG goals, as many as 8, primarily to life on land, climate change, and life below water, and even others. So our analysis shows that many countries are using WI research publications in their policy making. Some of the countries which are using our documents for their policy documents are Italy, Switzerland, United States of America, Kenya, Luxembourg, UK, Netherlands, Sweden, France, Chile, Tanzania, and Thailand. During last 1 year, our scientists discovered or rediscovered some of these species, as many as 13 amphibian species were discovered, entomofauna 2 numbers, and even 1 plant species was discovered by our scientists. We have also, in the last year, we have almost established our next generation sequencing facility, likely to be inaugurated by Honorable Minister during inauguration of ARS. We also propose to establish an interpretation center from Wildlife Museum under CSR. We have developed a proposal and we are likely to get support from ONGC for it. Last year we launched our Journal of Wildlife Science, and already 2 editions have come out. And our publications got a lot of media coverage, social media as well as the print media. 208 of our publications were reported in news, 17 by blogs, and on X, roughly around 2,500 of our publications were reported, and of course, few on Facebook as well as Wikipedia. Now, with our institute, we are trying to constantly improve the quality of our research and also improve the facilities we are providing to the researchers. You are aware that we have already initiated the travel grant program for researchers, both those who are doing their PhD and those who are otherwise working in the project. I'm very happy to say that already I think a meeting happened maybe 2 days back under chairmanship of Dr. Ruchi Badola Dean and 3 researchers who are doing PhD have been selected for the first batch of program. We have also started teaching assistantship. So one issue which was raised that when our researchers or our students apply outside for teaching job, they do not have any certificate to show their experience of teaching. So this initiative was taken, and all this we are doing from our own fund, the fund which we are generating and keeping in our revolving fund or in control. We are also, I am happy to tell you that we had been planning to upgrade our library through CSR. We have got a 4 crore rupee sanction for upgradation, and we have got the first installment of 1 crore already received in our accounts, and very soon the work of upgradation of library will also begin. Also, I'm happy to inform that the government of India has approved construction of a new hostel, and they will be providing funds in phases. So maybe the next year, the construction of next new hostel also may begin. I will be very keen to receive your constructive feedback on further improving the quality of research and the quality of facilities which are being provided to you by the institute. And finally, on the Himalaya Divas, celebrated every year on 9th of September, which is today, I extend a hearty welcome to all of you. As you will notice in the program, that the first session of this seminar pertains to our research on the Himalayas. I look forward to telling all of you and wish you a great seminar. Thank you, Jai Hind.
Thank you, sir. So that brings us to the end of this another session of something. And we will talk on time. And we will start with our first session. I welcome all of you once again. Today is the Himalayan Divas. So we will start our first session on the Himalayas. And I request Dr. S. Sajidkumar, Scientist Chief and Register, to chair the session. I request Dr. Amit Kumar, come over and co-chair the session. And I request Dr. Devi Gupta to come on with that and direct the session, run the session and the session.
Good morning once again. We begin our first session on the sustaining of the Himalayan ecosystems. So today is Himalayan Day, so wishing all a very happy Himalayan Day. I would like to introduce my co-chair, Dr. Amit Kumar, who is scientist D, and he is also the guy who works in the Himalayan and has done a lot of work on plants. And to my left is Dr. Vivek Dubey. He works on the aquatic ecology, particularly Himalayan rivers, where he's going to be the session facilitator. So we have 9 presentations in this Himalayan ecosystem session. And before I request the session to invite and introduce the speakers, I would like to say a few things. I request all the students, researchers who are sitting behind to please come forward and occupy some of those empty chairs here. Please, on the right here. Please come. Okay. As you are all aware, the Himalayan mountain ecosystems are one of the most fragile and highly well recognized for their biodiversity, ecological, environmental, hydrological, socio-cultural, and aesthetic values. But these values are seriously threatened because of anthropogenic pressures, climate change, and also uh, the natural processes that takes place and are impacted by development projects there. Uh, this particular session will be focusing on some of the projects that are happening in the Himalayan region. One project is on the National Mission for Sustainable Himalayan Ecosystem, which is a task force of WIM under the DST. And also we have some presentations on endangered species like Dehradun and also on the on the aspiration in the Himalayan region. So without taking much time, I will request Dr. Vinay Dubey, the session coordinator, to invite the speakers. First speaker is Dr. Sandesh Thakur. She is going to make a presentation on spatial temporal gradients in land distribution, patterns in the multi-use landscape of Kamen, Arunachal Pradesh, Eastern Himalaya.
Ms. Shagman comes from Hamirpur district of Himachal Pradesh. She did her master's in geology from DAV Punjab University. Subsequently, she joined the NFC project and worked on mammals in GSMP and biodiversity. Currently, she is pursuing PhD, researching on the mammalian community in the West Kamen region of Himachal Pradesh. In her free time, she enjoys hiking and exploring new characters. So, over to Shagman.
Very good morning to all of you, and I would like to say a few very happy also to all of you. So I'll begin this session with a brief introduction on I would like to begin this session with a brief introduction on the project mentioned, which is the National Mission of Sustainable Himalayan Ecosystem. It is one of the 8 missions under the National Action Plan on Climate Change and NDC. This project aims to understand the complex processes affected the Himalayan ecosystem due to climate change, long-term potential monitoring, and developing long-term ecological monitoring and developing mitigation strategies to safeguard the Indian Himalayan species, ecosystems and communities are major objectives of this project. So, Government of India and Department of Science and Technology will given the responsibility to coordinate and implement this project, and it basically established 6 task forces, of which task force 4 was included by the Institute of India. Biotech Institute of India started this project as the initial phase 1 5-year project in 2015, and it coming as a cellular and coming to my research topic of today, which is I am discussing space and time used by mammals in the variety of landscape of Arunachal Pradesh waste farming. The objective that I am discussing, I am going to discuss with you is to understand species richness in collective conservation mammals, to assess the distribution patterns of selected mammals, space of their responses to climatic factors, and to understand the temporal activity overlap patterns with anthropogenic disturbances. Coming back to the study site, West Tamil lies in the western part of Arunachal. It is connected wilderness wildlife sanctuary and starting wildlife sanctuary, which is in Ladakh. So, West Tamil lies in Arunachal lies in the Himalayan band of the zone of Eastern India, and the West Tamil has a basal area of around 7,400 square kilometers, which comprises of the elevation range as low as 300 meters and high as 6,000 meters elevation. The major forest sites are temperate broadleaf, temperate conifer, and alpine streams. There are 5 major types in West Tamil, namely Montana, Rishi, Shepherd type, Alba, and Ruby types. With the major being of Dehradun are exotism and vegetarian fiber. West Tamil also is a beautiful site of So the research methodology which I opted for my study is the intensive grid-based sampling, which I followed by to grid sample size for this study. I used 92 cameras with around 15,000 peptides for analysis. The sampling period was from December 2022 to April 2024 from 91 species. For the data analysis, I mentioned species richness and composition was assessed. Relative abundances were estimated, which is majorly the number of captures per guidelines. Generalized leader-rich models to Bayesian regression were used to study the response of species to biotic and abiotic factors and temporal activity overlap patterns of selected mammals were established overlap patterns. Based on the camera trap data, we found 32 mammal species as of now in the West Tamil basin. Belong to various orders like Carnivora, Arthropteryta, primates, Rodentia, and Lemurs. So here we see the elevation use patterns of mammals based on their core characteristics. First, let's look at the lower elevation code, which is usually dominated by species like the Montpellier, Biped, Golden. Moving to the mid elevation code, we majorly found species like Pumpkin, Muscular, Lepanda, and Moon. And then again, the highest elevation areas, about 440 meters, we found the frost, snow, and bucket dominated vegetation. Let's see very quickly done. You see here the majority of carnivores or mammals like birds, alligators, mountain cat are found in the elevation zone below 25 meters elevation, except for the leopard cat, which was important because the present elevation at highest. In this graph, we see the species which were majorly avoided above 4,040 meters elevation, such as red fox, snow leopard, and red sapphire. Similarly, for ungulate species, as we saw in the pie chart plot, this separation was quite apparent as species like were not reported above 3,200 meters. Species like U-shaped muscular market were not reported below 25 meters. Understanding the species distribution and habitat distribution for this animal is second objective. In our detailed animal, we use capture of species as a response variable, number of campaigns as a toxic to account for the variation in the effort to the size, and the abiotic features such as habitat and seasons, and anthropogenic pressures such as human presence and livestock presence as our predicted. Then we see the predicted relationship of alternative habitat and human habitation by Dehradun. Alternative distribution was mainly influenced by seasonal conditions, terrain, and human presence. It avoided rugged terrain. The relationship with human habitation shows initial increase, but it decreases as the distance to human settlements decreases, which can be attributed to the compounded effect of elevation, as we see here, the increasing elevation patterns in electricity distributions. Last year showed around 55% of overlap with humans and 10% overlap with livestock. However, we see here the bimodal response of clear to these questions that is avoiding the peak activity hours of human and livestock by majorly being active in the morning and evening. In Korea, we also observe similar patterns as we did in martin here. For weather-coated martin, we see significantly higher overlap with humans, around 74%, and with livestock, which is around 82%. For red fox, we see continual behavior, which is avoiding the human interactions and livestock interactions by engaging the activity integral periods of the life. But goal we didn't go for the overlap as we call it as structures. So we only observe the activity patterns when we see the peak time of goal activity is in the morning hours, in the afternoon we see a slight dip in the activity, and again during the evening hours we see a slight increase in the activity patterns. So the key findings of my study are the variety of the US photoreceptor species indicating its resilience to climate. Also, this coming holds as a potential site as presence of rare species such as spotted ginseng, pumpkin, photoreceptor have been performed on display. However, the preliminary findings showed no detection of large carnivores such as leopard and very few reports of HFT blackbears, which potentially indicates the probable location of local extinction from the area in the future. Our study also resembles with other studies conducted in coastal areas in Arunachal Pradesh, such as Nandala and Shimla, also highlights similar issues related concern over independent extinctions, especially of large carnivores. The major challenges and limitations which we occurred in conducting this study is that we do not include other species due to very low detections, and it was challenging to assessing the relationship with habitat and human activities. The inability to estimate animal densities reflect the challenges associated with animal excavation and wetland in tropical forests, where there are different species of inherently low densities. There is limited baseline data on rare species, which hinders in establishing trends or assess collaboration patterns. For the way forward, to expand the geographic coverage to factor variations by increasing sampling sites, we will also adapt alternate camera tracking methods such as arboreal camera tracking to capture semi-terrestrial species, and we will also assess the seasonal and global patterns of mammals with polar activities. We will be conducting proximate surveys with community conserved areas and comparative non-conserved community conserved areas to understand the nature of hunting practices. And last but not least, we will review establish standard criteria for selecting vulnerable and endangered species. I would like to acknowledge each and every member of NHU in the Wildlife Institute of India.
Thank you, Sharon. We take the questions later. We move on to the next speaker. Ms. Rita Kant will be making a presentation on occurrence and activity patterns of mammals and life forms in the Krishna Basin, East India. I will request the IT cell to please take the microphone before the next presentation.
This Rita completed her undergraduate and graduate studies in forestry and environmental science. She was associated with the WWF in India and Western India Private University. In September 2019, she joined the Wildlife Institute of India under this project. Since then, she has primarily worked in Himalayan region of Uttarakhand and Sikkim. Her main interests lie in ecology and behavior of animals. Over to Pooja.
Good morning, everyone. Thank you for the introduction. I'm Pooja and I'm associated in MNC Facebook project and I just now Dr. Satyawar. First of all, I would like to wish you all Dehradun, where we have confirmed 18 mammals of high global conservation significance, 5 endangered, 4 vulnerable, and 6 threatened species. As I said, Sikkim is a small state, but it has a huge diversity of wildlife. As a snow leopard, ivory, and southern yam, which is endemic to this region, and only breeding population of this area. Red panda, which is state animal, and duck pheasant, which is state bird of this area. Now coming to the methodology of my study, I did camera mapping in 4 into 4 kilometer square grids, and the elevation ranges from 500 to 5500 meter. The total 7,392 traffic minutes per sample with 108 cameras. The duration of my study was February 2023 to July 2024. We also assess the relative abundance with using camera tests per 100 trap nights, and also assess the activity pattern of mammals and diaphragms to see the temporal overlaps. These are some areas where we have done our camera trapping. So this time we have tried to cover protected areas as well as non-protected areas of Sikkim. The objective of my study is to estimate the occurrence and relative abundance of mammals and diaphragms. And the second one is to assess the temporal overlap of mammals and diaphragms. Coming to the result of my first objective, which is distribution of mammals across elevations. So we found that Asiatic black bear and dhole have the maximum elevation range of 4,000, while Asiatic black bear shows the lowest range of 1,500 to 4,000 meter. We have divided our elevation into altitude gradient, and we found that dhole have the maximum area at an elevation of 2,000 to 3,000 meter, followed by Asiatic black bear and common leopard. We also found that leopard cat, yellow throated marten, and red fox have the maximum elevation range of 4,000, while leopard cat shares the maximum elevation with the lowest elevation of 1,500 to 4,000 meter. The relative abundance index of carnivore shows that red fox has the maximum area at an elevation of 3,500 meter. And the ungulate distribution shows that ivory have the highest elevation at 5,000 meter, followed by Rushi, ivory, and southern Siam. And the lowest elevation was recorded of barking deer at an elevation of 1,500 to 3,000 meter. The RI of ungulate shows that Gural have the maximum RI at an elevation of 25 to 3,500 meter. Coming to the next slide, which shows the distribution of galleyforms, and the Satire Kabutar and Himalayan Munal have the maximum distribution at 4,000 meter, and Khalif pheasant have the lowest elevation of 1,500 to 3,000 meter. The relative abundance of galleyforms shows that pheasant have the maximum area at 25 to 3,500 meter, followed by Himalayan Munal and Satire Kabutar. These are some captures we got in our cameras, and we found that maximum species were captured in 25 to 3,500 meter. Coming to the next objective of my study, which is activity patterns of mammals and fishes. We assess the activity patterns of species among species, and we found that Asiatic black bear and dhole has the maximum overlapping, followed by golden cat and golden jackal. Musk deer and sparrow also share the maximum overlapping. As Sikkim has the highest diversity, but it has some challenges and limitations. As you can see in my background you can see like how they are sorry, the garbage in such beautiful landscape, and although wildlife are also shared in this landscape, and the insects are feral are using this landscape, and they are also feeding in this garbage areas, because it's very easy for them to feed. And we also got this one is a camera picture where they are living in such harsh conditions. They are also sharing the habitat with the snow leopard and pet resources, as well as they are sharing the habitat of Albany. This picture is from the cold desert area of North Sikkim, and the insects they are predating the livestock like Ina, red panda, sero. And according to the study of Jaipur, 40% of the young cows are Mukang are killed by feral dogs only. So after considering all these issues, we have checked the overlapping of feral dogs with wildlife, and we found that it has maximum overlap with red panda, talking deer, Sitae, and livestock. We have also did capacity building workshop with the Indian Army, and we sensitized them about the proper dispose of garbage. We also did some capacity building with forest department and trained them some tools and techniques of camera tracking and science survey. Now, I would like to show you that we got the highest elevation record of private tiger at an elevation of 3900 meter and got from 3500 meter. Previously, it was recorded from the boundary of Arunachal Pradesh and got from Nagaland. We also got the highest elevation record of Indian peacock at an elevation of 2900 meter from eastern Himalaya. We got the first capture of common palm civet and spotted lizard from Tamilaka Wildlife Sanctuary. Although it was recorded from Western Rhododendron Sanctuary, but it's a first record for the Tamilaka. Coming to the summary findings, the study confirmed the presence of 35 mammal species, 5 deniforms, including 6 vulnerable and 5 endangered. The maximum capture of Asiatic bagbear, barking deer, and black pheasant were captured, and the temporal overlap patterns reveal that mammals were predominantly diurnal. Coming to the way forward, more scale-defined analysis is required to understand the co-occurrence of mammals and deniforms in Sikkim. More intensive temporal sampling to understand the seasonal movement pattern, and better to understand the habitat use of mammals by collecting data on different covariates, and to study the impact of climate change by deploying ecological niche model, understand the future and climatic scenarios, and identify critical future refugia for vulnerable species. Now, I would like to acknowledge foreign people. Thank you.
We move on to the next presentation by Mr. Himanshu. He will be making a presentation on diversity and assembly structure of fish in the West Common River Basin, Arunachal Pradesh.
Himanshu holds a master's degree in environmental sciences. He is working as a junior project fellow on the MC project, and he recently joined the ACSR PhD program. He loves hiking and fishing. Over to Himanshu.
So, freshwater ecosystems cover less than 1% of Earth's surface, but supports 48% of all of the species present across the world. Provides us with clean water, food, and economic resources. But we are exploiting our rivers by sand mining and stone mining, polluting them with industrial, agricultural, and household runoffs, damming and disrupting the ecosystem and altering the water flow regime, impacting the species distribution and moving them towards species extinction. All these environmental variables along with climate change are changing the river flowing ecosystem and moving them towards extinction. So understanding its environmental impacts on fish diversity and dynamics is a preliminary step to predict a response for future changes we will be facing and supporting an ecosystem-based management. Good morning everyone, I am Mansoor and I will be presenting on the diversity and assembly structure of fish in the western river basin of Himachal Pradesh. This is the study area. We have surveyed 4 rivers till now in the West Tamil basin. It is Dehradun River, Shetland River, Tenga River, and Afra River. We have sampled around 280 points. Coming to my objectives, the first objective is to evaluate the fish diversity, abundance, and the distribution patterns in the West Tamil basin. To evaluate the assembly structure of fish in relation to environmental parameters. To analyze the linkages between fish species and associated functional traits. And to evaluate the current anthropogenic trends on the fish communities in the West Tamil basin. Coming to methods, we survey larger order activities every 500 meters and smaller order activities every 200 meters. In each sampling point, we divide the river segment into 3 segments and take our environmental variables and other parameters in every 50-meter range. Sampling was done from November 2022 to May 2023 and November 2022 to June 2024. We used cast nets for fish sampling, which is 1 into 1 centimeter mesh size, and drag nets and tube nets. Water quality parameters were recorded by using bias and multi-parameter kits. Morphometric and realistic data were recorded on field itself. Unidentified samples were preserved in 10% formalin. And further, the identification of fish species were done in wildlife biology lab by following various taxonomic keys. For fish abundance, we have used correspondence analysis. And to link the fish abundance with environmental variables, we have used principal component analysis. And to look how rates are impacting the fishes, we have used gain and speed analysis. We have also evaluated catch per unit depending on the biomass of the fish, which is weight of fish caught divided by effort, that is 10 hours and number of casts. These are the environmental parameters we have collected. For physical parameters, we have collected pH, water temperature, total dissolved solids, electrical conductivity, and withdrawal oxygen. For microhabitat, we have collected flow rate and water depth by using the flow. Total river weight can be divided by range finder. Mesohabitat, we have collected cascades, rapids, runs, rivers, and pools. And for dominant substrate, we have collected bedrock, boulder, pebbles, sand, silt, and clay. Coming to the fish diversity in the West Tamil basin, we have identified 16 species. Followed by several foresters, large cities and good communities. And the family, simply is the most dominant family, followed by Sisovia and mortality. Looking at science species abundance, we have identified few areas which have higher abundance. And whenever you cast net in these areas, there is a possibility you will get higher catch of fishes rather than other areas in the basin. Numerous external represents water materials, they are found from an elevation band of 1200 meter above sea level till 400 or 300 meter above sea level. And this group is a warm water group, Dehradun, Chana, Mandela, Australia's Penguins, Dehradun, Mandela, and Sri Lanka's Arunachal. They are warm water features and found in the lower elevation of the basin. And they are almost also found in the Brahmaputra basin in the Assam region. You see, you can see that the Nafra river is distinctively different from other rivers in the study area. The Nafra river is influenced by the dissolved oxygen, bedrocks and boulders, mostly our dominant substrate variables. This is because of maybe the habitat heterogeneity we have in the Nafra region, and there's so many small streams there which is draining into the main Nafra river, which is also increasing the dissolved oxygen parameter in the Nafra region. Rest, all other rivers have no such influence of other variables. And we see through fish composition, we can see that Pseudokinesis sulcata is referring the Nafra river in the Nafra region. Other fishes are almost uniformly distributed across the basin. We did a hierarchical cluster of fish assemblages, and we can cluster in two groups. One is cold water group consisting of 6 species. One is warm water group consisting of 11 species. These two sub-assemblies were identified as Dehradun Shedon sub-assemblies and Nafra Tenga sub-assemblies. Dehradun Shedon sub-assemblies is governed by flow, altitude, and water depth, and Nafra Tenga is mostly influenced by temperature, wind, river, with electrical conductivity. Coming to the traits, we will Smith analysis on the traits. We have selected 5 traits: length at first maturity, maximum length, age at first maturity, lifespan of good habit. So here you can see the maximum length and maturity, first maturity are influencing cytotoxic system and all these fishes are big size fish, and they tend to have maximum larger length and larger length and maturity, which is opposite to cytotoxic in the opposite axis. Pseudomonas formulary and other fishes, they are actually in-stream roaches or sucker fishes, we say. They are very small in size. They don't attain length and maturity at a very small length. That is why they are not influenced by maximum length and maturity. Now we can also look at the food habit. Pseudomonas sulcata and Dehradun analysis. They are also influenced by food habit. They are omnivorous. Opposite to that axis, Pseudomonas sulcata and Dehradun are also in large. They are mostly carnivores, and they are limited influence by food habit. So they are two different groups of different food habits. Coming to next on fish communities, we have categorized all our fishes into additional categories, that is data deficient, endangered, risk concern, not evaluated, ineffective, and vulnerable. And the threats we have collected on field itself, we have collected different threat categories in each sampling location, that is agricultural runoff, human settlement, illegal fishing, mining, river erosion, and waste dumping. As you can see, all the fish species are more or less taken by all the 6 categories, but we will be talking about what would be that is golden marshal, new segments as well as started marshal, and this saturated settlement. So, what would be is mostly influenced by river operation, human settlement, and agricultural runoff. New segments example which is influenced by illegal fishing, human settlement, mining, and waste. Illegal fishing because numerous sectors example structural marshes grow into large cycles and people used to fish for that numerous sectors example for their food sources. And our group cytotoxic system is influenced by all the threats in the form of the study area. Coming to my conclusion, a total of 16 species have been recorded from the West African. There are given as a high abundance followed by Tenga, Shedon, and Nafra. Environmental variables such as altitude, water depth, flow, and slope has positive influence on the cold water fish lake, results on review and temperature, wind, river, and electrical conductivity are positively influencing warm water fish like Dara, and two distinct subassemblies were identified in the basin, cold water and warm water. Dehradun and Shedon have one assemblage, Nafra and Tenga have one assemblage. The functional rates like maximum length and length was maturity was found to be associated with large size fish like Sagittarius, results on and dumping of waste materials was found to be the most common way to all the species recorded in this community. Going forward, more intensive sampling will be conducted for assessing the diversity structure of the waste community. Seasonal sampling is undergoing to establish the baseline data on breeding patterns and water temperature. And climate change scenarios of this distribution will be analyzed under differentiated socioeconomic pathways using primary and meteorological variables. I can acknowledge this paper. Thank you.
Thank you. I'll invite Mr. Meghalaya to go for presentation. Characterizing macro-invertebrate communities, species, environment relationships, and trade dynamics. The best common basin is
Meghavi studied masters in zoology from Maharaja Dehradun University of Dehradun. She has recently enrolled a PhD in Dubai and working under an internship project base. Her interests include entomology and exploring new places. Over to Megha.
Thank you, sir, for the introduction. Um, the mighty Eastern Himalaya. This region is well known for its snow-clad peaks and deep spiritual significance. But beyond the stunning landscape, there is a hidden world lying beneath the fast-flowing stream of Arunachal Pradesh. The tiny macroinvertebrates. These are the nymphal and larval stages of aquatic insects. These insects are playing key role in nutrient cycling, trophic relations, and also keeping the stream health intact. Macroinvertebrates are further divided into different functional feeding groups. These functional feeding groups are divided based upon how they eat and what they eat. All the functional feeding group plays a key role in the nutrient cycling and food chain. Starting with the shredders in the headwaters, they break down the leaf and convert the coarse particulate organic matter into the fine particulate organic matter downstream, where the collectors and grazers feeds upon them and they transport them towards the downstream where the filterers, they increase the nutrient flow by feeding into the FPOM. Macroinvertebrates are basically like engineers of the stream. Without them, the stream integrity will collapse. So, maybe our leaf shedding engineers are not good at spelling, but when it comes with the making water clear, they are always right at it. From being a crucial link to the nutrient cycling and food for the bird species, even the only alpine crane species like black-necked crane feeds upon macroinvertebrates, and even otters also leave the traces of macroinvertebrates in their sprains sometimes. But still, we know so much less about it. So let's dive into the world of macroinvertebrates. Good morning, everyone. My name is Meghavi Purohit, and today I will be talking about diverse world of macroinvertebrate, characterizing their communities, species-environment relationships, and trade dynamics in the West Kameng Basin of Arunachal Pradesh. We aim to investigate the diversity and community structure of macroinvertebrate in relation to the environmental parameters. Further, we also want to assess functional traits of macroinvertebrate community with regards to the environmental variables and what are the key environmental variables which are driving the diversity and community structures. As you are already aware of the study area through the previous talk, my study was also conducted in the West Kameng Basin of Arunachal Pradesh. We have so far considered 60 sampling points from the Dirang River, and we did analysis according to these sampling points only. Well, first to look for the macroinvertebrates, we need to target certain mesohabitat and substrates. Riffle areas, cascade, and the areas with high leaf litter and pebbles are the ideal ones where the macroinvertebrates are found in a larger numbers. We also assess other mesohabitats as well. After targeting the specific substrate and mesohabitat, we strategize our field sampling techniques. We conducted sampling at 500-meter distance from between the 2 points in the larger order tributaries, and in the smaller order tributaries we consider 200-meter gaps. We took samples of environmental variables from each point, and we did kick netting and handpicking method for the sampling of macroinvertebrates. Well, streams of Arunachal are not predictable all the time. Sometimes we even use sieve for collecting the macroinvertebrates. Talking about alternative methods, I would like to highlight an incident. During my field work, I also met this woman. Her name was Nepong, but she would like to call herself as Rachipuki Buari. She was basically collecting macroinvertebrates for preparing the traditional meal using the traditional equipments. And it was so fascinating to see that how traditional practices are linked with scientific methods through the ecosystem we study upon. We then took our samples to the laboratory for the taxonomic identification. Out of 2,200 samples, we identified 71 families and 8 orders, and further we divided them into the 6 functional feeding groups. We wanted to check how environment is influencing the composition of community structure of macroinvertebrates, and we incorporated substrate characteristics, mesohabitat, topographic and physicochemical characteristics in our study. Further, we correlated these environmental variables and filtered them out, and we transformed them to standardize the explanatory and response variable data to do the redundancy analysis. We then plot RDA, and we projected our communities in the environmental space, and we also checked with ANOVA that how our model is fitting. We observed our data through the assembly process, where the local communities grows over the time, and the community structure also changes through the biotic and abiotic filtering. And species trait also plays role in filtering of abiotic and biotic factors. We also incorporated occurrence, environment, phylogeny, and trait matrices, and we did hierarchical modeling for species community for understanding the trait environment dynamics. In joint species distribution model, we first prepared our data and prepared all the metrics. We checked how our models are fitting. Then we did MCMC sampling, and we checked for the MCMC convergence through the effective sampling sites for all the parameters. We further evaluated our predictory and explanatory variables through the cross-validation and other strategies, and also we visualize our predicted variations over different gradients, that is environment-species relationships. In the result, our result highlights the highest composition at 1700 to 2000 meter elevation in Arunachal Pradesh, where the family composition were higher. We also highlights the generalist trend shown by family Betidae, Heptagenidae and Hydropsychidae, whereas Cephalonuridae and Simulidae showed a very specific trends towards the distribution. After incorporating all the environmental variables, we checked for the correlation, and we removed temperature and TDS from the environmental variables, and we then visualized the distribution of macroinvertebrate communities in the environmental space. Stream orders, boulder, flow, altitude, pebbles, and slope. They were distributed in a higher loading in the RDA space, starting with the first combination, that is Ephemeridae and Brachycentridae families. These families showed positive response toward the higher order streams, and also they favored the areas which are filled with high DO and boulders. Whereas Simulidae showed the negative relation towards the DO, and it favored more silt like areas because it acts as a filteral functional feeding group in the ecosystem. Same with the Chironomidae. It showed the positive relation with the pH. Betidae is showing positive relation towards the flow because by its nature it is a rheophilic family, whereas flow and riffles are in the opposite axis. stoneflies, which are the Perlidae family, they are favoring the riffle habitats rather than the flow-filled habitats. And we also observed that how Hydropsychidae is preferring the slope and the areas with more pebbles. Now we also wanted to see that how each family is responding towards the each environmental variables, and we also incorporated also starting with the Ephemeroptera order. Here we see that the physicochemical factors are governing more towards the distribution of Ephemeroptera rather than the mesohabitat characteristics. Whereas the Trichoptera, Diptera and Megaloptera, they are showing that negative relation with the clay, that is a substrate, but they are showing the positive relationship towards the other substrate characteristics. And the specific example of the Cephinidae from the order Coleoptera, it showed only positive association towards the riparian vegetation and negative towards the other variables. We also wanted to check how the functional feeding groups that we already discussed before are linking with the environment. And here we got the result, like the scrapers and collectors are being very specific. Scrapers only favor the physicochemical gradients, whereas collectors are negatively correlated and they are highly negatively correlated with the mesohabitat. Pearsons being generalist, it preferred water depth, substrate and mesohabitat, all three characteristics. Predators, they favored physicochemical and mesohabitat characteristics only. Among all the environmental variables, we wanted to check key and focal environmental variables which are driving the community structure. And out of all, the mesohabitat gradients are followed by topographic gradients are the ones which are driving the distribution and community structure. This outcome is a very important outcome of the study, as we already began our study with where to look for the macroinvertebrate. We considered certain mesohabitat and substrate characteristics. So topographic gradients, if we look into it, so collector, they are increasing as the stream orders are also increasing. As we discussed before, CPOM is transported towards the downwards, where the collectors and grazers are feeding upon it. Similarly, shredders are declining as the stream orders are increasing. That means shredders are only presented into the headwater streams. Coming down to the altitude. Altitude is also showing the similar relation as a stream order. Shredders are lesser in lower altitude and higher order stream, whereas filterers, they are lesser in the higher altitude stream because they are actually at the base of the river. Coming here to the substrate gradient, where we also looked into the relation between pebbles and sand. So pebbles and sand both are negatively related with the collectors. Collectors were less where the high number of pebbles and sands were there. Whereas, because collectors are those which are preferring more of the cobble and boulder kind of a structure. Also, the shredders and predators, they show positive relation towards the pebbles and sand, they increase towards it. It shows that what we have already observed in field is reflected by our data. And in conclusion, we so far identify 71 macroinvertebrate families from the West Kameng River, and altitude, stream order, flow and slope identified as a dominant factors influencing community organization of macroinvertebrates. Functional feeding groups showed positive and negative response toward the environmental data. Piercers and predators group showed positive response toward mesohabitat and substrate, whereas shredders showed more inclined towards topographic and physicochemical gradients. Freshwater substrate characteristics had a stronger influence on the distribution assembly of functional feeding groups than the topography features. Macroinvertebrates are having very narrow thermal niches, and they are used as a biomonitoring tool whenever it comes to the stream health monitoring. But they are more than that also. They can answer some big questions as well. And with the aim of that, we are aiming towards more intensive sampling in the selected western and eastern Himalayan river and identification up to general level for the selected families as well. Community turnovers of the freshwater macroinvertebrates will be assessed across the altitudinal and latitudinal gradients of the Indian Himalayan region, with the highlighting the ecotones. The impact of climate change in governing the distribution of selected macroinvertebrate communities under different emission scenarios will be assessed. I would like to acknowledge all of these, without which this study would not be completed.
Thank you everyone and wishing you all very happy Himalaya.
Thank you, Nagari. Next speaker is Dr. Priyanka Kashyap. She will be talking about soil mineral community structure and its associates with soil physico-chemical determinants along the elevation gradient in West Kano district of Arunachal Pradesh. Dr. Priyanka is MSc in Biotech and PhD in Wildlife Sciences. She is currently working as a senior project for the state and she working on climate change impact on soil ecosystem of high altitude and climate change. Over to Dr. Priyanka.
Thank you so much for the introduction, sir. A very good morning to all of you. So today I'm going to talk about the community structure of soil nematodes and its association. Am I audible now? Okay. So I'm going to talk about the community structure of soil nematodes and its association with the adaptive factors along the elevation gradient of West Kameng district of Arunachal state. So my talk is organized into these particular subheading, and before heading on to my methodology and result, I'd like to brief you about the soil and ecological variables and why we are studying nematode as an indicator for the soil health status of any region. So as you can see in this picture, a small portion or a handful of soil act as a landscape for soil microflora and fauna, and soil microorganisms play an important role in functioning of soil ecological processes. This can be understood by this particular diagram, where soil biodiversity observation network showed a link between the essential biodiversity variable, which is the outer circle, linked, or you can say they are prioritized by the soil biome, with the policy sector, which is in the center, through the working of the soil indicators, which is the inner circle of the diagram. So here you can see the taxonomic diversity and functional diversity showing a worm-like structure. Those are nematode, which is included as an environmental variable, or you can say essential biodiversity variable for the soil. Why they are important? Because they take up several trophic levels occurring in the soil food web and also correlate well with the ecological processes. You can see they are present at the first, second, third trophic level, where they are, you can say, categorized based on their feeding habit. So the bacterial and fungal feeders, then we have herbivores, which are plant feeders, and then we have omnivores and predators at the higher trophic level. And they play important functions in the soil processes, for example, organic matter turnover, nutrient transfer, soil structure improvement, disease transmission, and prevention. So the stability of performance of any ecological function is dependent upon the stability of the soil food web. So that's what we are trying to do by using nematode as an indicator. So the current study area is basically data deficient when it comes to the nematode community structure. So we are trying to bridge this gap by documenting the community structure of nematode. So the study area, we have sampled from 1500 meter to 4000, starting from Dirang to Sela Pass. So what we have done, we have categorized into the 5 classes of 500 meter elevation. And we tried, we have done the stratified random sampling in each of the elevation class. So after collecting the sample from these particular stratas, we marked it, sealed it, and transferred it to the WII headquarters, where we divided these samples into 2 parts, one for the biological analysis and second one for the soil physiochemical analysis. So coming on to the biological analysis, we have extracted and isolated the nematodes using the Cobb sieving and decantation method. Then we fixed into the formaldehyde solution, later on dehydrated in the desiccator for preparation of the slides using ethanol and glycerol, and then they are identified up to the generic level for further analysis of the community structure. So what we are trying to do is we are using the nematode diversity transformed into the nematode specific indices, which gives you the information about the soil health status of the particular region. And that can be useful with the long-term monitoring of the soil of that area. So this slide is just to show you what are the instruments we required for the preparation of slide and nematode identification. So we have a slide warming table, then we have a microscope, and you are seeing the slide box, and this is how slides are prepared and preserved for further identification. So for the second part, soil physicochemical property analysis, we have analyzed for the reaction, the soil concentration, soil organic carbon, nitrogen, phosphorus, and soil moisture. They were correlated with the various soil nematode trophic groups. So the analysis that we have done is we have analyzed various nematode-specific indices. For example, enrichment index, structural index, maturity index. So they basically provide you, or they depict the decomposition pathway, nutrient flow, and the level of disturbance that is happening in the soil of that region. Then we have what we have done for this calculation of these indices, we have divided the nematode genera into the colonizer and persistent values, ranging from 1 to 5. 1 is for the enrichment colonizer, then the disturbance colonizer, that is CP2, which are mostly bacteriovores, and then the persister, which are mainly omnivores and predators. Then we have assigned to these CP groups based on the Bonger 1990 literature. Then Shannon diversity index was also calculated. Then central principal component analysis was performed to compare the nematode composition with the soil variables. Non-metric multidimensional scaling was performed to show nematode community position and investigate the community structure along the elevational gradient. Then we have also performed the Permanova, which is used to test if community are significantly differ for the elevational classes. And these analyses were performed in R, and pairwise multi-level comparison of nematode composition among elevation classes was also done to see the difference which genera differs among the elevation. So moving on to the result, through the correlation analysis, we have found that bacteriovores, fungivores, and herbivores show a positive correlation with the moisture and organic carbon and negative with the elevational. Then the adaptive parameters along the elevation gradient, we have found pH was moderately acidic starting from 1500 to 4000, which could be due to the leaching out of the basic nutrients and also the accumulation of organic acid, which is equivalent to the humic acid. Then next is a salt concentration. It decreased along the elevation. Organic carbon was higher in the middle elevation and the lower elevation. Same for the moisture, nitrogen, and phosphorus didn't show any significant relationship. Moving on to the nematode trophic group along the elevation gradient, we have bacteriovores nematode highest number at the middle elevation. So these are the preliminary results that I'm showing to you. Then fungivores was decreased along the elevation, which can be conferred by the enrichment indices, which I'll show you later on. And then we have total nematode abundance was significantly differ among the different elevational classes. MI, this is the maturity index, which shows the disturbance level. So higher the value, lower will be the disturbance, lower the value, higher will be the disturbance. So we have moderately values up to 2.4, which is significant different between the elevational gradient. Then we have a channel index, which is usually same for everyone, and it shows the decomposition pathway in the region, which is a bacterial pathway. Then we have a Shannon diversity, highest in the lower elevational area and lower in the fourth elevational area. Then this is the mean value of bacteriovores nematode. We have found 24 genera of nematodes in the particular region. 18 out of that, 18 were highly significant. And we didn't find any turbitrix in the higher elevational region. Moving on to fungivore, we have six fungal feeding nematodes, out of which Axonium, Bacteria, and Tylencolemis was a significant difference among the classes. And Bacteria was absent in the higher elevation, and present in the lower one. In herbivorous, we didn't found any Merlinius in the higher elevational zone of 35 to 4,000 and 3,000 to 3,500 elevation. For omnivores, we have Doralemus. Doralemus was the highest in the higher elevational region, and Thornema. For the predators, we have found 9 predatory nematodes in the particular region. And one of the important findings, we found Carcolemus at the higher elevational zone of the region. And it wasn't present at the lower elevation. It could be due to the vegetation and various soil ecological parameters that we need to do. We need to replicate the sampling and analysis to find out the region for that. Then we have an enrichment index and structural index for the 5 elevational classes. It shows a food web diagnostic, which comes under the quadrant B and D, having the higher nutrient and low disturbance region. So you can see in the D section, D part of the diagram, the graph, you are seeing, we will see some of the points that lie under the low nutrient and low disturbance, which is of the higher elevational zone. So next we have done a principal component analysis, and the highly contributing factors were the elevation Doralemus, that is a bacteriovorous nematode, bacterial feeders and herbivores, followed by the various trophic groups and the soil physiochemical parameters. We have also done a dissimilarity test based on the Bray-Curtis distance, showed that nematode community from elevational classes was significantly differ when assessed at the generic level. And you can see from the graph that higher elevational composition was different from the lower elevational composition of E1, 2, and 3rd class. So summary, we have found 51 genera were recorded from the region. 32 genera significantly differ among elevational classes, comprising of bacterial feeder, fungi feeder, plant parasites, predators, and omnivores. And nematode diversity were relatively higher in the class 1 and 3. Trophic grouping of nematodes included 24 genera of bacteriovores, 6 of fungivores, 8 of herbivores, 5 of omnivores, and 8 were predators. Carcolemus was found at the higher elevation class only. The EI values significantly differ among elevation classes, and structural index, which is an excellent mean to understand whether the system is matured or structured, where high value represents structured and the lower value disturbed ecosystem. Pair-wise multi-level comparison of nematode community composition showed the nematode composition in higher elevation showing different nematode composition of the lower elevation. So it may be concluded that elevation is a significant factor for the soil nematode diversity, as elevation with specific vegetation, which is under process, this is one of the factors that we need to analyze further. So this is a preliminary result only, has a different local environment and physiochemical property that affect the soil biota, specifically soil nematode being present at various trophic level in the soil food web. Along the elevation gradient, there is a substantial difference in the taxonomic composition, the energy flow channel, and nematode community structure, which provide an actual example of understanding the role of soil nematodes in key soil ecological processes in the region. As a study is an initiative to generate a baseline data information in the region, evaluating the community structure along the elevation gradient. Therefore, some similar studies are required to comprehend the relationship between the nematode diversity and soil ecosystem of higher altitudinal region. So way forward, we are doing an experimental setup of open top chamber in the West Kamen district of Arunachal Pradesh, which we have performed earlier in the NMC first in the Gangotri National Park. So that same thing we are replicating in the West Kamen. So yeah, this is under process. We have already installed it. Analysis, first sampling has already been done in the October month. So yeah, this is a way forward that we are doing. And of course, the replication will be done in another year for understanding the community structure in the region. So yeah, this picture I want to show you the difference between how the open top chamber is basically helping to increase the temperature. These sheets are polycarbonated sheets, so they trap the heat. And you can see this how snow is melted inside the OTC faster than the outside the OTC in the control plot. So I would like to acknowledge all these people. Thank you so much.
Next speaker is Ms. Deepali Ramsey. She is going to make a presentation on climatic warming and its effects on diurnal respiration in alpine meadows of the Western North.
Dipali holds a master's in environmental management from Perth University, Delhi. Currently as a PhD scholar and educator under an internship project. She is concentrating on understanding soil-plant interaction, particularly in the context of climate change. Her work involves establishing long-term monitoring blocks and exploring the role of soil microfauna in carbon dynamics. As a nature enthusiast, she loves capturing the beauty of the outdoors for photography and enjoys bird watching in her spare time. Over to Dipali.
Thank you. I hope I'm loud and clear. नमस्कार, कृपया अपनी कुर्सी की पेटी बांध लें। हम पूर्वी हिमालय से पश्चिमी हिमालय की ओर जा रहे हैं। इस सभागार में 6 द्वार हैं, 2 आपके पीछे, 2 दाएं और 2 बाएं तरफ, जिनका इस्तेमाल केवल आपातकालीन स्थिति में ही करें। धन्यवाद। From the verdant east, where the valleys hum and the river dances, to the towering west, where the meadows breathe and the peak enhance. Let me take you from the Arunachal Pradesh to the majestic Bhagirathi peaks, where the Gomukh Glacier and the origin of sacred river Ganges is located. Good morning, everyone. Today I am going to present an ongoing research on experimental warming and its effects on the diurnal respiration in alpine meadows of Western Himalaya, which is also a part of NMC Phase 2 project, focusing elevational amplification. Elevational amplification can disrupt the entire ecosystem's stability and functioning, ranging its effects from impacting soil fertility and plant health to shift in the species distribution to altered carbon cycling. Talking about altered carbon cycling, let me introduce 2 terminologies here. First is soil respiration. That is the total carbon dioxide produced from the soil surface, which may be produced due to the respiration of plant roots, microbes, and decomposition of organic matter. Second, ecosystem respiration, which is the total carbon dioxide produced from the ecosystem, which comprises majorly of soil respiration and above ground respiration. And these respirations are approximately 10 times higher than that released by anthropogenic fossil fuels throughout the world. And that is why it is in our focus. Today we are going to talk about 2 major objectives of this component. First, to assess the effects of experimental warming on the diurnal respiration rates. And second, to understand its regulatory microclimatic parameters. Our study site is the Alpine Meadow in Bhoj Basa, Gangotri National Park, Western Himalaya, at the elevation of 4,000 meters above sea level. And the soil type experienced by the study site is sandy loam. We have installed open-top chambers which are hexagonal in shape and made up of polycarbonate sheet as a part of experimental warming. The sunlight falls onto these sheets and gets trapped, thereby increasing internal air temperature throughout the day. Let's have a glimpse of the study site. So we have selected 3 different locations in an alpine meadow in Bhoj Basa. As you can see, in each of the location, we have 6 open-top chambers with their respective control plots. So in total, we have 18 open-top chambers with their control plots. We are using LI 8100A to monitor respiration rates, which is coupled with soil moisture and soil temperature probe. We are also using fine-scale long-term monitoring data loggers, logging the data of air temperature and soil temperature hourly. Today we are going to look at the data collected once a month from June to October 2023. Diurnal measurements were taken in each plot to capture the variations throughout the day. That means 3 times the recordings were taken in a day. First, at the initiation of the sunshine with respect to the study site. Second, the peak temperature. And third, at the departing sunshine, again with respect to the sites. For soil respiration, we have clipped above ground plant part with minimal disturbance and left it for at least 24 hours to get it stabilized. However, this step was skipped for ecosystem respiration. Talking of the second objective, so far we have included a number of abiotic parameters like soil temperature, volumetric soil water content, that is soil moisture, relative humidity, and some of the biotic parameters like vegetation type and cover abundance. We have identified around 39 different species in our study sites and have collected their specimens, which are further categorized into 4 major parts: grasses, sedges, perennial herbs, and annual herbs. Talking of the second parameter of biotic component, cover abundance, which is the percentage of the ground area that is covered by a particular species in a given plot or study. As you can see in the picture, if you are standing and seeing at the plot from the top, you might be able to identify 3 to 4 different species like Elymus, Nepeta, Calamagrostis. However, the species like Thalictrum alpinum, which grows horizontally onto the ground, is not seen when you consider only the top cover. That is why in our study, we are considering both the top cover as well as the surface cover, and later using Brown Blanket scale method to represent the percentage of total cover of a particular species. For June to October 2023 so far, we have more than 1,90,000 data points for each type of respiration in each type of treatment, more than 1,500 data points for each type of respiration, and more than 3,000 data points for each type of microclimatic parameter we are monitoring. We have used our software for the analysis and have employed one-way ANOVA test for the diurnal variations and pair t-test to see the impacts of experimental warming. We have also used a number of models and finalized to use linear mixed effect models to understand the relationship between respiration and microclimatic parameters. Let's take a look at the results so far. So as you can see in the graph here, the peak air temperature was around 11 to 13 hours, with June and August being the warmest month during the observation duration, and October being the lowest overall air temperature. Similarly, soil temperature also peaked around 12 to 15 hours, August again being the highest overall temperature month, and October showed the overall lowest soil temperature. We installed open-top chamber with the objective of increasing air temperature, and we achieved it. Under experimental warming, overall air temperature was increased by 1.47 degrees Celsius, and this increment in air temperature had indirect effect on the soil temperature, and we got overall increment in soil temperature by 0.65 degrees Celsius. Talking of volumetric soil water content, that is the soil moisture, July, being the month which experiences most of the rainfall in that study area, showed the highest overall moisture content, and October showed the lowest overall moisture content. We had some significant spatiotemporal variations for all the parameters. Overall, the moisture was decreased under warming by 15 to 17%. However, there was no change in the moisture during the monsoon month, in spite of the fact that the temperature was raised. Ecosystem respiration peaked around mid-cycle, that is 12 o'clock cycle, followed by a decline in the evening. Significant increases in ecosystem respiration were observed in the month of June in all the diurnal cycles at all the sites. However, in July and October, significant increases were only limited at the midday cycle. Maybe due to the fact that limited solar intensity and radiation reduces the energy for photosynthesis and carbon assimilation, thereby regulating the respiration dynamics. Soil respiration also peaked around 12 o'clock cycle and then declined in the evening. Very interestingly, under experimental warming, there was no impact on soil respiration. However, in the month of June, significant increases were observed in the site 2, which was also consistent with the phase 1 study and reported by Tiwari et al. 2021. In June, high ant activity was observed, especially in the site 2, plot 11. As you can see in the graph here, under experimental warming, the soil temperature as well as soil respiration was increased, and you can see in plot 11 the soil temperature was further increased by 4.11 degrees Celsius. And it is known to us through review that Ant nests and activity are known to be the hotspots of carbon dioxide and leads to increment in soil temperature. This year also we saw similar trends in plot 11 and 12, thereby confirming the same phenomenon happening at the sites. So far, the cover abundance data is still under analysis, but I wanted to show some of the obvious and clear observations made so far. As you can see in the experimental warming pictures, the plant growth is much higher as compared to the control plots. Also, you can see the purple flowers of geranium and awns of Calamagrostis and Elymus. However, in the ambient control, the reproductive phase is yet to kick on. So there is a clear-cut difference in the phenology so far. Talking of relationship between respiration and environmental factors, respiration showed positive associations with the soil temperature and negative associations with the soil moisture. When we just considered the microclimatic parameters, they explained 7.88% and about 4% of the variability in ER and SR. However, when we considered the spatiotemporal variations, that is, incorporating the data of plots which are nested in the site, including time point and sampling month, the microclimatic parameters could explain 85.71 and 85.31% of variability in ER and SR altogether, highlighting the importance of consideration of spatiotemporal variations, especially when your experimental design is so nested. Major highlights of the study so far includes that increased air temperature significantly influences the microclimatic conditions in our study site. Warming affected phenology and cover abundance, particularly enhancing the growth of grasses in the alpine meadow. We have seen that warming so far has increased the cover abundance of all the grasses by 28%. Also, the ant activity influenced soil respiration in our study site, underscoring the importance to include such important biotic components when undergoing the soil respiration studies. And lastly, the microclimatic parameters significantly increase the explained variability in respiration, thus highlighting the importance of its consideration. We will implement this extended monitoring of respiration and phenology to capture more detailed responses to climate warming in our study sites and are hoping to apply micro-scale findings to a broader spatial and temporal scales to better predict ecosystem responses under different warming scenarios. Lastly, I want to acknowledge DST NMC project, Uttarakhand Forest State Department, and all the administrative, academic, and finance department without whom any of the field would not have been successful. All the faculty members, all the co-PIs of the project, NMC team members, all the interns and field assistants who have made this success so far. Thank you.
Thank you, Deepali. Okay, we are on time and we now break for tea. Please be back here in the next 15 minutes. So we reassemble at 11:15 minutes. Thank you.
There is another announcement. Some of you have not done the registration. I will keep the registration forms available outside. Please do that.
चल रहा होगा वीडियो भी है लगवा दो मुझे एकदम ना वॉइस कम कर देना उसका लगता है से कर देना।
अच्छा ओके। हाँ तभी
पूछ लेते ना, और किसी का तो नहीं है ना वीडियो, और किसी का नहीं होगा तो अभी कम कर देंगे।
आफ्टर दिस ब्रेक आई विल check presentations. Thank you.
क्वेश्चन आंसर राउंड भी होगा।
अरे विनोद जी, ये स्पीकर लगा रहा
तो पिन जरा लिस्ट खोल, लिस्ट
AI is more biodiversity.
ये तो है। वो भी तो है, अभी सेकंड वालों का भी तो होगा।
साढ़े तीन मिनट
आज 3:30
क्या
बजे।
मैं देख सकती हूँ एक बार?
फिर हो गया, मैं जरा उसमें नंबरिंग कर दूँ। इनका बताना क्या है आपका घूमने में।
अच्छा
1 बजे डी
DIKSHA
क्या नंबर है कल? 1 बजे। अरे यार ऊपर से गिरना मुझे। हेलो जी सर, गुड मॉर्निंग सर। टीम्स में so I think the timing is after lunch. No, you can try on YouTube का लिंक वेबसाइट पे है, डब्ल्यू आई की।
हाँ ये डिस्कशन में उसको डाउन कर देंगे।
वो सर लंच के बाद चलेगा, जब आपका सेशन होगा तब। जी सर, मॉर्निंग में ऑन हो जाएगा सर, जब भी सेशन होगा उसी समय ऑन हो जाएगा।
इसमें रेड पॉइंट
ठीक है, पूजा का बताना, पूजा का क्या नंबर है पूजा का?
पूजा पूजा कुमारी ठीक है बाद ये और 1 2 3 4 5 6 8
है। ठीक है।
एक तरह ऑडिटोरियम में
आपका क्या नंबर है अभिषेक का?
एक्वेटिक सिस्टम है, नीचे है। लंच के बाद पहला सेशन है,
नंबर
अभिषेक ना
हम मंगा लेंगे आपको।
8 9 10 ये नहीं है ना,
एक ही तो है। हाँ
ये डिस्कशन नहीं है।
नहीं है, कुमार प्रधान का है ना।
ये नफा है ना।
नहीं हाँ, इसके नहीं 10 क्लब बस निकाल
निकाल दो।
यस उदय। सर
Good morning, everyone. Pastoralism is one of the oldest livelihood in human history, and it is common across all different continents. It is divided into two major types. One is nomadic pastoralism, where human and herd move from one place to another with no permanent settlement, and another is semi-nomadic pastoralism, where the people and livestock stay at one place, but during time they take their herds into another place for feeding. The nomadic pastoralism in general is divided into two parts. One is migratory, where herd move from pasture to pasture and from place to place with no permanent settlement. And another is silvopastoralism, where the herd is generally kept within forest areas for grazing purpose. And the example of semi-nomadic pastoralism is sedentary type, where in general cattle farming in ranches is done across different continents. Since this is the oldest human livelihood option, presently with conservation coming into play, there are certain conflicts arising because of it, because the conservation which we follow is generally in essence of Western conservationism, where the essence is to block every person, every usage from the forest and habitat areas. As such, the topic of today's presentation is pastoralism and conservation in the northwestern Himalayas, a case study of Tirthan and Sanj Wildlife Sanctuary. This study was conducted under the project Present Status of Livestock Grazing and Suggestive Plan to Phase It Out in Tirthan and Sanj Wildlife Sanctuary, which was funded by Himachal Pradesh Forest Department. And the study was conducted into Great Himalayan National Park Conservation Area. The study area comes under Biogeographic Region 2A, and it consists of steep valleys and high mountains. Here we studied we did the study in Sanj Wildlife Sanctuary and Tirthan Wildlife Sanctuary. Coming to the history of the both wildlife sanctuaries, Tirthan Wildlife Sanctuary was established in 1976, and after which Great Himalayan National Park was established in 1984, and the recent addition was Sanj Wildlife Sanctuary, which was established in 1994, and altogether with ecozone region, this whole area makes up Great Himalayan National Park Conservation Area. The rights of the people to both National Park and Wildlife Sanctuary were made by Anderson in 1886, and which have persisted for 137 years. However, the rights of GHNP National Park were settled, and as such, the settlement of rights for the people of Tirthan Wildlife Sanctuary and Sanj Wildlife Sanctuary has not been completed yet, so they are still entitled to their grazing rights, their foraging and harvesting rights. And as far as Sanj Wildlife Sanctuary is concerned, there are 3 hamlets within the Sanj Wildlife Sanctuaries, and people have not moved out, and they're still using the area as a winter village, and they move completely towards the summer pasture after April. This region is quite rich in biodiversity. Overall in Himachal, 3,200 flowering plants are present. However, GHNP alone corroborates about 1,100 flowering species. The early studies done in the region were by Royal, Brandis and Parker. However, there was not much information. So after 1994, the FREEP project, which is Forestry Research Education and Extension and Education project, was implemented by WII, where the first proper systematic study was done by Singh and Rawat for the floristics part. Similarly, for the faunal study, the first proper study was conducted by Himachal Pradesh Forest Department under Himachal Pradesh Wildlife Project in 1980 and 1983. After 1983, GHNP Park was established, and after 1993 study, it led to the implementation of FREE project, where WI again did mammal studies, invertebrate studies, avifaunal studies, and pheasant surveys in Great Himalayan National Park. prior to FREEP study, there were no studies in pastoralism done. So a study was conducted by Mathur and Mehra and Tucker under, and they found that about 33,000 to 38,000 livestock was still at that time grazing in the national park, after which where rights were settled and the grazing was banned completely. Other studies done under FREE project were landscape management, socioeconomic aspects, village micro plan and eco-developmental activities, as well as remote sensing and monitoring plans. As such, for this study, we did three objectives. Our first objective was to study what is the current livestock grazing pressure in both wildlife sanctuaries. Our second objective was to see the extent of migratory pastoralism and the habitat degradation happening. And the third objective was to come up with a grazing management plan for the sanctuary areas. Coming to the methods for assessment of livestock pressure and migratory pastoralism, we did it in three parts. First, we conducted questionnaire survey, which was quite opportunistic of Gujars, migratory shepherds, and local villagers. For grazing pressure assessment, we selected specific meadows which were used for grazing by both locals as well as migratory herders. For forage yield, we did biomass sampling, and from that we found out carrying capacity of individual meadows. Then for grazing resource and utilization, we first identified different meadows utilized for grazing, and after that we did phytosociological assessment as well as wildlife assessment using camera trap. These here shows the location of different camera traps which we set up. We actually use the migratory route to study the movement pattern of wild animals, wild herbivores as well as carnivores in the study area. For this, we divided the entire study area into 1 by 1 kilometer grid, as the area was quite small, and we sampled at the most accessible and the migratory route areas to look at how the wild animals were interacting with the livestock. So the approach used for assessing grazing pressure and habitat assessment, what we used was the parameter used were forest health and forest disturbance as well as wildlife sightings. So for forest health and forest disturbance, we assessed pressure on the habitat on basis of fuel, fodder collection, as well as how much frequency of fire and grazing happening in the study in the region. The indicators for forest health used were canopy density, forest layers, and DBH distribution of the forest. For forest disturbance, we looked into invasive species infiltration, vegetation affected by grazing, as well as presence of domestic livestock. For wildlife sighting, as a parameter, we looked into the presence and absence of wildlife in the study area. And for finding the presence and absence, we used both direct sighting as well as indirect signs like calls and scats. In the entire region, the habitat was divided into 5 major categories, namely settlement. This settlement here is within the wildlife sanctuary, then forest caps, forest bare ground, and alpine meadows. For our study, for phytosociological assessment, we divided the forest into lower temperate, upper temperate, subalpine, and alpine region. Coming to the forest structure of Tirthan and Sanch, a total of 137 species were recorded. Since we had much less of a time and due to some natural disaster, we used rapid biodiversity assessment approach, and as such, we were not able to incorporate sample the whole sanctuary. The sampling was mainly concentrated on the livestock route and the grazing meadows. So in total, we found 23 tree species, 39 shrub species, 55 herb species, 10 were gramonoid, and we only found single climber. In each individual sanctuary, the total tree species present were 17 in upper temperate forest. In in Sanj Wildlife Sanctuary and 9 in Tirthan Wildlife Sanctuary. Overall, the diversity was high as far as tree species is concerned in Sanj Wildlife Sanctuary. However, in alpine region, the diversity of herb and richness of herb was higher in Tirthan compared to Sanj Wildlife Sanctuary. The major tree species, we also found IVI, and the major tree species in lower temperate was Quercus semicarpifolia, PCS smithiana, and Aesculus indica dominated the habitats, whereas in shrubs, Deutzia stamiana and Berberis lycium were the most dominant species. In Sanj Wildlife Sanctuary in upper temperate region, Pinus wallichiana, Quercus semicarpifolia, and Prunus were the dominant species, whereas Cotoneaster, Berberis, and Rhododendron were the dominant shrub species. In Tirthan Wildlife Sanctuary, Quercus semicarpifolia and Abies spindro dominated the habitats, whereas Rosa vibiana, Berberis, and Sorbus were the most dominant shrub species. Coming to the subalpine region, Abies spindro, Pinus wallichiana, and Quercus semicarpifolia dominated in the Sanj Wildlife Sanctuary. Rhododendron lepidotum, Rosa serica, and Berberis jaskinia were the most dominant shrubs. In Tirthan Wildlife Sanctuary, Quercus semicarpifolia, Abies spindro, and Betula utilis were the dominant tree species, whereas Rosa serica, Viburnum grandiflorum, and Sorbus lanata were the dominant shrub species. Looking at the wildlife interaction, we found a total 357 independent records in Tirthan Wildlife Sanctuary and 96 independent records in Sanj Wildlife Sanctuary. Overall, we didn't get much wildlife in both wildlife sanctuaries, as only 13 mammals were present in Tirthan and 7 mammals in Sanj. However, most of these were domestic livestock present within the both sanctuaries. So we only got common leopard, Himalayan brown bear, and Himalayan black bear in Tirthan, and only yellow-throated marten was recorded in Sanj Wildlife Sanctuary as a wild species. These are these are the pictographs of the grazers. On the left side, it's the Tirthan Wildlife Sanctuary. You can see they have a systematic way and they follow a particular route going from different meadows back and forth. This here map shows the route taken by migratory herders in both Tirthan and Sanj Wildlife Sanctuary. First, talking about Tirthan Wildlife Sanctuary. In Tirthan Wildlife Sanctuary, we found 4 different group of migratory herders. 2 were Gujars and 2 were Gaddis. So this here green depicts the Gujar migratory route, and in red color depicts the Gaddis migratory route. Gaddis in Tirthan Wildlife Sanctuary are local people who migrate from village towards the pasture in early summer or spring and then come back by the month of September and October. So the Gujars start their migration from Luri, and Gaddis start their migration from Bathad and Mashyar, which are 2 villages right at the border of the wildlife sanctuaries. The Gujars' final rest camp is at Sarhan. They start their migration journey in the early March and reach there by the end of April, where they stay. After which they establish their permanent camps at 2 meadows. These 3 meadows collection shows the rotational grazing done by the Gaddis and Gujars. In Sanj Wildlife Sanctuary, these are the migratory routes. The green migratory route is by herders from the outside, and red migratory route are from the inside of the sanctuary. The outside herders start their migration from Ani, and they set up their permanent camp at Stogni and at this one is within the national park at Taktisod. So coming to the livestock holdings in Tirthan Wildlife Sanctuary, the major livestock holdings were of sheep, goat, horse, buffalo, and oxen. While in Sanj Wildlife Sanctuary, only sheep and goats were present. For livestock carrying capacity, the parameters we looked into were area, forage produce, grazing days, livestock units and carrying capacity. Outside PA, we sampled one site where we found that the pressure was very high. Similarly, in Tirthan, most of the area of the sanctuary fall between 1.6 and 217 hectare, and livestock unit at each individual meadows were high, and most of the meadows were overgrazed except Munna and Flach. Similarly, at Sanj, majority of the meadows were overgrazed. Coming to the grazing impacts, we found we divided the impacts of grazing into three major categories. First is physical impact, looking into soil compaction and soil erosion. Then was the social impact, which leads to human-wildlife conflict. However, the people did not record any human-wildlife conflict there. Then third is the biological impact due to the fodder and forest dependency as well as grazing and fire. To understand how this impact is affecting the sanctuaries, we did a fragmentation analysis, and here we can see the dark green area here. It shows this area, it shows the edge of the habitat which is fragmented, and the red area shows the habitat fragmentation which is taking place right now. So white is the forest area and colored is the meadow area. In here, this picture shows how the fragmentation is taking place. This is example of Baja meadow. This here is the Munna meadow where no grazing is taking place. However, it is still present in the red area where the habitat degradation is taking place. Similarly, is the Josh meadow in Sanj Wildlife Sanctuary and Nanthua meadow in in Sanj Wildlife Sanctuary. For this grazing, we found major threats was of high livestock density. We identified 3 meadows in Tirthan Wildlife Sanctuary and 5 meadows in Sanj Wildlife Sanctuary. Habitat encroachment was seen in 3 meadows in Tirthan and 2 meadows in Sanj Wildlife Sanctuary, as well as species like Polygonum, Romex and Impatiens were invasive in the area. Then overgrazing was observed in all meadows in the study area. Then there is also illegal poaching of the medicinal and aromatic plants happening there, which is generally not reported. Then there is also issue of illegal grazing happening. For this, we made 5 management recommendations. First was the carrying capacity has to be maintained at 200 to 600 ACUs, and then we have asked the forest department to identify and monitor the present permits of the locals, as well as relocation of Gujars and Gaddis from both the wildlife sanctuaries, as well as there is a need for the habitat restoration of meadows, as they are heavily overgrazed and degrading over the time period. Then we also need to do community awareness for the harmful effects of grazing in the study region. So coming to the summary, our major recommendation was to ban grazing in the 30% area in the sanctuary, and to identify and remove invasive species, and if possible apply early detection and rapid response in the area. Then we also recommended reorganization of Sanj beat into multiple management beats as it is quite big, and establishment of high altitude plant nurseries for in vitro propagation of the local species, as well as establishment of high altitude surveillance camp to control poaching and smuggling of medicinal plants. Finally, we also recommended a long-term research and monitoring project for both sanctuaries to assess the impact of pastoral activities happening simultaneously. Thank you.
Thank you, Rahul. We move on to the next presentation by Ms. Pooja Kumar. She'll be talking about understanding distribution and occupancy of endangered Himalayan red panda in Darjeeling, Himalaya.
Ms. Pooja completed Masters in Wildlife Science from Aligarh Muslim University during 2022. Currently, she is working under the Active Red Panda project as a She also registered for her PhD in August 2023 at ACS Design, wherein she wished to understand the ecology of red panda in northern West Bengal. Over to Ms. Lisa.
Thank you for the introduction, sir, and good morning, all of you. Today, on this Himalaya Divas, I'm here to add some more insights to these unexplored, or I can say very less explored areas, wherein I'll be discussing or adding more into the secret lives of these elusive red pandas in the Darjeeling district of northern West Bengal. Before moving on further to understanding these ecological aspects, I would like to play a video with a brief description and basically the natural history of the species, wherein you will see how the species was described, its species classification and the distribution of those two species of red panda. Sorry. The first red panda, because it was described nearly 50 years ago than the giant panda, which is basically found in the China. It's not pausing video, I need to explain. Okay, let it be. It was described by Frederick Cuvier, a biologist who described the species nearly exactly 200 years ago from today and quoted the species as quite the most handsome mammal in existence. Further, the red panda was studied in Sikkim and Nepal Himalayas by Bregan and Hodgson. This was the first illustration he provided of the species. And then up till from the description to mid 20th of the century, the red panda was classified in the under Procyanidea family because of its resemblance to raccoons. And in 1921, Pocock, he introduced and suggested to put it in a separate family of Ariidae. And in 1938, two subspecies of red panda were classified by Allen, which are Ailurus fulgens fulgens, which is Himalayan red panda, and Ailurus fulgens tiny, which is Chinese red panda. Further, if we talk about at present situation, in 2020 by Hu et al. A study, and 2021 Supriya et al. A reconfirmation came for the studies existent, which say the red panda species basically are two species. They did on the basis of genetic analysis of the samples and further classified them. The distribution, if you would have seen in the video, the Chinese red panda lies in the northern Myanmar and China region, and whereas Himalayan red panda lies in the other three ranging countries, which are Bhutan, Nepal and India. And India is the only country to harbor both of the species of red panda. So moving on to the basic overview and introduction of the species, red panda, which was, as I said, described 2 years ago from today, is an arboreal and solitary carnivore. It is a sole representative member of the Ailuridae family and is a habitat specialist species which has a residing niche in with temperate forest and in the understory of bamboos. This species is of peculiar interest as it lacks any direct linkage to its other small carnivores. As per the current status of the species, IUCN classified the species in 2015 as endangered. And as per Indian Wildlife Protection Act 1972, it is put in Schedule 1. CITES, which is a convention on illegal trade of endangered species, also classify the species in its appendix first. And as per IUCN 2015 data, if we see there is a current decline in the population, which they give as nearly less than 10,000 mature individuals in the wild. Talking about the threats which are pertaining in wild to the species, major threats are habitat degradation, its loss and the fragmentation, poaching and illegal trade, which is still currently prevailing in other countries, ranging countries like Nepal, anthropogenic pressures like activities, tourism activities, development activities, disease outbreaks in the wild, which needs to be further studied yet, and ill-mannered tourism, which is now proving to the most effective threat to the species in the wild. If we talk about the red panda distribution, red panda distribution is lies in a very narrow range, only in 5 Asian countries: India, Nepal, Bhutan, Myanmar, and China. It is endemic to the temperate and subalpine forests of the eastern Himalayas. And as you can see there, the start position shows what we will discuss today is my study area. As per the preferable altitude of the species is 2200 to 4500 meter. The same is our study being carried out in the selected area only. For the occurrence of red panda, as per the literature, in India, if we see the status, it is found in all 6 districts of Sikkim. In 2 districts of West Bengal, which is northern West Bengal part, those are Darjeeling and Kalimpong, and in 14 districts of Arunachal Pradesh, wherein during our project study only we added 1 more district, which was Anjao. In Nepal, its status, it's found in 24 districts, in Bhutan 13 districts, in China in the provinces of Yunnan and Tibet, and in 58 counties. In Myanmar, it's found in only selected northern Myanmar part. The study which I am going to talk about is a part of an IDWH Red Panda project, wherein the project objectives are to assess the distribution and abundance of the species and to assess and predict the habitat and distribution of red panda throughout its distributional range in India. With that, we also aim to develop a monitoring protocol for red panda survey in India. Study area, which is part of West Bengal, northern West Bengal. Red panda habitat lies in 2 districts, as I've told earlier, Darjeeling and Kalimpong districts. This area, Darjeeling and Kalimpong red panda habitat, is a transboundary landscape which encompasses in itself 4 protected areas which vary in range of altitude from 183 to 3660 meter. The major areas in the protected area with their geographical areas are given in the table below. Total area for survey in North Bengal is 220 square kilometer, which we identified for our survey, out of which Darjeeling has 167 square kilometer of area, which is covering both protected and non-protected area. If we talk about the significance of protected areas specifically, Singalila National Park, which is present in Darjeeling district, borders with Sikkim and Nepal in its north and northwest side, and Nuara National Park, which is present in Kalimpong districts, borders with Sikkim and Bhutan in its north and northeastern side. Talking about the other species significance in the area, apart from red panda, many other threatened taxa species are also present. Pheasant species like satire tragopan, blood pheasant, many rhododendron, around 20 plus rhododendron species are found in the study area. For today's presentation, we have selected, this is the study area for Darjeeling, wherein we have conducted a survey for within question of to understand the current spatial distribution of red panda in Darjeeling, Himalaya. We have estimated the occupancy of the species in the area, and then to identify the key environmental and ecological factors which are affecting the site-specific habitat use of the species. We have done the site-specific habitat use, and my third objective is to understand the daily activity pattern of red panda and its co-predators. And its predator, basically potential predators, specifically in Singalila National Park, and to understand how do they temporally overlap. If I talk about the major challenges which we faced while conducting a survey, was not only red panda being an elusive species already becomes a difficult task to study. Apart from that, the frequent occurrences of landslides in the area, low visibility, because red panda also known as the cherub in the mist all around through the season when we surveyed, and the dense habitat it's found in made our work more hindering basically. For the present study, the methodological approach what we took was first we identified the study area on the basis of altitude, which was the preferable altitude of red panda, and over that study area 5 square kilometers grids were laid. And based on those grids, when we went to the area, basically, we conducted a primary data by science surveying the area and conducting key informant survey. On the basis of the data collected from them, the camera traps were deployed on the ground in the area. And also, at each camera trap station, and at a 10-meter radius circular plot, habitat and environmental covariates were taken. At each circular plot, microhabitat and anthropogenic data collection was also done, which includes the presence of human settlement, the animal species, livestock in the area, and others. Talking about the camera trap results, camera trapping was basically done from March 2023 to December 2024, wherein we have deployed 64 camera traps with 6147 trap nights, which yielded in 1469 total captures with 20 mammal species in it, and we got 127 independent captures of red panda. Through that, the data conducted and taken out from the camera trap, we have calculated the RAI of individual species which were captured. And to us, the barking deer, which has the recorded maximum RAI following red panda, which has 2.06 with a standard error of 0.71. If we talk about the spatial heat maps on the basis of the camera trap data which we recorded, we have plotted the heat maps of red panda along with its potential predators within the area. Now, objective 1 results, which was occupancy. For occupancy, we basically have used single species, single session framework, wherein we have analyzed the data on the grid wise using RStudio, and all the variables, environmental and on-site variables were taken for the analysis, which yielded the occupancy results told the elevation is the most critical factor which is affecting the species in the study area, along with the human presence, which may be a question for human presence, because the area where red panda was most captured is a Singalila National Park, which I will show further. The area on here where the highest prediction occupancy was attained is Singalila National Park, whereas on its left side is a transboundary area where are the fringe villages across, like in India there is no settlement on Indian side across on the highest elevation, but down to the elevation when we come there are fringe villages. And on the left side, there is Nepal border. So those people, plus it's a highly tourism affected area. And along the national park itself, the trail and trekking paths for tourists are there, which even yielded us in camera trap records of like high density of human presence. If we talk about the second objective, site-specific habitat use, for that on-site variables were taken for site-specific habitat use, and we performed general linear modeling. Collinearity was tested among the variables which we took, and many models were run as per that. Out of those, these 8 models with different variable combinations were taken, which was best selected, and out of that, the model with lowest AIC value, which has the variables altitude, rock, canopy cover, litter depth, and snow, was selected. If we talk about the effect of habitat variables here, As I showed in the occupancy level also, the red panda is basically mostly affected by the elevation, and here also on site also it showed that elevation or altitude is the most contributing variable to the species. Plus canopy cover and snowfall also showed the positive effect. The data when we got the maximum captures of red panda was basically then after post monsoon, when there is snowing in the area. Litter depth, which is on-site data, also showed an impact, but that was not considerable. If we talk about the activity pattern of the species along with its potential predators, we have taken the time stamp data and then plotted the graphs for the activity in Oriana, wherein we saw red panda showing the diurnal behavior. Now here I would like to tell you, red panda basically most of the literature say is a crepuscular animal, but there are some studies which are radio collared in Nepal and in Wolong Reserve of China which contradict and also along says that the activity pattern which is shown here goes with that. If we talk about the temporal overlap of the species along with its predator in Singalila National Park, the highest overlap coefficient we saw was with leopard, then following with Asiatic black bear, and then with yellow-throated marten. I would like to conclude my work here because red panda habitats through this, whatever analysis we did and the data we collected, we can conclude key red panda habitats can be identified with the analysis of occupancy we done, and also at microsite level, altitude and canopy cover were found significant to affect the habitat use. And in the study area, red panda showed more of a diurnal activity with its peak after the sunrise and nearly at about 8 a.m. in the morning. Now, exclusively, if I say no red panda study exclusively has been done using camera trapping as a tool across its entire distribution range. This is the first study we have done and we are continue doing, and this we want to prove as a baseline information in order to prepare a conservation action plan, which is our main project objective. If I talk about the way forward, in the present study area, wherein in northern West Bengal, this area is also under my PhD study area, wherein we need to do the intensive study for the species, wherein we will do involve the community engagement and to understand their attitude and perception towards the species. And earlier, the dietary species has been done on red panda in the study area, but to show and to understand the change in its diets within that 24 years of period, we will also do dietary analysis. For that, we have collected the samples across wise cat samples. Intensive study in the Kalimpong district, which has a potential area for red panda, will be done. Across that, apart from that, the main aim which I am talking about is sampling protocol preparation. In the 5 out of 5 ranging countries, only 2 countries, Bhutan and Nepal, has the protocol till now, even after 200 years of the description of the species. And we want to prepare it and to plan and provide it to all the 3 states where red panda is present: Northern West Bengal, Sikkim, and Arunachal Pradesh. Through this, I would like to acknowledge the following people who have helped me entirely, and especially my field people and project teammates and other colleagues. Till now, red panda, in basic knowledge, if we say, is known to mostly feed on bamboo species. But this video, small video I'll play, and this was recorded in December 2023, this year, when I was on field. Here, red panda, after post-monsoon, when the bamboo shoots become adult, and it generally feeds on the young shoots of the bamboo, it is, in order to meet its nutrition requirements, it is feeding on the Sorbus cuspidata berries. This video was recorded in Singalila National Park of Darjeeling district. I've been working on red panda for nearly about 2 years and continue to work even for PhD and after that. But this species have always mesmerized me. and continue. With that, I would like to say thank you to all, and we wish, as part of our project, we come again in next ARS with robust data and everything. Thank you.
Thank you, Pooja. Now we have next speaker, Arjun Kumar Pradhan, will be making a presentation on conservation of red panda in Western Arunachal Pradesh and assessment of current status.
Pujan Kumar Pradhan is a project associate in the IWH Red Panda project. He has a master's degree in zoology from the University of North Bengal and is currently pursuing PhD at the University of Calcutta. For over 6 years, he has worked on certain mammal species in the Indian environment. Over to Pujan.
Thank you very much, Dr. Vinith. Am I audible? Hello, everyone. A very good afternoon to all of you. I am Pujan Kumar Pradhan. Today, I'll be talking about conservation of red panda in the Western Arunachal Pradesh and assessment of the current status. This study is also a part of the project that my friend already introduced about that, IDWH red panda. So my friend Pooja have given the comprehensive background analysis of red panda. So I'll be just giving the overview of this species. Red panda, also called the first panda, is a highly diet-specialist mammal found in the mountain forests of Himalayas about 2100 meters. It is listed as endangered in the IUCN Red List and protected under Schedule 1 of the Wildlife Protection Act of 1972. And its trade is prohibited under the CITES Appendix 1. According to IUCN, its population has faced severe decline in the last few decades as a result of indiscriminate poaching, habitat loss and fragmentation, and climate change. So let's have a look on the current distribution map. So starting from central Nepal, it is distributed to Bhutan. It is also distributed in Myanmar, China, and in India it is recorded in 3 different states: Arunachal Pradesh, Northern West Bengal, and Sikkim. So objectives and research questions for my current study are: number 1, to assess the population status and threats of red panda within the current study area. So under which we ask the question like, what is the current population trend of red panda in the study area within last 10 years? So, and also to understand the threats of the red panda. Second objective is to estimate the red panda abundance, which we ask, what is the relative abundance of the red panda in the study area? Third objective is about the predicting habitat suitability for red panda in Arunachal Pradesh. Here we identify the suitable area for red panda within the whole Arunachal Pradesh, and also the proportion of the suitable habitat that falls under the protected area network and outside the protected area network. So this is Arunachal Pradesh, map of Arunachal Pradesh. Red panda has been recorded in 14 districts of Arunachal Pradesh. And for this study, we are focusing on the two westernmost districts of Arunachal Pradesh, particularly Tawang and West Kameng. So study design. First of all, we selected the red panda habitat on the basis of the previous secondary literatures, published literatures, where we done the key informant survey to gather the baseline information about the red panda and also about its threats and population trend. The key informants during our survey were the hunters in Arunachal Pradesh and the forest officials, the Gaumburas or the village headmen, etc. We overlaid the 5 square kilometer grid over the selected red panda habitat and performed the non-invasive sampling techniques like sign survey and camera trapping to estimate the relative abundance of this species. The occurrence points gathered from science survey and camera trapping, along with the secondary data that we generated from the online repositories like GBIF and INET, along with the environmental variables and bioclimatic variables, we did the habitat suitability modeling using Maxent algorithm in R. So this is my study area, and here are the grids that overlaid. The green shaded grids are those grids that we have already done the camera trapping and we have the data. And the red grids are those grids where the camera trapping are still in the field, camera traps are still deployed in the field, and we don't have that data. So coming to the results, Objective 1, assessment of population status and threat. We conducted the key informant survey, particularly targeted to the very specific kinds of people in the villages. We surveyed 72 villages in Tawang with the total number of interviewees, 192 number of interviewees. And similarly in West Kameng, 46 villages and 125 interviewees. For this particular presentation, I'll be just talking about the data from Tawang only. So here is the graph showing the respondents, various age group of the respondents. So during our key informant survey, maximum age group is from 40 to 49. About 53% of the total respondents were aware of the red panda presence in their surrounding locality. Out of 53% of the respondents who said yes, we again narrowed down, and around 57% of those total who said yes have cited the red panda directly. Again, among those, around 81% of the respondents cited the red panda in the wild. And around 6% of them, they have seen the red panda in captivity. Actually, in Tawang, West Kameng, what happens is people, whenever they go to jungle, sometimes what they do is they just try to catch the red panda, and they will just take the red panda into the village, take the red panda back into the village, and for a few days, and they release back that red panda in the wild later. So that is how some of the people have seen the red panda in captivity. So again, around 85% of the respondents, total respondents, they have responded us like the decreasing trend of red panda in the wild around the study area. So we also assess the awareness of Wildlife Protection Act and conservation status of red panda, particularly IUCN conservation status of red panda. And we also we asked the particularly specific question for the threats of red panda, and people responded like this. Around 38% of respondents said the wildcats are the major threats to the red panda in particularly westernmost Arunachal Pradesh. And feral dogs, 18% of the respondents says feral dogs. So moving on to our next objective, estimation of the relative abundance. So we used encounter rate and capture rate as the proxy to the relative abundance. So during our sign survey, we recorded 18 signs of red panda in the Arunachal, 2 districts of Arunachal Pradesh, where with the effort of around 165 kilometers. So here is the sign location that we recorded during our survey. And this is the total number of camera traps. We have deployed 89 camera traps in Tawang as well as in West Kameng, with the total trap nights of 5,851, with the total independent captures of 18. So here is the RAI, Relative Abundance Index, of all the mammals that has been recorded in both of the districts. So there are 28 mammals we have recorded till date in the red panda habitat. And out of which barking deer is the most abundant species in both of the districts. So for third objective, we selected the occurrence points from the field, as well as we gathered point locations from online sources. And we verified the locations and further model of the covariates like environmental layers and bioclimatic layers and here the best model with the AOC value of 0.95 says only 3% of the total area of Arunachal Pradesh is highly suitable, out of which only 3.5% of the total area is moderately suitable. So these are the various covariates that we use, and isothermality, mean annual temperature by 1, terrain ruggedness index, TRI. These are the most influential and contributing variables, and the response curve has been given in the right side. So again, we try to calculate the amount of suitable area inside the protected area network. So it is showing that only 2.1% of the highly suitable area is inside the protected area network, and 23% is inside the moderately suitable area is inside the protected area network in Arunachal Pradesh. So according to elevational gradient, red panda prefers 2200 meters to 4000 meters elevational gradient. So we try to calculate how much area is inside the suitable area of protected area. So only 2.8% of the total suitable area is inside the protected area. So in conclusion, besides there are various conservation efforts going on in Tawang and West Kameng areas, red panda is still showing the decreasing population trend in the study area. Though low number of red panda detection in the study area may reflect either a small population size or it may be due to suboptimal habitat quality. Thus, we further will be doing the habitat assessment and analysis study. The habitat suitability modeling predicted only 7% of the total area in the Arunachal Pradesh as suitable habitat for red panda. That too, very small proportion of the suitable area falls under the protected area network. So, other effective area-based conservation measures like community conserved area should be prioritized and strengthened for the long-term in situ conservation of such landscapes. We also in further plan to study corridor connectivity analysis across its distribution range in India to identify the potential corridors and prioritize their management and conservation. So I would like to acknowledge following people. Thank you very much.
Let me say that I request all the speakers to come on stage. Questions for the last two speakers, if you can ask questions, please come here. We have received quite a few questions, so we run now in the interest of time. We already, we'll ask just 3 questions for students. I'll request the speakers to be very brief in your responses.
So the first question for Shagun Thakur, this is from Dr. G.S. Rawat. So he is asking any information on abundance of large carnivores, especially leopard and Asiatic black bear in the past in your study, based on secondary personal communication with past hunters.
Hello, thank you for the question, sir. So, in case of leopard, as per the local people, I have asked them. So they said it used to be there earlier, but now it has completely vanished from the area. And if about Asiatic black bear, we did get some captures in camera trap also, and we have seen the hunted skins of black bear in the forests, in the villages and campsites, but they haven't mentioned much about Asiatic black bear. But yeah, for leopard, they have said that it has been vanished currently, but it used to be there.
The second question is from Bilal sir. Interesting presentation and information from remote side. Any specific reason for using Bayesian regression analysis? Is Goral presence is influenced by canopy height or aspect?
Hello, sir. Thank you for the question. The specific reason for using Bayesian analysis was only that it handles the small size data comparatively better, as the case in my situation where the data was comparatively less. And for Goral, it responded, for the canopy height, it responded negatively with the canopy height. As the canopy height increases, it was avoiding such areas. And for the aspect, the model didn't produce much influential results for the aspect for Goral.
The next question is from Dr. Salvatore. Wonderful to see rare species captured from West Tamil. Red foxes are also found in alpine steppe regions. How is it that you are finding show preference to more forested areas?
So in my study area, red foxes are responding negatively to the canopy height. And I mentioned also that it was only recorded above 4,000 meters. So it was not responding positively to the canopy, but negatively to the canopy, not positively to the forest areas.
That's it.
Thank you. Thank
you. Sure. Now, there is a question for Pooja Pan. This question is from Dr. Manjari. Activity pattern of mammals. You concluded that most mammals are diurnal. Could it be because you did not sample bats, or did you not encounter bats at all? What is the proxy for activity? Movement and presence in camera trap? Is it a good proxy?
Actually, we didn't get the bat captures. So that's the reason the activity patterns of mammals are dynamic.
The second question. Oh, your second question, Pooja.
So the sorry, I didn't get it. Yes, ma'am.
So the next question is from Bilal sir. Interesting presentation. Based on your experience of camera trapping, what are your suggestions for temporal camera trap? Okay, improved CT design, sir.
So we have to do systematic grid camera trapping as we are doing in 4x4. So we'll also use 2x2 kilometer square grid. So to capture all the species like large mammals, small carnivores, as well as small animals. Sir.
The next question is from Gopi sir. He's asking excellent work. He's complimenting you. Many compliments. Your talk and abstract mentions Kian had lower capture rates. A better inference for the species will be to drive from. Yes, sir. Please. So, whatever you showed in your presentation, you said southern Kenya.
Yes. Yes. Yes, sir.
What a better inference would be that there are very different regions.
Yes.
Just drive from Kudamar into Solomon, that is the alternate. You can find all the 35 individuals. To say lower capture rates of southern young village, maybe I don't know.
Sir, actually, there are some camera traps which are still in field. But as I have already mentioned that because of floods and all, we were not able to access those camera traps. So that's the reason we have lower capture rate of southern cam.
next question is for Himanshu. Dr. Salvador is asking, did you observe any impacts due to a lot of road construction or different areas you sampled with respect to fish diversity and abundance?
Road construction particularly have no such influence because roads were constructed like 5 to 4 years ago, and whatever residues have been into the river have been washed away due to floods and all. So I don't think roads construction have so much of impact in the fish diversity.
So that's it for Himanshu.
Sir, thank you for the question.
So the next question is for Nihal. This is for, in fact, this is for all, Sharon and Himanshu. Dr. Gopi is asking, all of you use this Kameng River Basin. Is it not correct to use Kameng River Basin? Kameng River Basin itself is a sub-basin of Brahmaputra. Better to use watershed instead of basin. Yeah, government of Andhra Pradesh also recognized Kameng River Basin. You comment on this. Here, I just wanted to know, this Kameng River Basin is repeatedly used in the abstract notes and presentations. I just want to know, government itself, Arunachal Pradesh government itself recognizes only the Kameng Basin. The Kameng River Basin itself is a sub-basin of Brahmaputra River Basin. Now you are saying West Kameng River Basin. So, is West Kameng and East Kameng are just administrative boundaries? Not located in that, we could just say that it is watershed. It's smaller area compared to the entire Kameng River. This is my comment. Yes.
Okay.
So actually we are working only on the West Kameng district. So, yeah.
River is only one.
Yeah, yes.
River is only one.
Correct. Right. That's it. So the next question is for Priyanka Kashyap. Do you think it would be better analytically to consider elevation as numeric variable instead of categorical variable? And there are some suggestions you can give.
Thank you for the question and for the suggestion also. So, see, this is, am I audible? So for the elevation, see, we have to take two main variables, that is the vegetation and elevation. So this is a preliminary study. So that's why we take an elevation as a class, as a categorical variable.
So the next question is from Dr. Arun Kumar. Very well done, Dr. Priyanka. It's complimenting you. Really nice to see your continued effort on understanding on the soil nematode diversity in the western and eastern Himalaya. Curious to learn on your findings on the differences of soil nematode diversity in eastern Himalaya and western Himalaya.
Thank you for the question, sir. Yeah, this is our next step after analyzing, after completing our eastern Himalayan data. Then later on, we'll compare along the elevation and vegetation also. Yeah.
So there is another question from Dr. Dehradun. He's asking how the variations within an elevation gradient is incorporated, considered in the sample.
Can you elaborate a little bit?
So, Priyanka, so during your sampling, you haven't really mentioned, you only mentioned about the elevation differences, right? So how, within an elevation category, there are so many different habitats, how those things are considered in your sampling?
So I'll consider that later on. So as I said, this is a preliminary. So that's why I've showed what I have right now. So later on, I'll incorporate, maybe in next ARS, I'll incorporate then everything and let you know about this.
So that's all for Dr. Priyanka. The next question is for Dipali Council. This question is from Dr. Mandiri. She's asking, a plot showed greater than 4 degrees centigrade in temperature in the experimental setup. What was this special about this plot?
Okay, so that is my favorite plot, ma'am. Thank you for the question. In that plot, we saw not just in that plot. So, if you saw the graph, it was site to 3 plots 9, 10, and 11. The 10th plot showed further increment of 4.11 degrees Celsius because it had more many numbers of ant nests and ant activities. And as per the review, we found that due to that, the overall respiration and soil temperatures was increased. We have taken the samples of ant this year because first year took us by surprise. So second year we are taking more data to get a better understanding that what is exactly special for that plot. We are looking into that, but so far there is no difference in the composition, plant composition.
The second question is 0.65 degrees centigrade change in temperature was found overall in the experimental plot. To what extent is this going to affect flora and fauna? More plants, yes, but will the type of plant also change?
Yes, the plant composition so far is changing in our plots, and we have some significant spatio-temporal variations. That means even within the sites, different open top chamber versus control plots are giving us some different directions of plant composition. That is why I said the data is still under analysis to give you a good review of it. Thank you.
The next question is from Dr. Raman Kumar. As far as I understand, OTC were installed for the entire duration from June to October, while your primary purpose was to exclusively look at the effect of raised temperature. Do you think the OTC are influencing the plant community in other ways? For example, they might be preventing seeds from arriving. It would be nice to know your thoughts on this.
Thank you so much for this question. I really wanted this to be asked. So, before conducting this research, the same question was in my mind also. So, in the same landscape, we had deployed some separate open top chambers, and we have kind of covered the control plots in a similar manner, which are open from the top, to just see whether they are impacting the distribution of seeds, or is the open top chamber barrier for the plant growth overall. And from the preliminary findings of the first year, no, open top chamber is not the barrier. In spite of its height, it is just the temperature and set of other microclimatic parameters which is leading to the increment in overall plant growth.
The next question is from Dr. K. Ramesh. Sir, is complimenting for your very well articulated presentation. Suggestion is please provide sample size in the result slides, without which it was hard to comprehend the variation. And the question is, increasing temperature, increasing soil temperature appears obvious. Could you consider wind temperature and wind direction as potential variables to alter the variability, affect the variability?
Thank you so much for the suggestion, sir. I will definitely incorporate it next time. And as you asked the question about wind direction and speed, that was in our consideration list, but unfortunately due to restraints of resources, we couldn't have enough sensors to monitor that. But not just the temperature, we are also looking onto the soil moisture, soil temperature, overall humidity, surface carbon dioxide, precipitation, water vapor content in the air, and so on. The parameters which we have sensors to do, that is one of the technical constraint for us.
So that is all for
Thank you.
Deepali. The next question is from Dr. K. Ramesh for Rahul Kumar. Very important study, and hope you compared the study on grazing in GHNP during 1995 to 20. GHNP, the question is, the GHNP is one of the national parks that has final notification for national park status, meaning no anthropogenic activities allowed unless permitted by law. Your recommendation for grazing permit is for entire
Park areas.
selected sites.
Thank you for the question, sir. So the issue with grazing is in GHNP, as it is the only portion which is declared as a national park, so grazing is banned there. But Tirthan and Sanj Wildlife Sanctuary are outside of the national park ban because there are no right settlements. Second issue comes is due to migratory grazers coming through Sanj Sanctuary or in Tirthan Wildlife Sanctuary, the old routes are still open, they are not blocked. So what the migratory grazers do, they cross either Sanj River and then they go into national park in their old haunts towards Parkachi Meadow complex. So since there is no regular monitoring happening there, the grazing is still happening inside the national park. Although the administration may deny or people may deny, but it's present there. So all in all, uh, if we check the number of livestock and we do regular inventory of it, only then I think that we can regulate the grazing properly within both sanctuaries as well as national park.
Rahul, one question Mr. Ramesh asked you, and I want to ask you a question.
Yes, sir.
How different is the livestock number grazing intensity and distribution when compared to British Maharashtra and Law?
Sir, the work which they did encompassed, it was only focused on the national park region. So within the Tir, towards Tir side or towards Dhela side, okay, so the grazing is completely banned there. At that time, there were more than 30,000 livestock, both migratory as well as local ecozone people, ecozone livestock was also going there. In last 20 years, the people have started stall feeding because of the banning of rights in the national park. So right now the livestock coming inside both wildlife sanctuaries is approximately 5,000 to 6,000, and that's to the majority of it, 50% is from the illegal grazers, which are coming from either Ani or Luri. So yeah, towards from sand side, because the beat itself is quite big. It's 90 square kilometer area, and it's basically impossible to keep on. No.
The next question is from Dr. Amit Kumar. He is appreciating your work, great work. What's the average livestock herd size in GHNP? Both the current studies show comparable results in other parts of the
So average size for from herd size is about 250 to 500. It varies between that. And for gusjars, which bring large livestock like buffaloes and horses, it's about 60 to 70 per group, be it adult or calves.
That's all for you.
Thank you, sir.
The next question is for Pooja. Dr. Salvador is asking, since red pandas are known to be affected by canopy, did you investigate proper aspects of canopy, for example, structure? I'm not able to.
So, which I do great work. My question was that it is well known that canopy affects red panda and they are found in the canopy cover is very good. So, I was going to ask that, we will look at aspects of structure and how integrated is the complex in terms of that. With respect to canopy, that will be more ecologically insights into the micro and or the dwelling spaces of question.
Thank you for the question, sir. If we talk about the canopy cover or other variable estimation, canopy cover was basically at camera station was calculated like ocular estimation, oculary, wherein we just saw the canopy. See, when we talk about the bamboo species, which is very important for red panda, and the other canopy of slanting trees, which are the highest factor tree canopy. So when at lower elevation, we found we haven't collected like as you have mentioned, but what we found was the lower bamboo species variation in the canopy also, and the tree canopy variation also. So we have summed up that for the canopy cover. Thank you for the suggestion, sir. We'll definitely incorporate.
The next question is from Dr. Amit Kumar. is complementing your work, excellent work. Red panda can easily get come across in broadleaf, especially rhododendron crowding forest. You never know. He's asking, did you make any efforts in sampling rhododendron forest? Just curious.
Sir, if you talk about sampling red panda in the forest, we have done like arboreally also. We climb trees for their sketch collection or any indirect evidences. And there were a selected number of species, especially tree species, which were mostly favored. In them, the various rhododendron species were also favored for red panda, for its like basking, for its stay, for its as a latrine sites, what we call. Because if you go on higher altitude, the variation in the length and height of the rhododendron becomes a bit slanty, which red panda basically prefers. So it was most preferable for that.
So that is all for you. That's it.
Thank you.
There is no question for Pujan. I think sir wants to ask.
Pujan and both as a Puja. I have one question for both of you. How different is the work that you're doing now when compared with work done by Dr. Sunita Pradhan and Dr. Sarla Kali? How different is the distribution that we are getting now? Even if you look at the ongoing studies by other institutions and researchers in Sikkim or Arunachal, how does this your work is different or anything new that you've got?
Sir, if we talk about the studies done by Dr. Sunita Pradhan, which is baseline study on red panda from India, she has done in confined to the national park her PhD work, which was Singalila National Park of northern West Bengal. The methodology and the approach she used there in that time was conventional methods of trail surveying. The sightings or the indirect evidences, whatever were collected, were taken into consideration for analysis and providing results, and through that, the inferences were drawn. What we are doing for now is we have identified the habitat and the study area wherein we are surveying both protected and non-protected area also, which was confined there in her study to protected area only. And if we talk about the other studies and other organizations, what they are doing, if we talk about ZSI and other, the main approach they have is for genetic work, then to the ecological aspects and to know the species actual ecology in the wild, how its behavior, its arboreal nature and other. And about Sikkim, there also the conventional method of approaching is.
No, I'm asking the ecological studies. Simple cost distribution reports and your distribution modeling today. Is it different or you are more information? That's my question.
Yes, sir, definitely, because therein we are not confined, as I told earlier, to the protected areas only. We have taken into consideration the buffer area of the protected area also, wherein we have got even the sighting records and other from there. Thank you, sir.
Sir, that is what actually what I saw in the slides. Actually, most of the protected areas that are in, particularly in Arunachal Pradesh, they are like not very much suitable for red panda. Like most of the areas are not that much suitable for red panda. That is like we are doing work in like most of the areas are like unprotected. That is not under protected area network. So that is the I think that is what makes the challenging thing. Yes.
Okay.
Thanks.
I'm just told by my IT team that there are lots of questions on chat. I don't think we can take them now. And there are also other questions from many people here. Sorry, I did not ask them, but these questions will be given to the speakers and they will be going over the questions to everyone. So thank you all of you. You can all go back. We are running late, we are about 10 minutes, so I will request the coach to quickly sum up for this comments.
Thank you, sir. Good afternoon, everyone. So, frankly speaking, it was very nice to hear all the speakers and compliments to each one of you. And I was really happy to see you and yeah, I think also from this list that contribution in terms of science. It was really nice to see. And if you talk about in general that the current level of question, and even for that matter, if you talk about the patterns of diversity, it is still, you can say, I think you choose any road you have, unexplored, lesser explored, or underexplored. So that's true for even well-established areas of the country in general and Himalaya specifically. So I think it was very nice to see, studies all the way from East Himalaya and of course Darjeeling Hills. So once again, thank you very much for enlightening us with the knowledge. Thank you.
Thank you, Dr. Anil. I'll very briefly sum up my observations. If you look at the first 6 presentations, they were all under one project, like the National Mission on Himalayan Studies. So the ultimate objective is to try and connect and see how climate change will impact the species and their habitats. So 2 studies on mammals, just trying to establish baselines. All the students in this project and the other projects on Red Panda and Great Himalayan National Park, all of them have done work in some of the difficult areas in this country. So even going and getting a visual encounter or deploying a camera trap takes lots of effort in these areas. Interesting baselines on mammals, also on fish, on macroinvertebrates and soil nematode, and experimental studies being done on the alpine meadows. They are all just initial stage, first 2 years of work, and work by Rahul on pastoralism is a very interesting and important topic. And I think we need to look at the past and the present, and even the idea of saying that 30% we should reduce livestock, we should have some basis for saying so. So I think carrying capacity studies are important, but given the time frame, whatever he has done, I think we can try this experiment there. And the work on red panda is really interesting. I know it's very difficult to spot red panda, even in captivity in Palmaza Himalayan Reserve Park. You need to train your eyes to see these elusive animals. They've done great work, and I would say that this was a very interesting session, and a lot of information has come from these young people from the very remote corners of this country. I thank the research coordinator and my co-chair and the facilitator for giving this opportunity. Thank you very much.
Thank you, sir. Thank you, Dr. Satyakumar, Dr. Amit, and Dr. David Google for conducting this session. I now will straight away go for the second session. I now invite, this is on aquatic ecosystems. I now invite my colleagues, Dr. J.A. Johnson and Dr. Nehru Prabhakaran to chair and co-chair this session. I also invite Dr. Sunny Deoli to act as a session facilitator in this session.
Same answer. Just number नहीं डाला है.
हटा दो ना.
जी. नीचे नीचे.
Good afternoon. With the permission of the seminar chair, we will be proceeding to the next session, aquatic ecosystem. So from the Himalayan ecosystem, we are going to the aquatic ecosystem. As you are all aware, aquatic ecosystem is quite unique. In terms of understanding the species ecology, still the knowledge on species ecology and distribution is still the minimum level. Even though we have so many organizations dedicatedly working on the aquatic ecosystem, the reason is that the aquatic ecosystem is totally new to us. In the sense, it's alien to us actually. So we are part of the terrestrial ecosystem.
we know much because we are moving along the terrestrial ecosystem with atmosphere, air, protein, water, collecting information. We need a special skill, definitely. We need a special skill, special technique to understand the species ecology. So I hope that this session we have a wonderful 5 presentation. Definitely, I am hoping that they are going to enlighten and they are going to enhance our knowledge on the aquatic ecosystem as well as species ecology. And these 5 talks I have gone through, so all are species, covering all aspects of the aquatic ecosystem, right from the mountain, stream ecosystem, river and wetland, freshwater ecosystem, then again moving to the estuarine, then coastal ecosystem, then marine ecosystem. So it's going to be a very nice presentation. So here this session to facilitate, to assist with us, we have my colleague Dr. Deru as a co-chair and Dr. Shani Theory as a facilitator. So I'm handing over the session to Dr. Deru.
Thank you, sir. Without much delay, I would like to call upon the first speaker for the session, Mr. Vivek Ranjan, to speak about desilting of heritage wetland, assessing the wildlife values, potential impacts, and mitigation.
Vivek Ranjan, he is pursuing his PhD in human large carnivore conflict. He did his MSc in environmental management from FIL. Currently, he is working as a senior project associate in Heritage Project and Conservation Advisory at SSL. Over to you.
10 नंबर 10 नंबर है ना 10 नंबर है सत श्री अकाल सत श्री अकाल Punjab, the land of 5 rivers. However, after partition, the current Punjab state in India has 3 main rivers, that is Satluj, Beas and Ravi. And Harike Sanctuary is located on Satluj and Beas river confluence. And in 1952, to cater the needs of irrigation and water supply in semi-arid regions of southern Punjab and Rajasthan, Irrigation Department of Irrigation, Punjab started a project for making Harike Barrage in 1952, which was completed in 1957. And by the time in 1957, the total storage capacity of Harike wetland was 67,900 acre feet, which reduced significantly by more than 50% to 29,542 acre feet in 2023. The main aim of this Harike Barrage was to provide water and flood control in that area of the Punjab. However, due to the importance of wetland, in 1990 it was declared as Ramsar site, and finally in 1999 it was declared as Harike Wildlife Sanctuary by Government of India. This project was given to Wildlife Institute of India by Punjab State Government through Department of Water Resources to conduct a pre-desilting, peri-desilting and post-desilting phase. In 3 phases this study will be conducted. This study started in February 2024 and the current presentation is based on the preliminary study for pre-desilting phase. The main aim was to establish a baseline based on the ecological assessments, identify potential impacts of desilting and identify critical habitats in the Harike Wildlife Sanctuary. The photo here you are seeing is the confluence of Satluj and Beas. The darker is the Satluj and the murky one is the Beas river channel. The sanctuary, when notified in 1999, it was 86 square kilometer notified. However, when we have the official communication now, the sanctuary boundary provided by the Punjab Forest Department only covers 56 square kilometer. This discrepancy in the boundary was highlighted, and even in the 2017 when eco-sensitive zone was notified, that map is same as this 56 square kilometer map. Harike Sanctuary is located, falls in 3 districts of Punjab, the Taran Taran, Firozpur and Kapurthala. This is near to the Amritsar, 60 kilometers. It has a mosaic of habitats and number of ecotones, and broadly we have divided it in, it is divided in terrestrial, lotic and lentic system. There are 2 channels of Beas which divides upstream of the sanctuary before entering, and 1 channel of Satluj. These are the 3 main channels of Lotic system, and 2 reservoir areas, the LMB reservoir, which we say because of the left marginal bund on the southern flank, and this Riyasat or Kaliben. Kaliben is a smaller river channel which drains into this reservoir, and Riyasat is named after the Riyasat of Maharaja of Kapurthala. These are 2 important lentic systems, and the terrestrial systems are mostly the islands which has been formed due to siltation process and the embankment areas. To conduct this study, ecological assessment, we collected data from March to May in 2024, and for birds we used point count method at every 500 meter interval in terrestrial areas for 10 minutes at each point stations, and 1 river kilometer difference in aquatic areas. For mammal, we used camera trap based on 1 kilometer square grid, and as well as opportunistic camera trapping and boat survey in the aquatic areas. For herpetofauna, nocturnal visual encounter survey and opportunistic survey. For fish, we mainly depended on market surveys and trap nets in the shallow river areas or swamps and marshes. For vegetation, we use nested quadrate method for terrestrial areas. For trees, 10 meter by 10 meter quadrate, 5 by 5 for shrub and regeneration, and 1 by 1 for herb and grasses, and rake method for submerged vegetation, and 1 by 1 meter quadrate for floating vegetation. And we also had focus group discussion and interaction with the communities in Gurudwaras for socio-ecological information and their level of awareness towards the wildlife and conservation. We have almost covered more than 90% of the sanctuary through different sampling for different taxa components and socio-economic components. For bird abundance density, we estimated in R using and distance sampling for density and diversity at 78 point count stations, and NMDS was also estimated for the similarity and dissimilarities between different sites where the point count stations were located, and relative abundance, IVI of trees, regeneration, and LULC decadal changes from 1993 till 2023, and hotspot analysis to identify critical habitats both based on kernel density as well as spatial autocorrelation Morenci index. 187 species we recorded for birds, 13 of them are threatened and near threatened, and 20 scheduled 1 species. 56 were migratory, and the most diverse and highest species richness was found in lentic ecosystem. And sandy river banks, that is downstream of the Harike barrage, the highest species richness according to the different 11 sites, it was in the sandy river banks. And glossy ibis was the most abundant bird species in overall sanctuary, and pale sand martin in lotic system, and rose-ringed parakeet in the terrestrial system. Sandy river bank is distinct from other sites based on the bird species composition, and the total abundance is highest for lentic system. However, lotic and terrestrial system has similar abundance for birds. River tern has the highest density among the RET and NT species, and glossy ibis has the overall highest density among all the 187 birds we have identified there. We also recorded 5 heronry sites. Interestingly, 4 of these heronry sites are in the tall reeds and grassland. That's Phygmites, typha, and other species of grasses were there, and 2 ground nesting sites. 14 wild mammalian species were also recorded during the 712 trap nights, and hawk deer was the most abundant wild mammalian species. During 199 kilometer of and more than 199 kilometer of our boat survey in aquatic areas, during all the times we only sighted dolphin twice in the Beas River channel, and we recorded 2 endangered species, the hawk deer and Indus River dolphin in Harike, and 2 vulnerable species, sambar and smooth-coated otter. We also have location data for den sites of smooth-coated otters, where family of 5 to 6 individuals are there. and 11 of these species are Schedule 1. 18 species of herpetofauna, 5 are amphibian and 13 reptilian species. Ghadial reintroduction program is being conducted by the Forest Department, and from 2017 to 2021 they have released 94 ghadials, and only at 2 locations we have sighted ghadials in the sanctuary and 1 location outside the sanctuary in the canals. And most of the, as per the secondary information, most of the ghadial sightings are in the Byas Conservation Reserve, that is upstream in the Byas River channel, upstream of the Harike. This photograph is from the Satluj River channel, the ghadial sighting. This is very interesting because whenever the Byas River channel swells, the discharge increases in the Byas and the water quality improves in the Satluj. Then only the ghadial is sighted at the location where this transition of polluted Satluj water with the Byas water. And other times it is not situated, and there is no bare areas where preferably the ghadial basks, and however it is basking on the water hyacinth in that area. 25 fish species, Cyprinidae was the most represented family, and 4 fishes were vulnerable and 1 near threatened. You can see this is a chitla fish, which we found near a heronry site, dead in the polluted water of Satluj. We recorded 23 tree species with good diversity of 2.54 Shannon diversity, and total 288 quadrates were plotted. Lucina leucocephala, which is also one of the important invasive species. However, it was planted by the Forest Department earlier, and it is the most dense and dominant tree, and top 5 dominant trees are given here in the table. And tree health is good, but however, regeneration is lacking, and 64% of species show no regeneration and 40% shows poor regeneration. We have recorded 10 floating species in Lotic and 4 submerged species. Spirodella was the most prevalent floating in Lotic system, and Hydrilla in 43% in submerged, and 8 floating species in lentic and 7 submerged species, with water hyacinth as the most prevalent species in lentic system for floating species, and Vallisneria in submerged. We also checked the association between water depth and submerged species. So in both the Lotic and lentic system, it shows negative association. And 10 invasive species we have recorded from the Harike Wildlife Sanctuary during this survey. And when we see the LULC changes from 93 to 2023, major threat here in the sanctuary is encroachment for agricultural purposes. And the yellow areas shows the area which have been encroached, which was not, and changed, the land use have changed to agricultural areas. In the upstream part of the sanctuary, agricultural encroachment is a major threat, and most of the areas, every year it is increasing day by day by burning the tall grass and reed areas and adding the crop fields. The larger the island, the more preferred for encroachment by agricultural purposes, because it is more beneficial economically, because they bring in the harvester through boats, and which I will show in the pictures ahead. To identify critical habitats, spatial autocorrelation was not very significant for RET-NT species only based on the abundance. So we used kernel density estimation, and we can see the red areas, the high priority areas near the confluence of Satluj and Byas River. And based on the species richness at each 78 point count stations, bird species richness, it was significant clustering was seen in the second right picture in both the lentic systems, LMB reservoir and Riyasat reservoir areas for high richness area of birds. For hog deer, it shows significant clustering, spatial autocorrelation in ArcGIS, and it shows the high priority area near the confluence and in the terrestrial island areas near the confluence of Satluj and Byas River. In this picture, first top left, this is the aerial shot of lotus. This is complete lotus. It is spread in hectares and hectares of areas. It was the highest possible elevation for drone to take this picture. Still, it was not covered in one frame. And this is from the Riyasat reservoir area. The second top right, it is water hyacinth, extensive spread of water hyacinth in the sanctuary. And this is not also navigable by the boat. Thus, much larger area of the sanctuary we are not able to survey or sample. The bottom left is the submerged vegetation area which is, and the right is the Riyasat Reservoir aerial shot, where we can see the shrinkage in open water areas, and these are the major reasons why it has the storage capacity of the wetland has decreased drastically in. And these are the other major threats: erosion of banks. You can see 2 tractor trolleys being loaded on the floater and from the island, and these vast agricultural fields. And even on the mainland sites, every year this road increases day by day, getting into the sanctuary. And there are few major, I have highlighted only major potential impacts for positive. Definitely it will increase the storage capacity if the desilting is carried out, and it will enhance the flood control, removal of invasive species, and important negative points are the habitat alteration, definitely displacement of species where it will be taken out, desilting activity will be carried out, loss of habitat heterogeneity, disturbance due to noise. It will affect the ecology, existing ecology. It might also increase erosion if a catchment area treatment is not properly being taken care of. So, based on this identification of critical habitats, and we have marked 11 no decile zones for the sanctuary out of 56 square kilometer, covering 16.6 square kilometer area. These 11 no decile zone will be completely avoided for any kind of desilting activities, with a buffer of 100 meter on all sides of these no desilting zones. These no desilting zones are marked where our hotspot analysis shows the high priority areas, and where the submerged vegetation is also very important for microinvertebrates and small vertebrate species for breeding and other foraging for birds and everything. So those areas have also been marked in the lentic ecosystems as a no desilting zone. And these are the few shots which we want, aerial shots of the area and habitat which we want to protect and marked under the no desilting zone. The first top left is the confluence of Beas and Satluj. And few other prescriptions which we have given as precaution and mitigation. Timing and scheduling is very important, since Harike is famous for its winter migratory congregation, and it falls on the Central Asian Flyway. And thus we have suggested that October to February should be completely avoided for any desilting activities or preparation for desilting activities like pontoon bridges or anything. And night time should be avoided throughout the desilting process if it is taken up. And seasonal planning, that winters should be avoided completely because it's an important period. Sediment management is very critical, because if the desilting has never been carried out in this sanctuary, and from 1957 till 2023, and if it is carried out, its long-term viability and efficacy depends on the catchment area treatment, which is the source of like silt coming into the sanctuary.
Please conclude in 30
Yes, sir.
seconds.
Yes, sir. This is the last slide. And continuous water monitoring for adaptive management practices, and number of several site-specific recommendations we have also provided. I would like to acknowledge the following people for their guidance and support throughout the preparation and conduction of this study. Thank you all.
Thank you, Vivek. Now I call upon the next speaker, Saurabh Chaudhary, to talk on amorphs, habitat and stream correlates. Can we see indicator values?
Saurabh has done his master's in wildlife sciences from Manipur University. He is currently working as a project associate under the supervision of Dr. Sundara Rindu, and his interest includes ecological aspects of Hapuda. His hobbies were traveling, photography, doing music.
Good afternoon, everyone. Am I audible to everyone? Okay. So now going through all other taxa, mammals, nematodes, and everyone. Now we will dive into the amphibians, the anurans, and also into one of the rare and endemic jewel of the west of the Himalayas. So here in this image you can see that that is my focus species, Amelops formosus, and here we will see that how this species can be used as ecological indicators for the stream ecosystem and health in the Himalayas. Right now I'm checking for the Western Himalayas, that how it is dependent and what are the correlations. So moving on. So first, to initiate, I want to tell you guys that what are the ecological indicators and what are their importance. So the ecological indicators are the major information providers that let us know that how the habitat, ecosystem, streams, everything is changing over the time, and whether it is degrading and how much it is degrading. They can give us insights into all those aspects. So there are different type of indicators: biological indicators, chemical indicators, which includes pH and all, biological indicators which use species and all, which is one of my topics. Physical indicators like the flow, the canopies. Habitat indicators include that what is the habitat type, vegetation and all. So moving on to the next slide. So what is the importance of these ecological indicators? That is the information they provide, but what is their importance for the long term? So for long term, they can help us monitor these ecosystems and their health over the time. So we can know that over like 5 years back, was the stream good, and was this stream good right now, or it has deteriorated over the time. Second, they can let us know and make a proper decision making goals. Decision making, they can help us to know that how much there are hundreds of streams in Himalayas, but a lot of stream needs to be protected, which we can get into the insights because of these ecological indicators only. And they can also let us conserve both the species and the ecosystem side by side. So first, coming to the introduction of my species. So here I found basically 2 species my study area, both from Amelops genus. So my major focus was Amelops formosus, which is a hill stream frog or a cascade torrent frog, which is found normally, on record it is found from 1000 to 2500 elevation, but what I have recorded is basically found from majorly found from 1600 meter to and about till 2500 meters. And these frogs are highly or closely related to the streams. So that factor can help us know that whether they can be a future ecological indicators for these streams or not. And one more interesting fact about this species is they have natural color pattern. So we can even identify each individual and know about the population, their movement, their distribution. Then we can correlate it to the stream ecosystems and parameters. So going back to that only, I propose the 2 study rationale that why I need to do this work, because one of the factors, the population dynamics, is one of the important factor that will give us information about all the that will give us information of the species. Then only we can go into further detail that how the population is being affected. Second is the association of these species and population with the stream parameters, which include the chemical parameter which I earlier said, pH, conductivity, salinity, and even the canopy and boulder sizes, water flow, etc. So based on that, I used 2 objectives. First, for the population, I used the SCCR linear. As the species is more or less confined to the stream habitat, so I moved with the SCCR linear approach to know that how much species are there in the per kilometer length of the stream, rather than by the hectare areas. Second, that what is the association with all different stream parameters. So here you can see the Himachal map, and these are few of the study sites that we selected initially to do the work. And because there is not much literature available about the species, so it's more of a go and see the approach also, whether that, because streams are present everywhere, but the presence of the species is there or not. So we selected multiple sites across the Himachal, and out of this presentation based on three sites: the Chudha Wildlife Sanctuary, Shimla Catchment Wildlife Sanctuary, and Bandli Wildlife Sanctuary. So first, this is the map of Chudha Wildlife Sanctuary. Here I sampled 3 streams where the species was found, but there were still 3 more streams which show the absence of the species. And in Shimla Catchment, according to their proposal, they have almost 16 streams, but I found the presence of the species only in the 4 streams. And lastly, Bandli Wildlife Sanctuary, although being in a elevation range, the species was the water catchments were all to the all over the boundary regions of the Bandli. So Amlops formosus was not present, but the different species Amlops johnsari was present in this area. So here in this slide you can see on the left both the species Amlops formosus and johnsari and the habitats they prefer, or the habitats known to be where they are like abundant. So here in this what procedure I used for my field work, I sampled the 400-meter length of the stream in a 50 by 20 meter grids, continuous grids for SCCR approach, and visual encounter survey was done for the admixture abundance and the covariates. So what were the procedure to do that, because it is more of a, as I earlier told, unique pattern species. As you can see in this photo, every individual has a different unique pattern. So what I used, without physically disturbing the species, I photographed every individual, and on 6 alternative days, and checked that what were the recaptures and what were the and through that recapture I went into the density analysis. So along with the photographs, I collected all the stream covariates. Then we analyzed the data for the abundance estimation and density estimation using SCCR linear, and then we went what is the association of the abundance and density to the stream covariates. So here is the one of the initial photo results, like from Chuda Wildlife Sanctuary and Shimla catchment. Both the locations, every stream, in Chuda, 3 streams were sampled, 6 replicates were done on every stream, out of which 148 photographs were captured, and all those streams, all the 3 streams I have written the number of individuals I encountered, like 22 in Chora stream, 36 in Kanda, and 18 in Rondi. And in Shimla catchment, I sampled 4 sites, 4 streams, in which very less photographs were captured. Although the Shimla catchment wildlife sanctuary, while entering the sanctuary, you feel like it is one of the pristine habitat but which was quite shocking. I found very less individuals in the sanctuary. Now coming to the Johnsari in Bandli, because as I earlier mentioned, Amlops formosus was not present. So Bandli, Johnsari, there were 6 streams that I sampled, in which 328 photographs were captured, and number of individuals I have mentioned what all the number of individuals were found in different streams. So here you can see the abundance results of 3 different all the 3 protected areas. First for Chuda Wildlife Sanctuary, second Shimla catchment, and third Bandli. And species I'm not comparing, I'm just giving you the insights ki how much species were found in my major protected areas. And you can see the difference between my concerned species in the Chuda Wildlife Sanctuary and Shimla Catchment Wildlife Sanctuary. So coming to the spatially explicit capture recapture linear one. So going through that, we got the density in the all the 3 streams of the Chuda Wildlife Sanctuary. Here you can see the density of the individuals in 3 streams to be 84 individuals, 92 and 78, which is a quite good number of individuals compared to per kilometer of length it is. And same thing if we go to the Shimla catchment wildlife sanctuary, you can see the density decrease a lot, like 18 individuals per kilometer, 12 individuals, and in one of the stream only 2.7 individuals. And same if we go for the Johnsari in the Bandli, here you can see that one of the stream showed a higher number of higher density, and this stream was also one of the undisturbed stream which was on the outside of the protected area, not on the boundary or nearby. So now coming to our major insight, that what were the major correlation with the stream parameters. So our major species Amlops formosus showed a negative correlation with the flow stream. This is one of the interesting findings we found, because all over the time species has been known to dwell in a high flowing streams, but it is not actually positively related to the flow of the stream. The thing is that it needs an optimum flow, which is approximately 0.8 meter to 1.6 meter per second for its highest number of abundance or density. But as the flow increases, their in movement and their encounter decreases. And one more thing we found, the boulder size. we found a positive correlation with the boulder size. As the boulder size increased, a larger number, like because it is Himalayan stream, there are huge boulders. So those boulders played a quite good role in the presence of the species. And thing was that this was our focus and all. We were not that much concerned that how Jonsari is responding, because it is more of a resilient species in the Himalayas, lower Himalayas. So, but same results were found with the Jonsari also. It showed a negative correlation with the flow and the positive correlation with the boulder size. So this let us know how the streams, like over the time, especially in Himachal Pradesh, over the time how the streams are being channeled to all the villages, and people are getting it, people are polluting the streams, a lot of tourists and all, and how those things might impact these species, which I found after this, because I'm going to show you how this habitat alteration has led to that. On the left side, this is the Churdhar Wildlife Sanctuary streams. You can see there is no disturbance, no human intervention, streams are clear. But on the right side, where we found a very less number of individuals, Shimla catchment, here you can see there are a number of check dams and water holes that has been made to provide water to the human habitation, to the main Shimla. So that is the main reason that is impacting. There are still more analysis that needs to be done, like the pollution, which I have collected data but didn't do the analysis till now. So this all the procedures and all the works, what it gives us the insight, like for Amlops formosus is it was in higher abundance, higher density in Churda, and Jonsari was in higher abundance, which was also in the area which was away from the human interaction and less disturbed. And the thing is that both species showed a negative correlation with the flow, although being always known as a high flowing stream, and showed a positive relation with the boulder size, which is which I think which needs to be more analyzed, because boulder size along with the temperature and canopy is also I think playing a role, especially in Amlops formosus, to be present along with the sunlight that is passing, because the amount of sunlight passes that increases the temperature, and as the species is nocturnal, it is not found in the day, but that temperature is impacting the species by the night time. So what is our way forward? Like I told you, this is the presentation. This I present only 3 study sites, so there are still more sites that needs to be covered, which can give us more detailed information. And also these SCCR approach and linear approaches can be used for more different species of the Himalayas also. In the upper Himalaya, like Buphotis also can be done by this. And second, the third main insight is like this ecological insight, which I showed you right now. This can be done on more finer scales to show us that how this species specific to these Himalayas, and whether this genus or the species, can it be used as ecological indicators in future also. And this insight can help us to even properly plan to save the species and the stream ecosystem, which is one of the lifeline of the Himalayas. So thank you. Recently also one of my paper was published on the same aspects of the Amlops, and the same work was also presented in WCS 10. At last, I want to acknowledge Director of Wildlife Institute of India, Dean Wildlife Institute of India, and JICA for funding the project, and Himachal Forest Department with all their rangers, field assistants and forest guards, and my colleagues for letting this work being done as it is being done. So thank you.
Now I call upon the last speaker for the morning session, Diksha Mandlik, to speak about diversity of selected taxa of the Goa coast.
Diksha did her master's in marine biology from Pondicherry University and in marine environmental protection from Bangalore University. She has also worked previously with IISCR and CDP. She has joined the brilliant as well as
Thank you for the kind introduction. Good afternoon, everyone. I am Diksha Mandlik, and I am presenting about diversity of selected taxa of the Goa coast. This is a pilot study, part of the bigger project, which is Coastal and Marine Biodiversity Profile of the Goa state. And for this study, as this is a pilot study, we have selected 2 taxa, and the data has been collected for 2 months, which I'll be presenting now. So India's coastline has a very rich diversity of habitats. It supports.
Can you just go closer to the
Yeah. Am I audible now? Right. So India's coastline has a rich diversity of habitats, and it supports various specialized habitats, including coral reefs, mangroves, and seagrass meadows. East coast is predominantly sandy, but when we go to the west coast, it's sandy, rocky, and also marshy areas in mangroves. So rocky shore is quite critical when it comes to ecosystem, because it's a critical interface between marine and terrestrial ecosystem, and it also withstands a lot of stress and harsh conditions due to the fluctuating variables, including salinity, temperature, water availability, and nutrient availability. And due to the presence of water, high tides, low tides, there are distinct zonations found in the faunal assemblages, which can be vertical and horizontal. And as this is an interface between 2 habitats, 2 large ecosystems, it's quite susceptible to anthropogenic activities as it's easily accessible. And a lot of species that are commercially important are found here, and thus it's ecologically crucial. So Rocky Shore has huge boulders or tide pools, which are inundations in the rocks, and they can be partially or completely covered during low tides. So we have flora such as algae, lichens, and fauna such as sponges, sea anemones, polychaete nematodes, which are the marine worms, crustaceans, mollusks, and echinoderms, as well as tide pool fishes. So Goa, which is our study site, it's the smallest state falling on the west coast of India, and it has a coastline of 105 kilometers. For administrative purposes, it's divided into 2 parts, which is northern and southern Goa, and it's decided by the flow of river Zuari. And predominantly, it has rocky coast with sandy pockets, and it also shows a lot of specialized habitats like coral reefs, mangroves, etc. So the taxa that we have surveyed, which are mollusks and crabs, it's due to their abundance and the throughout the shores, and also they are ecologically important because they have a huge impact on the fisheries from the state. There are 130 genera and 39 families of brachyuran crabs that are true crabs. The anomuran crabs, the studies are still undergoing, so we don't have a confirmed number. And from the estuarine areas, there have been several new reports of mollusks and crabs from Goa. There are 86 gastropods and 19 bivalve species found. And as Goa is a very famous tourist spot, it is very susceptible to tourism and thus the pollution that's incoming as well as climate change. These are our 4 sites, Anjuna and Baga, they fall in the northern Goa, and Rees Magos and Holland, they are in southern Goa. So all of these are again rocky shores with sandy pockets, but the thing with Baga and Rees Magos is that they are creaky, they are marshy because there are creeks flowing nearby. For crabs, our methodology was that we laid a transit parallel to the waterline, and we took 3 replicates of that. This transit was 10 meters in length, and crabs were counted along the transit on the both sides by visual efforts. And the specimens, a few of them representative ones were collected and the others were photographed. The collected specimens were preserved on the site, and all of these were later identified using species identification guides. For mollusks, we had a line transit of 10 meters laid parallel to the water line, and 3 replicates were taken. Then five replicates of quadrates were taken on alternate sites, and the mollusks were counted. The mollusk species were counted in the quadrats, and few of representatives were collected. All the pictures and specimens were later identified using species identification guides. These are some of the crab specimen that we collected. So we found 16 species of crabs belonging to 12 genera and 7 families. And Rees-Magos showed the highest abundance of families, and Portunidae dominated with 3 representatives for the family. Highest abundance and species richness of crabs was found at Baga, and Charybdis helleri and Graspus albolinatus were leading. They were found at every station and in very high numbers. When we look at the Shannon index, Rees-Magos has the highest Shannon index as well as the species evenness, and Graspus albolinatus was the dominating species in all the stations, but also contributed to these high numbers. And we think there are reports that these species, Graspus albolinatus and Charybdis helleri, they are quite adapted to the mechanical stresses that sandy shore puts upon them, and better suited to the hypoxic conditions to their high abundance. Also, Rees-Magos, the interesting part about it, that it's a heavily polluted estuarine area because there's a poorly managed waste facility just near the shore. But it also supports high crab diversity, which could be because of the ability of these species to survive the stress, hypoxic conditions, and high nutrient load. These are the mollusk species that we found. There were 42 species of mollusks, out of which were 30 gastropods and 12 bivalves, from 37 genera and 18 families. Baga had the highest family count, and it was at all of the stations, it was dominated by the family Veneridae, which has clams. So Holland has the highest abundance, whereas Baga and Anjuna showed highest species richness with 19 species each. And Planexis sulcatus and Gyranium natator were the species that were found at every station in very high numbers. Mostly Planexis sulcatus was very abundant everywhere, which gives the number for Holland. When we look at Shannon index and species evenness, Anjuna was the leading site, and because Planexis sulcatus was present there in high numbers, it's better adapted and had better survival rates. And the high abundance and species richness at Anjuna and Baga could be due to the fact that it has large boulders and rocks present throughout the coast, and that provides niches and stable habitats, which gives opportunity for the species to attach to and have a lesser stressed niche. In conclusion, our study shows that there's high crab diversity in Rees-Magos and highest mollusk diversity in Anjuna. Also, Rees-Magos has high Shannon index value and species evenness, which says that the resources at the site are better distributed, and the species are, there's no one single species that dominates it, but it's better utilized by the species. Same goes with Anjuna. So the biogenic structures and bioturbation processes which aerate the sand and it helps in nutrient cycling, and this is done by intertidal organisms which control the ecosystem health. And crabs and mollusks are abundant, are some of the most abundant. So they definitely are great indicators of ecosystem health. So for low diversity sites, restoring habitats is very crucial so that overall ecosystem health could be improved. And diversity profiling studies such as ourselves, they help us understand what could be the potential biodiversity hotspots, and for lesser or low diversity habitats, they can provide management and conservation suggestions. Interestingly, we also studied the cetacean strandings during the study period, which was April to August 2024. And total of 27 cetacean strandings were reported. 10 of them were from north and 17 of them were from the southern coast. And out of them, 12 counts of porpoises, 14 counts of dolphins, and just 1 count of Kogia sima, that is the pygmy dwarf whale, was seen. The standings are very essential to look at when we are profiling any area for biodiversity because it gives a real time data on the species count, species presence and abundance at the shores. And very interestingly, 9 of these strandings during April and August were reported from this Varkas, Zalor, and Kavalosim pocket, and which should be further looked into as a potential seasonal stranding hotspot during pre-monsoon area. Further, as we go along with our study, these are just 2 taxas that we studied in our pilot study, but we plan on studying and seasonally monitoring these habitats. A few of them were the flora includes algae, seagrasses, mangroves, and others like corals, poriferans, marine worms, mollusks, crustaceans, etc. And for that, we are going to use line intercept transits, underwater visual counts, boat surveys, and as our study would be seasonal, our study would look into the seasonal aspects as well. We'll look into more environmental variables. I would like to acknowledge these people, the director, the dean, the registrar, and the research coordinator of WII, Forest Department of Goa, which is our funding agency, Mr. Pranay Sawant from NIO, who helped us immensely during our field work and post identification studies, Ms. Pooja Mitra from Terra Conscious and Drishti Lifeguard Network, who provided us standing data, and Girish Gaonkar, who was the local field assistant. Thank you.
Thank you, Diksha. Now I request the audience and students to please send your questions to the dais quickly. So most of the questions now we are receiving from the faculty side only. Even the previous session also we received more questions from the faculty side. I encourage the students and researchers to ask more questions. So with this, we are now breaking this session for lunch, and we will be reassembled around 2:30 again. Over.
Yeah, just one announcement. Lunch for all research scholars and students have been arranged in the lounge of old hostel. For officer trainees, faculty members, and our IT and audiovisual unit staff, lunch is at the old guest house. So enjoy the lunch, do justice to the lunch, and give me the feedback if anything needs to be improved. Please come back at 2:30.
दबाने लग गया। कर रहा हूँ, देखो देखो दिख रहा आपको? हेलो देखो।
हेलो। हाँ हो रहा अनम्यूट है।
Kunal Gokhale, who will be talking about population estimation of fishing cat in Koringa Wildlife Sanctuary.
Kunal has done his master's in geology from Pune University. He is currently working as a project associate in the project Ecology and Conservation of Fishing Cat in REE and Andhra Pradesh. His interest lies in ecology and behavior of small wildlife. Over to you, Kunal.
Thank you for the introduction. My topic is population estimation of fishing cats in Koringa Wildlife Sanctuary. So let's come to the objectives. As the title suggests, the objective is to estimate the population density of fishing cats in Koringa Wildlife Sanctuary. And the secondary objective is to understand the activity overlap of fishing and with other mammalian fauna. So let's talk about basic ecology of fishing cats first. Because of various threats, fishing cats are listed as vulnerable according to IUCN norms. They are highly reliant on mangrove habitats, wetland habitats, and marshland habitats. So the major part of their diet includes fishes, and then the secondary choices of prey are rodents, birds, reptiles, crustaceans, and amphibians. The threats includes degradation of habitats because of human settlements, industrialization, and conversion of their habitats into agricultural lands. Unsustainable fishing or overfishing, which is also happening in our study area, Koringa Wildlife Sanctuary. Then poaching and hunting. So this is the global distribution map of fishing cats. Fishing cats are found in countries like India, Sri Lanka, Pakistan, Nepal, Bhutan, Bangladesh, Myanmar, Cambodia, and Thailand. So study area. Our study area is Koringa Wildlife Sanctuary. It is spread over 235 square kilometers. It is situated in the East Godavari district of Andhra Pradesh at a place where Godavari River meets Bay of Bengal. I'll come to the methodology. So to understand the ecological challenges related to accessibility and tidal wave inundation, because our study area is along the coastline of east coast of India. So to understand the tidal wave conditions, we did initially Reiki survey. After Reiki survey, we did a camera trap session from March to June this year in 1 square kilometer grids. Total trap night effort was 2,279 trap nights. So using this data from camera trapping, we estimated population density of fishing cats using two methods. First is conventional capture mark recapture, and second is SECR linear, which I'll talk about later. And we also generated heat maps using our package P heat map, and also showed their activity overlap with other mammalian fauna in Koringa Wildlife Sanctuary. So now I'll come to the second method of population estimation, which is SECR linear. So why SECR linear? Now you can see in our study area, the grid size is 1 square kilometers, but all the camera trap locations are along with the streams of the rivers Koringa, Tulibhaga, and Gaderu rivers. So because of the accessibility issue, we could only deploy camera traps along the streams of these three rivers. So camera traps were only deployed along the streams. So this resulted into a trap layout to be in a linear manner. So our package SECR has an extension called SECR linear, which is used to estimate the population densities of animals in linear habitats. So these are the results. So the effective sampling area was 67.5 kilometers. So in SECR linear, the area is the area is linear, so it is calculated in kilometers, it is calculated in length. So number of individuals we identified were 50, unique individuals was 52. The estimate was 92 and the density is 77 per 100 square kilometers. So the sigma derived from our results is 855 meters. So to compare, we compared our sigma with another study of telemetry study on fishing cats in Thailand. So from their data, we derived sigma as 1,520 meters. So there is, you can see there is significant difference between our sigma and the study telemetry, the sigma in the telemetry study of fishing cats in Thailand. This difference, the cause of the reason behind this difference, possible reason might be that the density in the Thailand in that their study area is lower than our study area. So the home ranges must be higher. So the sigma value is relatively higher in their study. Now these are the results of capture mark recapture. Again, we had identified 52 unique individuals, and effective sampling area of CMR was 63.7 square kilometers. Estimate is 56 and density is 87 per 100 square kilometers. So we generated activity overlap graphs also. Fishing cat only showed significant activity overlap with only 2 mammalian species, which is jackals and rodents. So these are the heat maps which are indicating the presence and distribution of mammalian fauna in Koringa Wildlife Sanctuary. So jackals are the most abundant and widely distributed species in Koringa Wildlife Sanctuary. And fishing cat presence, you can see it is mostly in the southern region and the northern region of the sanctuary. Smooth coated, we got only captures of smooth coated otters from only 2 camera trap stations. And so this is for the comparison. And conclusions. So we got a very large number of fishing cat captures. So this highlights the importance of Koringa Wildlife Sanctuary as a major stronghold for the species conservation. We got captures of only 7 mammals from our study area. These were fishing cats, golden jackals being the most abundant species. We also got the second one, which is more abundant, was rhesus macaque, and then fishing cats. We got few captures of wild pigs also, which are introduced in the sanctuary by locals, and only 1 capture of Indian palm civet, and more than 100 captures of rodents. So only few studies of, we got only few captures of fishing cat from our study area. So this emphasizes the need to carry out studies related to the distribution and the population of smooth-coated otters in Koringa Wildlife Sanctuary, or at least in the streams of or the catchment areas of Tulibhaga, Koringa and Gaderu rivers. We also conducted questionnaire surveys related to conflicts with the local fish farmers, which are present in the western part of the Koringa Wildlife Sanctuary. So I haven't presented it here because the data related to that was not sufficient. But whatever data we had, it indicated that there is not much conflict with fish farmers. The results say, the questionnaire survey indicated that fishing cats are entering into fish farms, and they are damaging the fences which are on the boundaries of the fish farms. Jackals are also doing the same, and they are catching or preying on the fishes from the fish farms also. But it is not causing any killings or poisonings or electrocution of fishing cats or any other animals. in the fish farms. So I would like to thank all these entities. I'll play one short video which is showcasing one interesting behavior of fishing cats. It is scent marking its territory. Thank you.
Thank you, Kunal, for being very, very kind. Next, I call upon Abhishek Dutta and Joshua Dhanuraj who will speak about plays and rituals on the way. Special control of plays and equal artistic monitoring efforts.
Abhishek has done his engineering from VT University, Karnataka. He did his master's in wildlife science from Wildlife Institute of India. He joined WI as a project associate in the year 2019 in Kappa Dolphin project. Currently, he is working in IDWH Humpback Whale project. His interest lies in understanding the citizens. Over to you.
Thank you for the introduction, Dr. Sanjay. Good afternoon, everyone. To begin with, oceans are the largest ecosystem which holds and nurtures few of the largest species of mammals. And coming to their assessment, it is very difficult because compared to the terrestrial ecosystem, the difficulty in the oceanic system increases. And this story is about one such organism, one such creatures, which are known as the gentle giants of the oceans, Arabian Sea humpback whales. With this, I would like to introduce our talk, Tales and Whispers from the Oceans, giving a brief introduction on the species. It's a subpopulation of the Arabian subpopulations, which is restricted to the Arabian Sea area, and it is distributed along the coast of the Persian Gulf, including Pakistan and India, and also it extends up to the Sri Lankan and Maldives islands. And their current status is about approximately 25 mature individuals across the globe. That is restricted to Arabian Sea. Which puts them in an endangered list globally, and it is protected under Schedule 1 of Wildlife Protection Act. And since it is restricted to Arabian Sea, it has a very distinct morphological characteristics, which is the fluke pigmentations, which is very distinct from the rest of the populations. And genetically, these are unique population because they have been separated around 70,000 years ago from the rest of the populations, and they don't migrate when compared to the other populations which do between the hemispheres. And coming to their acoustic vocalizations, even compared to the other populations, they are acoustically also isolated, and the singing patterns are much more unique. All the Arabian Sea singing patterns are much more unique, and it has a very small sample size of males which vocalates. And since humpback whales are known to have a cultural exchange of songs when moving between the populations, since these are not moving between the populations, so there is a less improvements of vocalizations, and their songs remain same continuously over the decade. And this also has an acts as a effective barrier and also has an inbreeding thing. And what we don't know is what is the current status in India and their sightings or movement pattern across the Indian coast, and also majority of the work is being done in the western Gulf coast of Arabian Sea, and data is deficient in Indian waters. So with all these things, we focus on addressing our first objective, which is trying to understand their occurrence pattern by integrating the data from the Fisher Folks survey in the Indian coast. So we did a sociologist survey from the Fisher Folks of the entire west coast of India, covering around 6 states, and we did we did an assessment where marking from 200-kilometer buffer from the coast with a grid cell of 20 by 20 kilometer grids, and around 28 fishing harbors were selected and surveyed, which around 2,500 numbers of fisher folks were surveyed, which around 1,876 positive response came up with the Arabian Sea humpback whale sightings. So with this data, how we actually interviewed is we had a catalog of representing all the baleen whales in the Indian coast, and with this we have an unique identifications for the humpback whales because the pectoral fins and their full body breach is a unique identification characters. With all this, we asked the fishermen folks where they are sighted from the coast and what is the distance of them, and we approximately took the grid cell of that area, and the last sighting of was recorded and the season of it was also recorded. The right side map indicates the occurrence patterns of the humpback whales across all the seasons, and mostly every state has an hotspot of where the humpback whales are sighted in most of the seasons in a year. So the fisher folk interview had all the demographic variables also, but to validate their interview, validate the Arabian humpback whale sightings, so we gave we actually took 5 variables from the interview and gave the weightage based on the number of crews which the fisher folk was surveying, and the experience which the interviewer had, the fisher folk had, and the days of fishing, and also the distance from the coast which they are monitoring, which they are fishing actually, and also the identification cue which they use to identify the humpback whales. Collating all this, we had a grid-wise extract for the last 5 years of data, and with that we extracted a seasonal oceanographic data from Copernicus. The 5 variables were chosen which indicates the primary productivity and also which influence the distribution of prey species, and it was the extraction was done for 4 seasons, which is summer, monsoon, post monsoon and winter season. And we did a generalized linear model to predict the perceived occurrence pattern in the western coast, and it was replicated to all 6 states and across all the 4 temporal seasons. So what we got was primary productivity. Chlorophyll was significant in sighting the humpback whales, and the probability of the occurrence increases with the increasing of the primary productivity in the region. So this is the predicted map. You can see in summer there is a shifting of the occurrence pattern towards southern regions, and the GIF images actually tells the productivity of the net primary productivity in the Arabian Sea region, and it's clear and followed by the monsoon regions. It is clearly showing that the sightings is clearly shifting towards the southern regions, which correlates with the Malabar upwelling zones, which is happening in the southern coast of Arabian Sea and where the Indian Oceans meets. And followed by the post monsoon, the sightings is shifting towards the northern regions, where the northeastern monsoon has an influence on these regions, and the productivity also increases in this region. You can clearly see in the GIF images, and also the sightings of the humpback whales which is moving towards it. And coming to the winter, it is completely shifting towards the northern region, and because we have limited with a 200 kilometer grid, the humpback whale also tends, it's a subpopulation which roams around the Gulf countries also. So the humpback whale sightings mostly tends towards the upwelling zones which is happening in the Gulf states. So when you see the overall pattern, how the shifting of the humpback whale happens in the entire all the 4 seasons in a year, it's showing that clearly that they are using the net primary productivity, the productive areas of the oceans. And there is a study from the Gulf countries also, where they have got a similar lizards, where they have actually mapped by tracking the satellite tagged humpback whales of around 9 individuals. They also have a clear shift movement from the movement from northern region to southern regions during these upwelling zones. And net primary productivity has a very strong influence in sighting the Arabian Sea. And as mentioned earlier, the vocalizations also is very important, and also it is isolated populations. So we try to hear what the whale says. And with this, our second objective comes, it's a pilot study to develop a passive acoustic monitoring devices and developing a uniform protocol to replicate across the hotspots. So this is a study area. From our previous maps, the Malvan region was selected because that was the sites where the sightings was high during all the seasons, and the study was conducted during the summer season. And we deployed around 6 hydrophones. Initially, 4 hydrophones were tested for managing the different columns, and this is the hydrophones which we use for recording. So how we deployed it? So initially we selected a site away from the coast. We did a bathymetry scan and to know the depth contours, and we selected the site of a particular depth, and we deployed it in the bottom and mid column of the entire of the ocean. So we actually for the bottom we deployed it using the below images. We can see we actually mounted it on a rock. But for the mid column, we actually measured and anchored it towards the bottom. So this is the results findings which we got. The mid column was the best zone, which reduces the noise from other benthic organisms. We recorded around 600 hours of data, and the last hydrophones recorded a sound of a baleen whales, and we got around 12 different occasions over 6 days, and the day 6 you can see has the highest call recordings, which is around 30 calls in a day. So with our current instrument, we could with the sensitivity of the instrument, we could gauge that the sound was produced. The source of a sound produced was around 1,200 meters from the hydrophone where we are deployed. So this is the average frequency of the calls of each day calls which we have recorded, and it ranges from 100 to 700, the average ranges, but the entire ranges was from 100 to 1500 kilohertz, and this is the wave patterns of sound was of time versus kilohertz graphs, which shows how the sounds were look, and total of around 3.43 minutes of sounds was recorded in a single hydrophones, which had around 192 hours of recordings. The pattern which we got was downward shifting pattern of sound waves from higher frequency to lower frequencies, and there was a humpback whale recording also done near the Karnataka, near Malva, which is 200 kilometers away from the Malvan regions, but it doesn't match the sound which we got doesn't match with the humpback whales, but it has a potential patterns of a bright whale, but there is a huge frequency shift in it, so we are not sure of what whale it is, but we are sure that it is a baleen whale. And this is a acoustic recording. If you can hear, this is a very faint acoustic recording we got. So concluding all of this, the seasonal distribution of the humpback whales is influenced by the oceanic process, and also the net primary productivity is one of the factors. And for effective long-term monitoring, it is crucial to synchronize our passive acoustic monitoring devices with the seasonal occurrence. And deployment at the mid column was increases our likelihood of capturing the vocalization because it reduces the background noises. And way forward, the same work will be replicated in the Lakshadweep Islands, because that is the sites we are missing right now. And all since we have done a very pilot study, and the remaining remaining states will also be monitored, where the potential sites of humpback whales has been cited. And for long term, we don't have any sufficient data. So for long term monitoring, satellite tracking, surveys, and passive acoustic monitoring will be done to understand their ecology. With this, I would like to thank the following people, and thank you for your patience hearing.
Thank you.
Now I call upon all the speakers to speak for the question and answer session. Please officially say that. Okay. Vivek, the first question is there's a comment from Dr. Raman Kumar. Very comprehensive study on a very important aspect of wetland conservation. Best wishes for the future phases of the project. The question is curious to know the extent of harvesting of water stems in the area and its ecological impact.
Thank you, sir. There is harvesting of taifa by the local communities for thatching and their coral uses. Lotus, very small amount because harvesting of lotus is not very much done by the local communities there. And it has ecological impact mostly for the bathymetric study. It's a infringement. That area is not navigable. And these areas, but however, are used by, we also cited jacana nesting in those lotus patches and a very large number of jacanas in those areas where lotus was present.
The next question is from Kumar K. Confluence of rivers act as an ecotone and support good diversity of different facts. Your KPE estimates for birds is also showing same results, which is obvious. Will it be good to see habitat zones of different bird species which will help in protecting those crucial areas in the sanctuary?
Thank you, Ankit. Yes, we will definitely try to do after the winter survey, which is also an important time for birds in Harike. And since we didn't have for individual RET species, we didn't have very large amount of data for such, but definitely we'll do it in the coming phases.
Another question is, study started in February 2024. Will it be good to analyze the regeneration rate of trees?
I don't think this is any problem for regeneration status of trees for if the project starts in February or March. I don't think this is a problem.
The next question is from Kiki. Question is, you use the point count method for birds in your study area, but most of the area is covered by water hyacinth. What is the significant impact of the presence of water hyacinth on the migratory bird population?
I can better answer this after the winter phase of this study. Thank you for the question.
Vivek, you have more questions that I will hand over to you.
Okay.
So, next speaker, Saurabh. The first question is so the question is, what is your opinion on using passive acoustic survey in addition to visual surveys? Are these frogs vocal?
Yeah, so I forgot to mention that I can't use acoustic surveys because these frogs are not vocal to us. They may be using the supersonic or hypersonic voices, but we are not able to listen. So that for that we have logistic constraint. So we can't do right now the acoustic surveys for that.
The second question is, how do you identify?
So as I mentioned in that, and I also showed in my slide that these adults have a unique pattern. Every individual has a unique pattern, so you can easily identify them by these patterns. So these are the major things that helped us in the population.
Second question is from Dr. Kushi. Very good, very, very good question. Did you find any effect of seasonality and elevation in the distribution of the species?
Thank you for the question. Yeah, this is one of the aspects I'm still looking at because I have seen there's a change with the rain. That before rain and during the rain, there's a population or what do you call it, encounter, change in the encounter. So that will be impacting the species. And for the elevation, so the elevation range I selected is basically the range where both all the study areas have an elevation range of 1800 to 2500. So it was the same. So I couldn't use that as a factor for the population or the knowing about into this species.
So, next question is from Dr. Abhijit. The first question is, did you consider seasonal population fluctuation?
Yeah, like the same as the last question. There is a seasonal population fluctuation, which is like I already told you that this work is still ongoing, and I haven't analyzed the complete data set. So just before the rainy season, during the rainy season, there is a observed change in the population.
Second question is, your adult preference is against larval contact.
So,
Can I explain?
yes, sir. Yes, sir.
So you have said that the pattern is crossed.
Yes, sir.
You are finding the water flow is negatively forwarded.
Yes, sir.
But these are the habitats for the larva.
Yes, sir.
Which is completely contrasting. So your finding is actually you cannot say that this is the finding for the species because the species occur into completely different form.
Yes, sir.
The larval habitat and adult habitat are very different.
Yes,
so
sir.
you should not refer to the species. That's what I wanted to say.
Yeah, sir. But sir, what I was trying to say is that the larva stage is maybe they might be preferring this habitat, but the thing is that I was not going into the larva, what do you call the insides. I was majorly looking at the adults only.
Then you should not say that this is for the species pattern.
Okay, sir.
I think Saurav, you can now discuss this.
Yes.
So, the next question is from Esther. Generally, Amalok species are found in the fast-growing habitat of horse trees. However, according to your study, more species were abundant in the boulders rather than growing habitat. Are there any morphological features which makes them different from the other species of homologs which prefer growing habitats such as suckers, etc.?
No, the amlops has the disc shape for the same for for these habitat for fast flowing. They have discs on their toes and slight webbings on the back toes. The thing is that in my area there is no another species like both the amlops species are not found in one habitat itself, and the change like whatever the preference it is like even the Formosus is also was known as the fast flowing only. It dwells in fast flowing stream, but the encounter, which like the encounter rate for us, it was decreasing with the flow of it was showing a negative relation with the flow of the stream. That's what I was trying to say.
Thank you, Saurav. There are quite a few questions for you, so you can collect these questions and please get in touch with you. So next I call upon Diksha. So question is from Lucas Jain, while understanding your study, did you explore or consider pressure due to fishing for seafood industry?
Lucy and Goa, can you ask from there?
Basically, my question was that while you do this survey with the estimated for the getting information about this taxa, did you consider the seafood industry in Goa and the pressure that that has on the population?
we will be considering that, but our study is mostly profiling the biodiversity. The fishing data will be getting, we are touch with the fisheries department of Goa and will be getting our data from them. This was a pilot study, so we did not take into account any environmental or otherwise variables.
So the next question is from Dr. Anand Kumar. Did you assess the habitat, microhabitat parameters?
So no, we are just focusing at this in the study. We focused on the whole of rocky coast as of now. So we did not really look into microhabitats or habitats. We saw that most of our species counts we were getting, our transits were mostly laid on the rocky shores. We did look into sandy and rocky and what species are we getting from where, but that was the extent of it for the for these studies, for the pilot study. But we'll be definitely taking that into account as we go further.
Second question is, what is your target size for sampling industry?
It was 1 meter square. The quadrants were 1 meter square, like 1 meter by 1 meter.
So the next question is from Dr. Gopi. Passport standing and effect may be different, in size. As standing is a random phenomenon, identifying hotspots and linking with management will be difficult. So southern Goa standings are more. This could be due to issues in neighboring states. Karnataka. Your comments.
Yes, certainly that would be definitely taken into account because if we look at the sizes of the mammals and then we look at the coastline of Goa, it's quite tiny, and when in oceans there are no boundaries as such, so that's definitely possible. But what we looked for these strandings, we studied them from April to August as the pre-monsoon and monsoon season. It is hypothesized that it's because of the monsoon winds that are being brought towards the coast. The strandings are towards the shore. They are pushed towards the shore. But yeah, definitely it's possible that it's from Karnataka and Maharashtra.
Next to the compliments from Dr. Dehradun. So there is also a suggestion. Nice with lots of information. Suggestion moving forward. I recommend modeling species richness and diversity, accounting for size of the habitat, disturbance, including commercial activity and other habitat variables.
Yes, thank you, sir, for the compliment. And yeah, we'll definitely take that into consideration as we go forward.
Thank you, Disha. And I call on the next speaker, Kunal Dole. We have a question from Raman Kumar. You outlined the rationale for using SPCR linear. It is used for organisms that have a linear distribution. Can you clarify whether this assumption is applicable to fishy cats? It appears you choose the linear SPCR sampling simply because you were able to only access the river channel in a linear manner.
Thank you for the question. Since we first tried to use the framework of grid-based sampling only, but the sampling area that we covered was not covering all the study area. So that's why we use traditional approach, capture, mark, recapture, and we also experimented SCCR linear to estimate the population densities.
The next question is from Said Pratap, Diploma of Science. He compliments your presentation. Very good presentation.
Thank you.
Few questions. In Hyderabad, the district of West Bengal, fishing cats share same habitat with jungle cats. Any such interaction observed in Koringa?
No, there is no presence of jungle cats in Koringa.
And second question is depth of water preferred by fishing cats for hunting.
That we did assess.
in southern districts of Bengal, it has been observed that fishing cats don't eat the head portion of the fish. Any such observation in Korea and reason behind the same?
Yeah, I have observed a fishing cat eating whole fish after praying.
So next question is from Dr. K. Ramesh. Um, did you consider any specific sampling unit for deployment of camera traps, or was it done randomly or opportunistically?
No, the average distance kept between two camera trap stations was 674 meters, and it was first deployed in a grid-based manner only, in 1 square kilometer grids, at the height of 30 to 45 centimeters from the lad.
So the next question is, since your camera trap locations are spatially continuous, the heat map may have to be re-looked into for accuracy, spatial construction.
Okay, we'll consider that in our study.
The next question is from Dr. Gopi. He compliments the presentation. My compliments as a suggestion. We consider lunar illumination to tidal cycles to infer how they influence fishing and distribution and abundance in Korea.
The activity of fishing cats becomes away from the streams when there is high tide, and when there is low tide, the fishing cats come outside to prey in the streams.
So I have a few questions. When we look at the distribution of or abundance of fishing cats across the streams, it is mostly close to the streams or the creek mouth.
Yeah.
So this is also something to do with the vegetation, because the architecture of trees on the stream edges are usually the rhizophora trees it out in the stream edges, while the abyssinian trees are mostly the interiors, which has less complexity in architecture. So is there any such relationship?
I have observed that there are open areas along with the streams where we set up the cameras. So, but why the captures are there and the fishing cat activity is there? Because they are coming in those areas only for preying on the fish or birds also.
So, thank you.
Thank you.
So the last question is Abhishek. There is a question from Dr. Manjari. Instead of statutory records, you can consider mounting the recorders on patrolling boats.
Okay. Not every state has marine national parks in it and having a patrolling team for regular monitoring. So we can try installing with the fisher boats or the trawlers, that we can do. But currently we are having a stationary monitoring device. So we can try. And
second, consider using hydro mods, probably cheaper.
so the device which we are using has a higher sensitivity compared to the hydro mods. We have tested it, and also it has a long-term monitoring this thing, and it is very sturdy, and it can go up to 100 meters. And hydro mod has a very limited depth range of 30 meters only. So I hope you answered the question. Thank you.
Thank you all the presenters.
Yeah, thank you very much for so please come and collect extra question. There are a couple of extra questions available later on collect from Dr. Sami. So now we are about to end this session. I will request my co-chair Dr. Nehru to give his remark.
Thank you, sir. Though it's a short session with 5 presentations, all the presentations are covering a different ecosystem, from wetland, freshwater streams, estuarine habitat, coastal and marine, and particularly marine. So we had a wonderful, diverse array of talks. There were very interesting information. Of course, the marine species remains the impact where the project is still a lot of things to be uncovered as far as this particular species and its subpopulation is concerned. Likewise, the Goa biodiversity profiling. though it is one of the most accessible coastline, but still, when we look at the amount of scientific information available on the coast and estuarine beaches, it's still we are long way to go. So it's a good attempt. I congratulate all the speakers for making up a very good presentations. That's all.
Thank you, Dr. Nehru. So for me also it was really a wonderful session. Normally everybody is talking about aquatic system. I love that with a lot of scientific rigor. Really they are enhanced my knowledge. And there are a couple of observations I would like to say here, so the presenters they can make it note. The presentation first one, desilting of Karike wetland by Vivek. So there is one quick observation. The scientific name, when we are, as researcher, as biologist, whenever we are writing, we should be highlighting, italics, bold, or or underline somewhere. So basic rule actually, you have to be really careful when you are presenting. Second thing, the fish survey was conducted with the market survey. So market survey, whenever you go to market, we can't rely on that. Actually. People may be bringing from several sources actually. There will be a nice, same wetland itself, you can associate with the fisherman and get a nice rate actually. So these are the two remarks. Next time we can improve it. Coming to the Amalak's frog, so it's a wonderful effort, it's a great effort to identify the indicator quality of the species. And of course, he was trying to incorporate some of the stream covariate. And but still this species may be associated with some of the other microhabitat parameters such as moisture, temperature, and food resource. Maybe for food resources may be coming to this extreme. And instinct cover, that is another one important parameter. There is a thermoregulatory species. The cover is one of the important parameters that will be the microhabitat feature in that area. So if you incorporate these kind of microhabitat variables, you will get better results. So you can pinpoint what variables it will indicate actually. Coming to the third presentation, the diversity of selected taxa of Goa coast, though it's preliminary work, but you have to incorporate more things actually. Especially the WAV quality, always we will gather the ecological information, environmental variables. Just now picking up the inventory, it won't help actually. So when you pick up the environmental variable, that will showcase the spatial temporal distribution of species across the spectrum. Secondly, the sampling the size of plot size that was already indicated. 1 square plot, it may be suitable for a small creature, but you need to think about when you want to capture the many variety, you need to rationalize your sampling effort. Similarly, the species like they will be living inside the soil or rocky area. As you mentioned, the rocky shore it has a different setup. So you need to use multiple strategies to collect the species. Sometimes you have to put a trap or hook or several strategy you have to follow. Only 9 certainly will not help actually. Moving to the fishing catch study, it is one of the elusive species. Little known aspect, actually the main aspect, the food resource utilization pattern still is a great area actually. So right now this Kunal has often this species they end up conflict with the fisherman or the fish farmers associated with that area, going away to central area. You need to look on resource
So Suresh, Dr. Ashish here, and Dr. Frank, Dr. Suresh to chair the session, and Dr. Ashish here to co-chair, and Frank to facilitate the session. So we have an online talk in this session from Anupam. So she has already joined. So let's over to you.
Thank you, Dr. Vilash, and welcome to this session on alien ecology and conservation. This is particularly interesting to me because I work on birds, and so does Ashish. We did hear a very interesting set of presentations in the aquatic systems, and looking at the list of presentations listed out here, I think they're going to be really, really exciting. And we are, through those presentations, we are actually going to be traveling across different parts of India, and there are going to be presentations from Lakshadweep and from the Andaman and Nicobar Islands. So I think this is going to be a very interesting session. So we are going to break after the first presentation. We're going to break and then have tea and then come back and continue with the session. So for this session, I have Dr. Ashish Shah as well as Dr. Frank, who is our alumni, master alumni, and wonderful to have them. And I will hand over the mic to Dr. Ashish to follow up the presentations and you will introduce the presenters.
So we started the day with presentations from Himalayas. Now when we are coming towards the end of today's proceedings, let's start from the deep down south. And the first speaker will be Srija, who will be talking of the birds in the wetlands of Tamil Nadu. Over to Dr. Frank.
So Ms. Srija is from Hyderabad. She has done a master's from Pondicherry University in marine biology with dissertation in Andaman Islands. So she is currently working as JRF at Satan. Over to you, Ms. Srija. Yes, you're audible, Ms. Srija.
Southern districts of Tamil Nadu. So firstly, wetlands are very important habitats because they are the lifelines for both humans and wildlife. So they provide different kinds of ecosystem services such as groundwater replenishment, water storage, water purification, and also it's a major source of income for most of the coastal people who are living in the coastal regions. So there are different kinds of threats to the wetlands, starting from a landscape level where because of the various changes in the land use, to a micro level of pollution, there's a lot of invasive weeds. So one of the major root causes of this kind of pollution is also the human interference with the wetland system, because humans are highly dependent on these wetlands. They also tend to modify the wetland habitat based on according to their needs, in the process of which there's a lot of threats that are caused to these wetlands. So my study species for this project is the spot-billed pelican, which is Pelicanus philippinensis. It belongs to the family Pelicanidae. Looking at the distribution, we can see that it's distributed in the Southeast Asia. And focusing on the State of Indian Birds report produced in 2023, we can see that the loss of the pelican sightings in the regions of Gujarat and some parts of Western Ghats, which was earlier reported as part of their distribution range in the Cornell Lab of Ornithology. So these birds are generally the water birds. They are piscivorous in nature, that is meaning they feed on fish, and they are colonial nesting birds. They also, the young ones are etricial, meaning they need high levels of parental care. So the study area, we have 3 districts in the southern Tamil Nadu, that is Kanyakumari, Tirunelveli and Thoothukudi. We have selected 51 wetlands across the 3 districts, taking Kundankulam, which is one of the oldest pelicanidaries, as the center, and we have considered 51 various wetlands across the 3 districts. Now looking at also the annual trend produced by State of Indian Birds Report, we see that there's drastic decline in the abundance of these birds in Tamil Nadu state. So it's evident as to why we have to study this particular species. Now, looking at the literature review, we have various national and international studies. So the national studies mainly are focused on the status and distribution of spot-billed pelican, and a good amount of work has been done on breeding and nesting behavior. However, there's not much on the other aspects of ecology and conservation. So the international studies mainly from Sri Lankan region, where they have some studies on status and conservation of spot-billed pelican. Now, the importance of the study is the study, of course, has different components, including movement pattern of spot-billed pelicans and the land use changes over the decadal range affecting the wetlands. But for this presentation, I'll be mainly focusing on the water quality of the wetlands, the vegetation structure, and the human disturbance. How is that particularly affecting the habitat usage of the spot-billed pelican? Now, with respect to the objective, so I've said we have chosen 51 wetlands across all the 3 districts. So the criteria for choosing the wetlands is first the distance to Kundankulam. So we have considered wetlands of varying distance to Kundankulam. Then the size of the wetland, the depth of the wetland, and also the earlier records of pelicans. So we consider wetland where the pelicans were recorded, and as well as those wetlands where they are not present. So counting the birds, we followed the total count method for 45 minutes at each wetland. The number of vintage points varied based on the size of the wetland. Then we also followed the double observer method in order to reduce the error. So we also characterized various kinds of habitat characters such as vegetation sampling. Vegetation sampling, we have considered, we have divided the vegetation into 5 major categories. And within the 20-meter radius from the perimeter of the wetland, we have seen what is a percentage of the all 5 major types of categories present. We also noted some other important factors like the presence of open water. Since most wetlands in these districts have water, but they are covered with the invasive weeds and other hydrophytes. And open water is very important for foraging aspect of, in terms of foraging for the pelicans. So we considered the percentage of open water and the presence of mounds and artificial structures for perching, and the presence of submerged tree vegetation in water, which is very important in terms of their nesting. We also looked at, we tried to see what is the fish diversity present in the different wetlands by interviewing the locals in that region. Then we did water sampling to understand what is the physiochemical structure, the quality of water that is present in the wetlands. So we used a multi-parameter probe, which collects information of about 13 different factors, including the dissolved oxygen and oxidation reduction potential and so on. Then we took measurements of depth and light penetration using the Secchi's disk. We also collected data on phytoplankton and zooplankton using the phytoplankton and zooplankton nets and did the laboratory analysis to identify them. We also considered some landscape characters using both the field measurements and also with the help of GIS. So we calculated things like distance to the nearest breeding ground, distance from the main primary source of river. In the field, we also collected some information on threats and disturbance, like the presence of water diversion from the wetland, or the presence of fish tender in the wetland, plastic pollution, and various other threats and disturbances. So for data analysis, we have mainly used N-mixture p-count modeling, in which we considered 38 site covariates, which were further grouped, and then 3 observation covariates. So the results that we've got, the primary results are like, of the 24 wetlands that has been surveyed in Tirunelveli, we found pelicans in only 7 wetlands, and 8 out of 17 in Kanyakumari and 4 out of 10 in Thoothukudi wetlands also had pelicans. So we saw more of pelicans in the coastal districts compared to the Tirunelveli, which is the main breeding center, as Kundankulam is placed in Tirunelveli. But the highest count of individuals that has been seen is from Kundankulam. Then, the result of the N-mixture pecan modeling, we've got that the distance to the breeding grounds and tender are very important detection covariates. And fishing, the number of pedestrians, the percentage of agriculture area around the wetland, ammonia, oxidation reduction potential, and fish diversity formed very important factors in determining the abundance of the spot-bill pelican. So looking at these results, we see that distance to the breeding ground and fish tender, both of them are negatively associated with the predicted detection of spot-bill pelican, meaning that increase in the distance to the breeding ground and those wetlands where there was fish tender had less number of the predicted detection of the spot-bill pelican was less. And looking at the factors influencing the abundance of spot-bill pelican, the fish diversity was positively correlated. Ammonia was negatively correlated because it's a landscape which is dominated by agriculture. There's a lot of mixing of water between the wetlands and the agricultural lands, and the ammonia levels are very high. So, however, high levels of ammonia is not good for the fish and other wetland species. Again, pelican being one of the top predators, so also gets affected in a negative manner. The number of pedestrians showed a positive relationship. That could be because these wetlands are very huge and the pelicans are generally occupying one part of the entire wetland, which is slightly away from the highly used area, because of which it could not show any significant, there could not be much disturbance to the pelicans as such. Then oxidation reduction potential also showed a negative relation. There's an optimum level of oxidation reduction potential which is necessary for the better survival of fish. So either too high or too low levels also affect the predicted abundance of pelicans as well as other species. So the percentage of agriculture fields where we have taken 500 meters wetland buffer and we have seen what is the percentage of agriculture fields, and we have categorized them as high, medium and low. So wherever there is high agriculture area surrounding the wetlands, we have seen that the predicted abundance of the spot-bill pelican is also greater. And fishing here also is a very interesting pattern. We've seen that the wetlands which has more fishing also has greater predicted abundance of pelicans. But one important thing to note here is that we have not actually scaled the fishing, whether the fishing is small scale done with just the hook and line, or it is done at commercial scale as we saw in case of fish tenders. So if we look at the earlier slide, we see that in fish tender had a negative relationship of pelican detection, but fishing had shown a positive relationship. So we can maybe come to a kind of conclusion saying that it's not just the fishing as such is not as a threat, but the level of fishing or the scale at which it is done is a very important factor in determining whether the pelicans are actually using the wetland or not. Now, the different kinds of disturbances that we have noted, like as represented here, lots of algal blooms, there's plastic pollution, there's dumping of waste. There also the fire and air pollution is something adjacent to the Kundankulam area, which is the main pelicanary. There's a cremation ground because of which there's often fire and lots of air pollution. So this is something I think which needs to be given importance. Even the buffer region of the protected areas or nesting grounds also needs to have some kind of protection where it does not disturb the birds inside the pelican. Now we also look at this image where the rounded figures in the red actually are the dead pelicans. So we have seen a lot of dead pelicans in some of the wetlands, and because there was not enough water in Kundankulam and there was a dispersion of the population compared to the earlier years, there was less bird population nesting in Kundankulam, and also they spread across other wetlands. And we've seen that in a lot of other wetlands, there were pelican deaths. So we're not yet sure, or we are yet to investigate as to why this has happened. So the main outcome of this objective that we're looking at currently is we want to actually assess the water quality assessment of the different wetlands at a landscape level, and also understand what are the different kinds of localized threats, and also identify these important limiting factors which influences the habitat usage of the spot-billed pelican. So in the end, I would like to thank the SERB, the funding body, Forest Department, Director of SACON, WII, all my field assistant, lab mates, and everybody who has contributed in doing this particular study. And thank you. Hello.
So thank you, Shrija. I guess.
Thank you.
Okay, so I'll pass the mic to the research coordinator.
Srita, I was wondering if you can hold on, we can finish the question and answer right away rather than connecting again.
So yes.
If there are any questions.
So are there any questions for Shrija which can be passed on now, because this is an online presentation and we have challenges in getting her back on. So if there are any questions. In the meantime,
Yes. Yes, sir. Srija, it's am I audible, Srija?
Yes.
Yeah. Srija, very good work, very nice, fantastic work. I was just wondering, there are a couple of questions. The first one is, you said there is an overload of ammonia when you test for the water quality. So this you attributed to the agricultural practices, but it also could be due to the fact that the water deposition, because Mondakulam Heritage has been active for many years, more than 100 years. So now, because of the excess and if the flushing mechanism in this particular irrigation tank is not good, so the ammonia content both in the water and in the soil will be high. So not necessarily because of the agricultural practice, because of the bird itself per se is not.
Okay, so you are saying because of the birds itself, there could be high levels of ammonia. Is that what I got you right?
Yes.
Okay, so that maybe that could also be a cause, but no, looking at so I could what you are saying could be right, but looking at the scale of the landscape, I mean looking at the wetland size and the number of birds that are actually using it, I don't know how much it would actually contribute to like high levels of detection of ammonia. I mean, it definitely contributes to the ammonia levels. Yes, that could also be a possible reason. I mean, we cannot clearly say that, it is because of exactly just agricultural mixing.
Yes, I'm correct, Srija, to whatever understanding we have. We have been working on this landscape for many years, and we are actually, we are actually looking at how the nutrient inputs from the birds penetrates into the wetland and then further into the agricultural field. So ammonia does have an impact. And the second thing is, when you look at factors influencing the nest site selection, I was just wondering, do you also incorporate the nesting tree availability in the landscape and nest material availability? These two factors are very crucial compared to availability of agricultural fields or some other factors.
Sir, can you please repeat? Like I could not hear the exact two factors you mentioned.
Nest tree availability and nesting materials.
Nest. Correct, yes. So actually, we have considered the nest tree availability when we said the tree submerged in the trees submerged in the waters. Generally, the acacia, the prosopis, and the palm trees in this particular area we have seen. But most of the wetlands that we surveyed are not breeding grounds. They're mostly the foraging grounds, and we had very few breeding grounds, in fact. So maybe because of that particular reason, it did not show up in the model. But what you said is very right. Hello?
Does it answer your question? Yeah. Okay. So I have a question. So Srija, thank you for your presentation. My question is related to the question that Dr. Gopi had. So you were measuring water qualities as one of the variable for the bird habitat use, but the bird itself is changing the water quality, right? Bird is pooping there, defecating there, that would alter its say BOD, COD and other factors that you're measuring. So how can we say that water quality is the one driving the habitat selection of the bird rather than because the bird itself is changing its habitat?
Okay. Okay, so we just didn't consider the water quality alone. We also took the other factors, but since water quality is very important in terms of like maintaining a good fish abundance and diversity, we've considered that also as an important factor. Yes, like you said, I think every species also contributes to the system that it lives in. But we are trying to focus on how the environment as such could be like an factor which could be influencing, like in all the wetlands that are present, they are using some wetlands, but they're not using certain wetlands. But we also seen wetlands which are not used by pelicans are also quite degraded in terms of their quality. So I think it is very based on the locality and what are the kind of situation available. It depends. So we cannot actually exactly segregate how the bird is affecting or how the like what we are trying to look at is only one direction. But like you said, yes, there is always a two-way influence that's happening. The effect of the habitat on the bird and the bird on
Yes, and
effect of bird on the habitat.
Srijan, another point to note would be that these birds, wherever they nest, they need not feed there. So they can even fly like 10, 15 kilometers away to their feeding grounds and bring the fishes to the nesting.
Sorry, like I am not able to actually hear what you're saying clearly. Like, can you, if there's in the chat, then I can view what they're asking.
Okay, Sridhar, thank you so much for your effort. There will be many more questions that will be sent to you, and you can accordingly respond to that question. Because you are so far away, we will not be able to call you back again. So, yeah, so thank you so much, and you can disconnect now. You can stay online, but disconnect from this presentation. I've been informed by the research coordinator that we have a break for tea. So we break now, but just as a passing remark, in the morning we had a lot of attendance. I see a lot of empty seats now in the center row. And so post lunch, I think the numbers have trickled down, and I don't want that to happen after tea because the students have put in a lot of effort to make these presentations. So please come back after tea.
So what should we do? Should we reduce the number of chicken paneer or rice? So we'll break for tea. We assemble here at 4 o'clock. 17 minutes. So let's be back.
लास्ट है हाँ लास्ट वाला थर्ड यही सेशन अच्छा वही चल रहा है कहाँ पे डालूं मेरे बंद एक बार उसको चेक कर लिए फट रहा है क्या ठीक है 20 to 7
ये अगर आप पीपीटी बनाओगे 16:9 में बनेगा।
बनाया तो 16:9 है, पर ये स्ट्रेच होता है नहीं होता? कुछ पार्ट्स में प्रेजेंटेशन एक दो थोड़े से स्ट्रेच
बन- चल रहे भाई, चल रहे। अरे चाय पीएंगे हम।
मेरी डाल दे। अल्लाह था। यही बात की जगह लगा दी ना। तस्वीर आगे मिल गया सारे भर के ये रखता हूँ और ग्लास साफ कर दिए। नहीं खाली भारी खाली है, खाली सब खाली है। एक ले ले, एक ले ले। बस हो गया पीछे होने चाहिए। ग्लास की क्लोज कर। हाँ तो लास्ट में कर देना है। लगा दी होनी है और ये गिलास क्लोज कर दो। अभी नहीं कोई ये गिलास क्लोज कर दो। यहाँ नहीं रखते साफ कर दो कर दीजिए। गिलास दे दे। हाँ ये 3 गिलास दे दे। गिलास दे दे। कपड़ा मार के तू ले जा। अभी ये तो वहाँ ले जाना है। हाँ गद्दे में। आप रखनी है ये साफ ठीक है डॉक्टर। हाँ ये साफ गिलास ले ले वही रहा तू ले गया साफ है वो। एक और साफ ये बता रहा हूँ वहाँ चाहिए। और फिर एक लोग था तीनों चारों जाएगा एडवांस बोतल की 10 रुपये भर दियो। बोतल सुबह भर दी सारी है। वाइप कर दिया वहाँ वाइप कर दिया। पूछ ले बाद में लगाना है नहीं लास्ट ये पानी बस अभी लास्ट में लास्ट में बाहर का होता है इसको बाहर की ऐसी रहेंगे कोई दिक्कत है। इसीलिए इसमें पानी भर के एडवांस में रख देंगे सब बात कर देंगे।
कुछ नहीं हो रहा। ये डायरेक्ट थोड़ी है, तो यूपीएस पर वही तो उसमें लाइट नहीं आ रही, वो पुराना है।
So welcome back everyone. Please settle down. Those of you still coming, please come quickly. Settle down and let's start the session. I'll hand over the mic to Dr. Ashish to invite next presenter.
My first talk for this particular session was from the coast of Tamil Nadu. Now we are going to coast of Lakshadweep, and we'll have Mr. Rathi Mitra talk about the colonial nesting birds in the Lakshadweep Island.
So Mr. Rajiv Mitra completed his Masters in Environmental Science. So after his Masters, he has worked extensively on various research projects contributing to the conservation of bird populations across India. He is currently working as a project associate at the Royal Institute of India, where he is leading a study in the Lakshadweep archipelago, assessing the current population status of nesting seabirds in the region, and also using satellite telemetry to track the fine-scale movements of select seabird species. Over to you, Mr. Rajiv.
Thank you so much. Hello, am I audible? Hello, yes. Thank you so much, sir. Good afternoon, everybody. I think it's fair to say that the first thing we all learned about geography is that the Earth is around 70% ocean. And in this ocean, life originated and it expanded, evolved. And there is one group of animal that actually made these oceans their home. It's the seabirds. Globally, around 800 million seabirds, consisting around 350 species, they have literally made these oceans their home. They are found from the Arctic to the Antarctic and even in the tropics. They have literally expanded everywhere. However, all is not well with the seabirds. With the rising fishing industry, global fish stocks plummeting, bycatch, line fishing, and even egg harvesting has seen the population of seabirds dramatically falling. If you look closely, 56% of 314 well-studied seabird species colonies have seen a dramatic decline. And interestingly, within this, 80% of the species are least concern. If you look at the seabird colonies in the Indian Ocean, similar such trends have been observed. There are roughly around 500 nesting colonies across the Indian Ocean, and within which 70 or 80 of them have been well studied. And even in that, 46 well studied tern colonies, 14 of them have been extirpated because of the concerns I mentioned before, and 8 are constantly declining. Now, if you look closer to our northern Indian Ocean, the Chagos-Laccadive ridgeline holds some of the most important seabird nesting colonies. Chagos Archipelago, Maldives,
and the Lakshadweep archipelago, or the Lakshadweep Islands, they have some of the most important seabird nesting sites. From the Indian region, it's the Lakshadweep archipelago where historically seabird nesting sites have been recorded. There previously used to be 6 or 7 sites where seabirds used to congregate in large numbers. However, due to sea level rise, egg harvesting, coconut plantation, much of the islands are lost. However, only in a small island called Piti, seabirds continue to come. And there is also a small colony in Cheryabani or Walepani reef where occasional nestings have been reported. And today, I will be giving you insights into the nesting population of seabirds from Piti Island. Piti, also known as Pakshi Piti, is a very small, around 4-hectare landmass area. It's a very small island. It is around 25 to 24 kilometers from the capital Kavarati. And historically, Alcock in 1902 was the first person to report nesting of seabirds in the Piti Island. However, Alan Octavian Hume reported nesting of seabirds all across Lakshadweep archipelago. Following them, there have been very few studies that was carried out in Piti Island, and all of them talk about an important trend, a dramatic decline of seabird populations. Some of the studies, although questionable, were basically guesstimates where observers did not land on the island. They did it from a boat or a vessel or a ship. But overall, the picture is quite clear. The seabird populations in Lakshadweep has seen a 95% decline. With this rising concerns, the Lakshadweep Forest Department reached out to the Wildlife Institute of India to assess the current nesting population status of seabirds, to understand the current trends, and also to look at the post-nesting movements of select seabird species. However, today, for this presentation, I will only be talking about the population status and their trends of the nesting seabirds in Piti. Now, Piti, as I mentioned, a very small area, but accessing this island had always been a challenge. This island is covered by a barrier reef, which makes landing on this island impossible. You cannot land on this island with any boat or any vessel whatsoever. And even to reach the water surrounding the island, you need right oceanic conditions. And then to reach on this island is an issue of another level. You really have to swim 100 to 200 meters in calm oceanic conditions to reach the island. So my observations were basically possible 6 times, from February to April, twice every month. And on these occasions, 3 times, I could not land on the island. So observations had to be made from the boat. The other three occasions, I managed to land on the island from a small channel of the sandy beach area where I had to swim 100 to 150 meters from a boat which was far away. And then after landing on the island, we actually systematically counted the seabird population and their nests. Now, recent reports have indicated that the dwindling seabird populations, especially elegant tern in the California coastline, if the population is really small and if it is disturbed, the birds would abandon the nest. So keeping this information in mind, we actually did a direct count method. We used a direct count method. We observed the terns, observed the nesting seabirds from the periphery and counted their numbers. Now, looking at the results, our findings indicated that this small island not only hosts large number of nesting seabirds, this isolated land also provides crucial habitat for different species of wintering birds. And also, this island is used by species of seabirds which are on passage, going to further in the Indian Ocean, southern range, or even in the northern range. And for now, I will discuss about the non-nesting seabirds. The first is the great crested tern. This species is commonly seen all across the Indian coastline. And this bird, rightly so, was also very commonly documented in Piti, more or less around a maximum of 300 individuals we recorded from Piti Island. And these birds, their major nesting colonies are there in the Chagos-Laccadive ridgeline, specifically in Maldives and in Chagos archipelago. This was further followed by the lesser crested tern, a very similar looking birds, but differentiated by the orange or reddish beak. And ringing recoveries have indicated that in the Western Indian Ocean or the western part of India, the lesser crested terns which come here actually come from the Persian Gulf, where they actually breed. Now, another interesting bird, is the bridle tern, one of the four species in the Onychoprion genus. This bird actually was observed roosting on the western part of the island, where there was rocky areas where they are known to breed. Even though I did not observe this species breeding, but historically there has been reports of this species breeding in PT Island. The nearby known nesting site of this species is in Vengula Rocks, Maharashtra, in the Maharashtrian coast. Followed by this was the common tern, a common winter migrant. This species breeds in the northern latitudes and uses the Indian Ocean basin as a wintering site. A maximum of 70 individuals I observed during the study, which appeared to be a stable population from 50-70 individuals, and maximum was 70 birds, which I observed. This was followed by white-cheeked tern, a very similar looking tern. You can identify, it is very difficult to identify in non-breeding plumage, but if they are in a sitting position or they are taking off, you can look at the white rump of the common tern and the gray rump of the white-cheeked tern. That's how you can identify these two species. These are endemic birds of the Western Indian Ocean. They are found in the Western Indian Ocean. They breed in Persian Gulf and in the Red Sea. This was followed by a very interesting sighting of roseate tern. This species we recorded for the first time in the waters of Lakshadweep archipelago. 4 individuals were observed on 2 occasions. This bird is again identified by a creamy white belly, long tail streamers, and a red beak. Now, to talk about these birds and their population trend, you can see that clearly there is a spike in March, where greater crested tern and lesser crested tern, both of their population peaked. So why it happened? Both these species actually, their nesting period starts from, April, May, and then followed on. So what we believe is these birds were congregating in good numbers in PT Island before dispersing to their respective breeding sites, maybe some in Chagos, some in Persian Gulf, some in Red Sea. While the other species like the bridle tern, as I said, they could be nesting after my field work finished, but we could not say for sure. The common tern and the white-cheeked tern, it appeared to be their wintering habitat. These birds were 60 to 70 individuals were observed very frequently in this region. And the water surrounding PT Island is very rich. It's a protected area. 40 square kilometer of area is protected. It's very rich with nutrients and of course fish. There is no fishing taking place in this island. So hence the area surrounding PT provides an ideal foraging site for these birds to actually forage on their crucial food resources. Now, interestingly, I also documented 3 species of tube-nosed birds, Procellariformis. They are known to have a tube nose, which actually gives them, an extreme sense of smell. These birds, like the Wilson's storm petrel, which actually breeds in the Southern Ocean, near Antarctica, South Georgia, Falkland Islands, Gough Island, all these small islands in the Southern Ocean. And these birds are austral breeders, means breed in December, January, that time, and they would invariably come up with their migration to this part of India and the northern Indian Ocean. Flesh-footed shearwater, a near threatened species, again shows a similar migration pattern. Persian shearwater, another species which we documented for the first time from Lakshadweep Islands, and these birds have only 3 known breeding sites. That too, one in the very famous island of Socotra, which is in the Yemeni territory. Now, I will now be talking to you about the 2 most famous birds for which PT is actually known for, sooty tern and brown noddy. So the sooty tern is actually a species which is distributed from 35 degree north to 30 degree south latitude throughout the equatorial belt, and they are actually the bird which is known to nest in PT Island for centuries. A maximum of 1,000 birds we documented, and they were mostly congregated at the central part of the island. This was then followed by the brown noddy, extremely interesting looking species with a gray cap and brown color. Not to be confused with the lesser noddy, which was recently sighted in Maharashtrian coast, which the gray color comes in front of its eyes. So the 500, approximately, the 500 birds maximum which you observed in this PT island, but these two species had spatially, different or distinguished nesting colonies. Their nesting colonies did not intermix with each other, which is an interesting find, because in other places there are reports of these two species having intermixing colony, which I did not observe during my study. Now, to talk about the population trend of sooty tern and brown noddy, you can clearly see there is a spike in population of sooty terns, from 400 individuals recorded in February to around 1,000 individuals in April. So what is happening with this population? So these birds are known to show, great movements, and they are acyclic breeders, meaning that they don't have particular season where they would nest. However, as soon as April comes or May comes, this entire Indian Ocean belt or the Arabian Sea belt gets influenced by the monsoonal currents and the monsoonal winds. So what happens is, different tracking studies from Seychelles and Bird Island in Seychelles for the last 5, 6, 7 years have shown that sooty terns from Seychelles, Bird Island, they are known to migrate to this part, maybe for breeding. And then for the brown noddy population, you can see there is a stable population of 400 to 500 individuals. And as most of the seabirds, they show extreme site fidelity, nesting site fidelity. So what we believe is that 400 to 500 birds are constantly present in this island. And after their breeding, they would move and use the ocean basin and again would come back into the same island for nesting. Now, if you look at, interestingly, if you look at the, in March, if you look at the nesting, if you look at the nesting population numbers, we could clearly see that these birds have actually shown good trends. Now, what about the clearing threat of egg harvesting? The Kurup and Pandey, they have reported egg harvesting from this region. So I wanted to look at whether this egg harvesting trend was still happening in this region. So for this, I surveyed in all different islands, Agati, Kavaratti, Androd, Kalpeni. I asked fishermen whether egg harvesting is still taking place. The answer which I got was really interesting. They said that because of the change in economic viability, they no longer go to the PT island to collect the seabird eggs. The improvement of the islands, of more tourism coming in, and with the development of economy, the people would not go there and gather the nesting anymore. Even you can see in this nesting trend, there was no nest, and supposedly there were no nests or eggs were lost because of egg harvesting, if it had taken place. Other threats, there has been sea level rise for global warming has been a threat for most of the seabirds. If you look at one of the prime example in Dry Tortuga National Park, Florida, the seabird population crashed from 3 million to 350,000 because of the rising sea levels within the last 8 decades. In Lakshadweep, there has been a sea level rise for 0.4 to 0.9 millimeter per year. This is actually a threat that needs to be looked upon in future. However, a natural threat was from a natural predator, ruddy turnstone. These are Arctic breeders and known to have a diverse range of diet, and they are known as nest destroyers. There was this white tern colony in Australia back in 2003-04, where a ruddy turnstone population literally decimated the entire nesting population. As these seabirds only lay one egg, so if you are predating on one egg, the entire population is lost for that period of time. I observed around 30 to 40 individuals in all of my visits walking regularly inside the colony, and I observed 9 occasions in direct observations where these seabird eggs, specifically sooty tern eggs, were directly hunted or predated by ruddy turnstones. However, I do believe that all hope is not lost for these seabirds. As you can see in this image, brown noddy with its chick. The information which we gathered during our survey, it actually forms a baseline and adds into the ongoing seabird conservation effort or population estimation effort across the Indian Ocean region. And as I mentioned, my fieldwork was only possible till April. With May, the onset of monsoon, fieldwork is shut because all the local transportation, island to island transportation is closed. So now we are planning to deploy time-lapse cameras on the nesting colonies to understand the seabirds' nesting behavior. Yes, sir. And I will be updating you in future regarding the further updates of the seabird colonies and their nesting behaviors. By this, I would like to conclude. I would like to thank the following people, specifically my friends and family in Lakshadweep, without whom this was not possible. Thank you so much.
From Lakshadweep, let's move to Andaman, where Ms. Aarti will be talking about Andaman Swifts in Nicobar Island.
Ms. Aarti has done a master's from Pondicherry University in marine biology. She has been doing ornithology studies for more than 3 years. So currently she is working as a project associate in IEWH project on a species Andaman river nesting. Over to you.
Thank you, sir.
Please stick to time, sir.
Yes. Good evening, everyone. Let's listen to the chirps of tiny black bird from the Nicobar Caves. Birds are always fascinating to watch. The birds, only few of them, inhabit the cave ecosystem. One such special bird is Andaman edibleness swiftlet. So here I am talking about Havabil of 10 degrees south. I am presenting my study under IDWH project Population Status of Andaman edibleness swiftlet in Nicobar Islands. Edibleness swiftlet. Edibleness swiftlet is distributed across Southeast Asian countries. It has 6 subspecies. One among them is Aerodramus fusifagus inexpectatus. It is an endemic bird that is residing only in Andaman and Nicobar Islands. It is least concern according to IUCN globally due to its wide distribution. Andaman edibleness swiftlet. This endemic species in Andaman and Nicobar Islands, I have studied across Andaman and Nicobar Islands, but today I am limiting my conversation to Nicobar Islands, which is 10 degrees south to the latitudinal channel, which is separating Andaman and Nicobar archipelago. It is a cave-dwelling aerial insectivore, and it is special for one reason, because its nest is made of saliva. It is regurgitated from its sublingual glands. It is having a great price in the market, rates from $2,800 to $14,000 per kg in the market. It is causing a threat to its population. There has been illegal trade going on this species from 16th century onwards. Even during British colonialism, this bird have been exploited with its nest being collected throughout the islands. Till 1960s, this trade have been happening. Even now, the trade is not banned and it is not under any international trade law. Even the bird is not protected under Wildlife Protection Act 1972. Why is the nest so special? The nest that is a specialty in Southeast Asian cuisine. It is used for making soups and it is also used in medicines in whole Asian continent, especially in Chinese medicine. There has been previous studies on this bird. The pioneer study was done by Dr. Ravi Shankaran from 1995 to 1998. He published a report in 1998 which shed a light to the whole population of Andaman and Nicobar Island population of the bird. He concluded that there has been an 80% of decline in the population of Andaman and Nicobar Island edibleness swiftlet. He employed many conservation strategies for the Andaman populations, and now it is monitored by the Forest Department with the help of the local people. But the Nicobar population, this case is really different. When he did the survey, the Nicobar population was protected by the tribal people. They owned the caves, and the nests in the caves were sustainably used by them, and the population was not so less. So he didn't do any conservation strategies on Nicobar Islands. And there were many studies on the Andaman population throughout the years, but for the Nicobar, after 1998 survey, there have been no population survey or any other studies happened. So from Dr. Ravi Shankaran's report, we could understand that a total population of 210 nests were reported. The one nest indicates one breeding pair, that is estimating up to 2 birds. So the Nicobar Islands are divided into 3 subgroups. One is Car Nicobar group, one is Central Nicobar, and next is Great Nicobar group. So the Car Nicobar group, he found that 2 caves had the presence of edible nest swiftlet, which is having only 3 nests. And in Central Nicobar, there were 14 caves with around 1,427 nests. And in Great Nicobar, with 27 caves having the presence of edible nest swiftlet, only 580 nests were reported. So what happened after 1998 survey? There has been a devastating 2004 Sumatra Andaman earthquake happened in Nicobar and Andaman Islands, which actually subsided the Nicobar and uplifting the Andaman. So there has been severe damage to the cave network in Andamans as reported by Manchi Shirich in 2015. So similar might be the case of Nicobar. And also, there has been many socio-economic changes happening to the Nicobarese communities because they have been using natural resources for their sustenance. After the tsunami and the earthquake, there has been a depletion of the natural resources due to the submergence. So they had to find some other way. So the nest of the caves were really vulnerable for the nest collection. This is one of the caves that have been closed and sealed due to the tsunami. So before our survey started, we had no idea on the cave locations. From previous studies, we do have little bit idea, but it was not possible for us to survey with that information. So we had to consider to do a questionnaire survey with the local people to locate the caves and the nesting populations. The total population survey was started from January 2024 and it lasted till July 2024. 207 field days were there, and we could find that in 8 islands, the edible nest swiftlet were absent, which is from the previous studies and also from the questionnaire survey. 3 islands were totally inaccessible due to the rough sea. So we could actually survey 11 islands. Out of the 11 islands, 8 islands had edible nest swiftlets recorded. So total survey gave a result of 2,851 nests, which is approximating to 5,702 birds. And 3 islands, the edible nest swiftlets were no longer reported. So this is a island-wise distribution of nests in Nicobar Islands, which shows that Nankauri Island had the highest number of nests of 250, which is followed by Kachal. This is actually cave population. So in Car Nicobar group, there were 2 caves with 30 nests in it, and in Central Nicobar group, 6 caves had the presence of edible nest swiftlet with 567 nests. But in Great Nicobar, only 1 cave was present with edible nest swiftlet with just 4 nests present in it. So there have been a population shift when we consider 1998 survey and present survey. So what happened? The cave nest count. Cave nest count was 2010 in the year 1998. 2010 nests were present in 1998, which now decreased to 601. But the total nest count, there is a change. Ravi Shankaran only surveyed the cave populations of edible nest ciflet. But now what happened? The total nest count is 2010 itself in 1998. But now the total nest count increased to 2,851 nests. So where do the rest of the population went? Where are they? Where did edible nest ciflets go? It shifted to human-made structures. So we could find around 2,215 nests from human-made structures alone. From Central Nicobar group, I could find a 115 nests count from just human-made structures. But when we look at Great Nicobar group, 2,135 nests were just counted from 2 human-made structures alone. This is one of the structure that is in Great Nicobar Island, which is a submerged temple isolated from the terrestrial area. It had 63 nests. This is a special case, Navy jetty in Great Nicobar. So the highest population of any nesting site is present here, having 2,072 nests in total, which is counting up to 73% of the total population counted in Nicobar group. That may be because it is a Navy jetty at its defense area, it is protected from local people. So where did the cave population, what happened to them? It is decreasing in a drastic manner. So cave population have to be protected and monitoring have to be done. In Andamans, 3 major populations of caves have been protected from last 10 to 15 years by the Andaman and Nicobar Forest Department and local people. So that method can be employed here in Nicobar also with the help of Nicobaris. House ranching. What is house ranching? House ranching is a method used throughout Southeast Asian countries for sustainable harvest of edible nest ciflets. This can be employed in Nicobar group where there have been a shift of population to human-made structures. This is a potential strategy to conserve population. I conclude by saying this. This is a house ranching found in Vietnam. There has been suitable conditions for birds to nest in these houses. This is the acknowledgement. I thank everyone of my family in islands. They have been great help in my survey. They are still in contact. They considered me as their family and took me in and helped me a lot. And I also thank my colleagues and friends. Thank you everyone for listening to me.
Thank you, Arti, for such interesting presentation. Next, we move to Mr. Harshad, who will be talking about JIPS versus assessment across India.
Mr. Harshad completed his Masters in Wildlife Conservation Action from Bharati Vidya Institute of Environmental Education and Research in 2019. He has served in both in situ and ex situ conservation, contributing to the field of wildlife conservation. Currently, he is working as a project associate at the Wildlife Institute of India, where he is leading the first pan-India nesting population survey, focused on the conservation of India's critically endangered JIPS species. Over to you, Mr. Harshad.
Hello everyone, am I audible? Yeah, so Good evening. I hope most of you have seen this photo before. This photo is widely used when it comes to the discussion about the population decline in India. Once very commonly found, now is critically endangered and has experienced about around 97% of population decline. Over the past 2 decades, there have been various conservation efforts taken from the government of India. First and most being the ban of the veterinary anti-inflammatory drug diclofenac. Subsequently, there were also uh, breeding centers which were taken forward, and given that the vulture population was very low, and with all this, we have created a lot of captive population, and also the recent studies have shown that the vulture population in wild have started to stabilize. There have been a lot of efforts to document the existing vulture population across the country, but there are some sites where the population is still been monitored, but the documentation of every site is not available. Documentation is only available from the select sites or the states like Madhya Pradesh, Gujarat, Tamil Nadu, Kerala, Karnataka, where the forest department themselves carry out the population surveys. Apart from that, the organizations like WWF India are also monitoring certain sites in Madhya Pradesh and Rajasthan where they are trying to estimate the population. With this, the data is very scattered, and more importantly, the vulture nesting sites where they used to nest before have shifted, or the population might have declined, and it can be because of various anthropogenic activities. So as a need to document all this information in a whole, this study was brought up from IDWH Vulture project. The pan-India assessment was carried out for the critically endangered gyps vultures, and today I will be specifically speaking about the 3 critically endangered gyps vulture species, which are the white-rumped vulture, the Indian vulture, and the Gyps tenorestris or the slender-billed vulture. Uh, so there have been to carry out the field surveys. The field surveys was carried out from February 23 to July 2024, and before the field surveys were taken up forward, we did an intensive literature surveys where from the inputs of the local forest department and also from the local conservationists, we mapped out all the known nesting locations of the vultures, and later on the field visits were carried out. And during the field visit, aspect of the project was on and also envisaged to create a national vulture database. So to create a national vulture database, there were certain things which we considered. So overall, around 27 variables were collected from the field sites, like the nesting site name, number of active and inactive nest, the species of the vultures nesting over there, the environmental factors, the habitat factors, and if there are any threats. So this is how the field visits were done and the observations were made. So we visited almost every known site of the vulture across India, and I will be talking about these 3 species in detail and about the status of nesting across India. So starting with the Gyps bengalensis or the white-rumped vulture, which is more widely spread. So earlier the Gyps species was distributed across India, and supposedly it was known from 206 sites across India. And Gyps bengalensis, white-rumped vulture, is a tree nester. So it was basically spread across a lot of protected areas. And currently, the situation is it's only limited to 49 sites across India and across 11 states. So, and it's largely concentrated across the Himalayan region the Kangra Valley, if you can see over here. And a lot of population from the south has been lost, and it's now only concentrated in this Mudumalai area of India. And for this vulture, we recorded almost 450 nests, and majority of the nests were recorded from Himachal Pradesh. For this species, we also recorded the trees which they were nesting on across the different populations. And to our surprise, in Kangra Valley, this species were mostly recorded on cherry pine trees. From the Mudumalai and the Gandhi Sagar Wildlife Sanctuary, this species was recorded from Terminale Arjuna tree. From Kana and Mahamaya in Assam, the species was recorded from Sal, whereas from Indravati and Kodarma, the species was recorded from the toddy palm. While on the other side, the Srivardhan and Maua, it's recorded from the coconut trees. And lastly, the Sohelwa Wildlife Sanctuary, we recorded the trees from the Haldu. So coming back to the Indian vulture, so Indian vultures are the cliff nesters, and they usually nest across the cliff and use the rocky outcrop of the cliff to nest. So as you can see, the distribution was also distributed in such a way across the Aravalis, the Western Ghats, and the Annamalai and Nilgiri ranges in the south. Across 94 sites, it was known to be previously nesting, but the current scenario is very different. Though the number of sites have increased to 108 sites across the 9 states, there has been a loss of population. There has been loss of nesting sites across the southern India. Though the species have been seen continue to nest in the central India, and for this species we recorded almost around 1369 nests, of which majority of the population was recorded from Madhya Pradesh and Rajasthan. So let me take you to the largest nesting colony of long-billed vulture, or the Indian vulture which we call. This is the Chambal River in Kota. It's on the banks of the Mukundra Tiger Reserve, and it encountered about 91 nests recorded during our survey. So let me take you to the closer look. So this is how the cliff of the site looks like, and the vultures use this ledges for nesting. So this is how the vulture use the site. Coming back to the Gyps tenorostris, or the slender-billed vulture, which is the most critically endangered bird in my study, and earlier it was distributed across the Himalayan and Gangetic plains, and across 47 sites it was previously known to nest, but now it's been only restricted or limited to the 12 sites, and that too in the state of Assam. And we could only locate 20 nests of this species across these 12 sites. It's very alarming in the sense, and similarly to the white-rumped vulture, we also tried to locate what kind of tree are they preferring. So this species is preferring more of the cotton trees across India, across the sites. So after these 3 focus species, we also had a chance to record nesting of other species during our field visit on the nesting sites of the previous species, and we could also record 35 Himalayan griffins, 35 Himalayan griffin nests, and only 2 bearded vulture, which was in Jammu, and only 5 nests of red-headed vulture could be recorded. So along with the vulture site survey, we also tried to locate the major carcass dumping site across India, and we could only locate 21 major carcass dumping site and 5 vulture restaurant. So vulture restaurant is a site where the vulture restaurant is basically managed by the forest department, where the carcass has been supplementarily provided to the vultures for the feeding. And It has been only recorded from the states of Maharashtra, Uttar Pradesh, and Himachal. Apart from that, there will be a lot of unreported, undocumented nest carcass dump sites, which I would have not visited. So maybe in future or maybe by some other service, I will try to do that. So quickly summarizing the findings. So from 206 sites to 49 sites, white-rumped vulture is only concentrated in Himachal Pradesh. From 94 sites, the sites have increased for the long-billed. But the number is only restricted towards the central India. And for the central slender build, 47 sites have been brought down to 12 and limited across the Assam. Whereas now, if we talk about how the species is spread across the protected and non-protected area, so white-rumped vulture, most of the sites, as you can see, are outside protected areas. So almost across 30 sites are outside protected areas, and majority of the nests are outside. This indicates that the white-rumped vulture may need more protection in future. Whereas on the contrast, the long-billed vulture or the Indian vulture has almost 62 sites inside the protected area, and majority of the nests are well protected under the protected area network. And there were no sites which we could record for slender build inside the protected area. So with this, all this nesting data, I would like to give out the numbers. So a minimum breeding individuals of this white-rumped vulture can be accounted for 900 individuals, whereas for the indicas 2738, whereas for the slender build only 40 breeding individuals. I think this is how the scenario across India is, and there can be a lot of study done from this data, and a lot of management efforts can be put on. And with this, I would like to conclude, and I like to acknowledge the Funding Authority, MOEFCC Director Dean, PCCF and Chief Wildlife Warden, and the forest staff of all the forest department personnel of all the states, the contributors to this data, friends, family, and colleague. Thank you.
So now we have Ms. Neha, who will be talking about Nicobar Microphone.
Sneha Pandey is a research scholar at Royal Institute of India. Her expertise on the field of remote sensing and GIS. She has been working in the habitat monitoring component of the IDWH project for more than a year now. And today she will be presenting her work on mapping the distribution of Nicobar megaphone. Over to you, Sneha.
Thank you, sir. Good evening, everyone. So the presentation today, through this presentation, I would be highlighting our findings about the distribution of a lesser known species, which is Nicobar megapode. So this work which I'm presenting today is a part of a larger project. It's under the habitat monitoring component of the bigger IDWH project. Nicobar megapodes are medium-sized ground-dwelling birds, and it is characterized by large feet and claws. In fact, the term megapode actually is a Greek word where mega means large and pod means feet. It is a mound-dwelling species, and it uses environmental heat source to incubate its egg. The bird is characterized by dark brown plumage, and it has a reddish skin around the eyes, on the face. According to IUCN, it is categorized as vulnerable, and it is also under the Schedule 1 in the Wildlife Protection Act of India, 1972. This bird belongs to family Megapodidae and order Galliformes. There are 22 different species divided in 7 genera, which are broadly classified as scrub fowls and brush turtles. This species, Nicobar megapod, is also commonly known as Nicobar scrub fowl. This species is endemic to the Nicobar archipelago, which comprises of 22 different islands. From India, from this habitat range, there are two subspecies. First one is Megapodius nicobarensis, which is confined to the central Nicobar Islands above the Sombrero Channel. And the other subspecies, which is Megapodius nicobarensis eboti, it is confined to the Great Nicobar Islands, which is below the Sombrero Channel. Moving ahead to the habitat of this species, the habitat is characterized by flatter terrain, and the species is mostly found within a distance of 100 to 200 meters from the shoreline. The habitat which it prefers is characterized by moderately dense forest comprising of littoral and beach forest. As a general behavior, the species prefers coastal habitats more than compared to innermost dense terrestrial forest. And the substratum is, it usually has high composition of sand and sandy loam soil. If you look at the picture on the left bottom of the screen, it depicts the mound of this species. So the bird, it builds large mound nest using soil, leaf litter, and other debris. These mounds are usually closer to the shore, and an average canopy cover over these mounds are 58%. Being a coastal species, there are several specific threats which it faces. First of all is vulnerability to climate change, and specifically sea level rise. Since this is a coastal species and the habitat is usually near shorelines, a sea level rise impacts the habitat of the species. Second, the species habitat is also prone to natural calamities like tsunami. One example is that the tsunami of 2004 has almost declined 70% of the total population in the Nicobar Island. And the surviving population which exists now, they are still facing the aftermath of the tsunami impacts. It faces anthropogenic pressures like hunting. So post-tsunami, the Nicobaris, they often face lack of food resources in the island, and therefore, they used to hunt the bird as well as their eggs for food resources. There are some developmental projects also, including the Great Nicobar Island projects. Some of the natural predators which which are endemic to the islands often predates on the birds as well as their eggs, which includes raptors, reticulated python, etc. Also, the habitat receives less coverage through protected areas. Although there are several studies which has focused on the population status of Nicobar megapods, their distribution, as well as habitat preference. But so far, the approach of modeling the distribution of the species and their habitat remains unexplored. With this information about the species, the information about its habitat, and recognizing where the research gap is, our broad aim was to map the distribution patterns and potential habitats of the Nicobar megapod for their long-term habitat monitoring. To do so, we had two specific objectives. The first one was to map the distribution of the species using Maxent algorithm. And once we got the output, our next objective was to map the suitable habitat for the species. The study area is the Nicobar Islands. It comprises of 22 different islands and is categorized into 3 subgroups. On the northernmost part, we have the Car Nicobar Island, which is dominated by human settlements. The Central Nicobar, which is also known as the Nanpuri group of islands, is above the Sombrero Channel, and The third group is the Great Nicobar Island group. You can notice that the entire island, they usually have a flatter terrain except for some parts in the Great Nicobar and Little Nicobar Island. Although there are several protected areas in the Nicobar Islands, however, the habitat of this species is often outside the boundary of the protected areas. In fact, the Telingchan Island, which is completely declared as a wildlife sanctuary in the Nancowry group of islands, is the only wildlife sanctuary which focuses on protection of the Nicobar megapode. The impacts of tsunami of 2004 has led to submergence of the wildlife sanctuary of Megapode Island. It is now completely submerged and there are no surviving population of Nicobar megapodes on that specific area. Our methodology comprised of 5 major steps. First one was we gathered species occurrence points through secondary data. These included published literature as well as points gathered from eBird platform. Next step was rarifying these data sets. Rarification is a statistics tool which helps us to filter out the data sets and get unique occurrence point. Third, we had selected environmental variables through literature review on the habitat ecology of the species. Fourth, we followed species distribution modeling in Maxim. And lastly, we had made an attempt to delineate the suitable habitats for the species by creating binary maps from the SDM output, which depicts the presence and absence of the species. Here I present the overall methodological flowchart which we had followed. First of all, we gathered data through literature and eBird. The total number of occurrence points which we could collate for the Nicobar megapode was 225. Following rarification in ArcGIS, the total number of occurrence points which were used for the model are 102. Based on our understanding through literature, we have selected few environmental variables and done a correlation analysis through which we have finalized 11 different layers. And using the total occurrence, filtered out occurrence point and the final layers, we generated species distribution modeling output. Now, the SDM maps in itself do not tell us much about the presence or absence of the species. There are certain thresholds which help us to generate binary layer. We have used minimum trading presence threshold to generate binary layers for the presence and absence of the species, and we have tried to do a visual interpretation and validation through satellite imagery. We had 3 primary sources for the occurrence point. First of all, we have gathered 56 locations through a published paper wherein they have shared the latitude and longitude value of where the species has been sighted. Second, we have collected 99 occurrence records from eBird, and there are 70 occurrence records with the latitude and longitude value in a report published in the year May 2012 by SECON. The total number of occurrence points were 225, which were rarified and filtered out, and finally we had used 103 occurrence points for a model. We had used total 11 number of variables corresponding to ecological and habitat requirements of the species, which includes canopy height, forest cover, land use and land cover, distance from beach, distance from road, distance from built up, elevation, slope, land surface temperature, annual mean temperature, and total precipitation. Since 7 out of the 11 variables were at a resolution 30 meters, we moved ahead and resampled the variables at a resolution of 30 meters, and the projection used was WGS 1984. Coming to the results, our model had an AUC value of 0.92. This map depicts the distribution, this SDM output from our Maxent algorithm as well as the occurrence point. As I've said earlier, this map in itself doesn't give us much information about the presence or absence of this species. Therefore, in the coming slide, I would be presenting the specific binary maps for the entire range as well as selected items. Based on our output, SDM output, the top 4 contributing layers were distance from beach, forest cover, elevation, and the height of canopy. Among these 4s, the maximum contribution was made by the distance from beach, which was 47.7%. We have tried to understand the environmental relationship through the variables which we have used. If you see on the right-hand side of the screen, these are the response curve of our top 4 most contributing variables, and what we could infer from our modeled output was that Nicobar megapods are likely present at location which are from a specific distance from beach. And through our output, we could determine this range to be 100 to 150 meters away from the shoreline. It also prefers area with a moderate canopy height, which comes out to be between 0 to 15 meters. And it has a tendency to prefer specific forest cover, which correspond to the moderately dense canopy height, which is 40 to 70%. The species has affinity, like it is usually found in flatter terrain, so it prefers lower elevation, and we can see that as the elevation increases, the probability of the species being present there decreases. So the preferred elevational range based on our model output was between 50 to 70 meters. Here is the presence map of the species. The total suitable habitat which we could identify through our models was 148 square kilometer spread throughout the different islands, throughout the 22 different islands in the Nicobar group. The Great Nicobar group had an area of 85.1 square kilometer delineated as suitable for the species, followed by the Nancowry group of island, which comprised 56.38 kilometers square. And lastly, our model also delineated the presence area in Car Nicobar and Baltimore Island, which is 6.62 square kilometer. Although the species is not found there, but since the terrain is flat and the habitat conditions are similar to what is there in the Nancowry and Great Nicobar group, therefore a very small portion of area has also been identified as suitable for Car Nicobar and Baltimore. There are 22 different islands, but I would like to take you through few of them. So if we see here, this is from the Nancowry group of island. We have, I'm showing Teresa and Nancowry. If you see on the left hand side of the group, the presence area have distinguished between the suitable and non-suitable coastal habitat of the species. It is not selected the mangrove forest or coconut plantation as suitable. Other 2 islands for which I'm presenting is Great Nicobar Island and the Kachal Island. Over here as well, if we see at the left hand side of the screen for Kachal Island, the area having higher cliffs and rubber plantation has not been delineated as suitable. So to summarize, the potential suitable area for Nicobar was 148 kilometers. Our top 4 contributing variables were distance from beach, forest cover, elevation, and canopy height. Our models, the results aligned with what is known about the species ecology. It preferred moderately dense forest cover, having a canopy height between 5 to 20 meters near shoreline. The accuracy of the results can be increased through field validation, and it will provide insight into future strategy for effective management of the species. These are the references which has been used in this study. And I would like to acknowledge the Director, Dean and Registrar of WII. I would like to thank Research Coordinator, and we are also thankful to Dr. K. Shivakumar for his guidance. And we also acknowledge efforts from our ex-SPA, who was Mr. Debanchan Sarkar. Thank you.
Thank you.
Now we come to the final presentation of this technical session. I would like to invite Mr. Varun, who will be talking of the birds in Thar Desert.
So Mr. Varun K is a quantitative ecologist interested in grasslands and deserts. His current work focuses on the ecology and conservation of threatened birds in the Thar Desert, and that is what he'll be talking about today. Over to Mr. Varun K.
Thank you. Good evening, everyone. It's been a long day of many presentations, and I hope you all have the energy for one more. My colleague Harshal already gave a brief introduction on what is happening to raptors of the country and particularly vultures. And I'll take some of the things from there and talk to you about threatened birds, particularly raptors that are found in the Thar Desert of India. As most of us already know, the Thar Desert is the most arid part of our country, found in the northwest of our country, with most part in India and some surrounding areas in Pakistan. The area is home to some of the largest open natural ecosystems that are currently left in our country, and also a very important area for conservation of many threatened species. Within this area, which I have denoted in red here, we have two protected areas, the Desert National Park, which is around 3,000 square kilometers, and the Jorbid Conservation Reserve, which is around 10 square kilometers, and three important bird areas, one in Desert National Park, one in Jorbid, and another one in Diyatra. This area at the same time is very important for many bird species, some of which have been classified as threatened and near threatened by the IUCN. As of we know that there are 19 threatened species that use this landscape during different times of the year, and many of them have a bulk of their breeding population restricted to this area. At the same time, the area is also a very important location on the Central Asian Flyway and is used as a migratory route by many species that use the western arm of the Central Asian Flyway. Most of the conservation in this landscape is currently focused around one of these threatened species, the Great Indian Bustard, and this involves creation, at least in Jaisalmer where the bird is still found, this has involved creating a lot of grassland enclosures to ensure habitat restoration and promote vegetation recovery. I'll be talking about some aspects of this going ahead in my presentation. While this area is very important for conservation and conservation of birds particularly, it is also facing massive land use changes in the last few decades. The main cause of this has been expansion and intensification of agriculture with the advent of irrigation in this region. And in recent times, this area has also been a hotspot for development of power infrastructure, with both wind and solar energy projects coming into this landscape in a very big proportion. In this broad context, we wanted to see what is the status of threatened birds in the Thar Desert. Apart from the Great Indian Bustard, we wanted to see what is happening to the other species that also use this same landscape. I'll be presenting data that was collected over the last 6 years through multiple projects, and would try to synthesize the population status for some of the species, the key threats that they face, and some other ecological aspects that we have observed. Before that, I would just like to just introduce you to some of the species that I'll be talking about. In the desert, we can categorize the threatened species into three broad categories: the breeders, the birds which are winter migrants, and the ones in the middle which we don't know much about. For this study, I focused only on the breeding birds and birds for which breeding was suspected, and asked the following questions. First was, what is the population status of some of these threatened species in the entire desert landscape? Second was, which species are being benefited by habitat restoration activities, the creation of enclosures which I mentioned to you earlier. And third, what are the key threats that some of these species are facing? Now, coming to the ecology of some of these species, the first one, the lagger falcon, is a resident breeder for which around 60 to 70% of the population is supposed to exist only in the Thar Desert. The red-headed vulture is found across India and in some neighboring countries like Cambodia. The Egyptian vulture, again, it's found in many places of the world, but there is a breeding subspecies that breeds only in the Thar Desert. The tawny eagle, another species distributed in Europe, Africa, and Asia, but having a subspecies that is restricted to India. The white-rump vulture, about which you have already heard from Harshal in a previous talk. The Indian vulture, again, which you have heard about. The white-throat bush rat, which is perhaps one of the most enigmatic passerines in the country, and the Indian spotted eagle, which has been recorded from this region. These were the main target species, and the data for this was collected broadly in this black outline that you see here. This is Bikaner and Jaisalmer districts of the Thar Desert, where WIA through its multiple programs has been working since working extensively in the last decade. As you can see, here depicted here in violet is the frequency of detecting either of these threatened species in a particular cell. And you can see that the two red polygons, which were the intensive study areas, are very important even from a regional perspective, as these areas house a lot of these threatened species and with good occurrence probabilities. Coming to what we did, a lot of the results that I'll be presenting were ancillary information gathered from population status surveys conducted for the Great Indian Bustard. The first amongst them was a systematic one-time survey conducted in the district of Bikaner, where around 13,000 square kilometers of the landscape was surveyed through vehicle transits of around 1,500 square kilometers, mainly through support of citizen volunteers and researchers of the institute. From 2023, we have been intensively surveying a smaller 1,200 square kilometer area in the district of Jaisalmer, and have been repeating these surveys in multiple seasons. At the same time, for the white-throat bush chat, we selected a smaller 500 square kilometer area and laid 154 kilometer of line transits and conducted these surveys in the winter of 23-24. At the same time, a lot of other allied activities were ongoing, and this included nest searches that happened in the last winter, camera trapping and carcass surveys under power lines and wind turbines that have been ongoing as part of the Bustard program since 2017, and some specific spot mapping exercises for the 3 rare vulture species that are found in the region. Coming to the results, the first species that we were interested in was the lager falcon. We found that the occupancy for the species was around 60% in the Bikaner district and 75% in the area that we surveyed in Jaisalmer. The species was found both in the flat grassland areas of the landscape as well as undulating sandy areas. The density of the species was found to be slightly higher in the Bikaner district, with 5.6 individuals estimated per 100 square kilometer. Pulling all of these things together and extrapolating it to a larger habitat, we estimate the population in the desert area of Bikaner and Jaisalmer to be around 2,000 birds. We also looked at what are the factors that are governing the encounter rates and densities of the species, and found that the density for the species increases as the proportion of grassland increases. In Jaisalmer, we were looking at whether the occupancy and density of the species is improved by habitat restoration measures, particularly the creation of enclosures. Here, the browns show unrestored areas or areas without any enclosures, and yellows are areas with enclosed grasslands in it. We found that occupancy was increasing only slightly, but there was a big difference in density of the species in unrestored and restored grasslands, indicating that the species was perhaps benefiting from grassland conservation activities that are happening in the desert. We also recorded multiple nests of the species, and the species was found to nest in Prosopis cineraria trees and even on electric towers. But the electric tower part was a double-edged sword. We also found that the bird is very susceptible to collisions with power lines and deaths due to collision with wind turbines. And our estimates suggest that in Jaisalmer, which has around 1,000 turbines and 1,000 kilometers of power lines, around 120 birds are dying annually, which is around 10% of the population. Prima facie, we have seen that the fecundity of the species is quite high. It gives around 4 eggs, and the breeding success is also not too bad. But given these direct threats, a lot of these juveniles do not survive until adulthood, and the populations are perhaps in an ecological trap of sorts in areas where renewable energy has spawned in big proportions. The next species of interest was the tawny eagle. We found that it was the occupancy for the tawny eagle was much higher in Jaisalmer, which is much more flatter than the areas surveyed in Bikaner. At the same time, the density was around 10 times what it was found in Bikaner. Our population estimates were around 3,200 birds in the entire desert region, which is around what we expected it to be. Pardon me for just taking you back to the lager falcon. The global estimate according to IUCN and BirdLife for the lager falcon is around 10,000 to 25,000 birds. But based on our results from area which is supposed to be around 60% of the area for the species, we expect that this estimate of 10 to 25,000 is a gross overestimate, and the status of the species is much more precarious than we thought of it earlier. The tawny eagle, on the contrary, had a population estimate that was similar to what was estimated beforehand, and it did not show any great trend with proportion of grasslands in the area or with restoration activities that are happening. We observed a few nests of the species, and most of them on Prosopis cineraria trees and some of them on electric towers. The breeding season for the species was in the winter, and we are currently planning to monitor some of these nests to see breeding success and other aspects of breeding. The species was relatively less prone to collision and electrocution with power lines, and did not face that direct threat as much as the other two species that I'll be talking about. The third one was the Egyptian vulture, which again, just like tawny eagle, had a much higher occupancy in the flatter areas of Jaisalmer and a very high density in the Jaisalmer landscape. Similar to the lager falcon, exactly opposite to the lager falcon, the population estimate for India according to IUCN and BirdLife is around 3,000 to 4,000 birds. But our surveys have found a lot more, and we already and the estimate from our data is around 1,500 birds in Bikaner and 13,000 birds in Jaisalmer. And again, contrary to lager falcons, the Egyptian vulture preferred disturbed, fragmented landscapes and not pure grasslands, and actually had a slightly negative effect due to restoration activities. The species was seen to be a summer breeder, and we are currently monitoring a few nests to understand a little bit more about it. This species was also very prone to deaths due to power infrastructure, and we estimate a lot of birds dying every year due to collision with turbines and power lines. This was by far the most commonly seen raptor under power lines during our carcass surveys. Now, coming to the three larger vulture species, it was not possible to estimate their population through traditional distance sampling based vehicle surveys. So instead, we collated all the information that we had about the species from multiple data sources and did a spot mapping exercise to arrive at a general figure of what their estimate in Jaisalmer would be. So our estimates are 20 to 30 predated vultures in Jaisalmer, and for which we have found 2 nests already and more efforts are ongoing. 50 to 70 white rump vultures, and the Indian vulture, which is not known to breed in the desert for sure. We still do not have any data that says that the Indian vulture is breeding in the desert landscape. The last one was the white-browed bush chat. Again, like I mentioned, this is one of the least understood species, least understood passerine species in the country, and based on a lot of literature review, expert consultation, and eBird records, we narrowed down on a 500 square kilometer area around Desert National Park to see what is the population status of the species. We found that the species likes very specific type of habitats. It does not like extremely disturbed agricultural areas, nor does it like old growth grasslands. It likes something in the middle. It likes agricultural fallows or moderately grazed grasslands that are recovering. And we found that its occupancy and density was slightly higher in areas that are being restored actively through enclosures as compared to areas agropastoral landscape outside. While these were some of the results that we have seen through 6 years of rather ancillary data collection in the entire desert landscape. We have since 2023 started doing some more formal research on some of these species. We are currently having a nest search and monitoring program which has started from the last year. We are also looking at carcass utilization surveys, particularly looking at interaction of raptors and free-ranging dogs around carcasses. And a study with some aspects of movement ecology is also planned. Some of these activities are currently being done under a RRCF funded project on focusing on carcass utilization by avian scavengers and dogs. And going ahead, we plan to increase scope of some of our surveys, plan to do a large-scale survey in the Jaisalmer district this year. We have identified around 50 nests already, some of which you saw in Harshal's presentation earlier. We plan to monitor these nests over the longer term and see demographic parameters, nesting success, and many other things such as that. The mortality surveys and the camera trapping is expected to continue in the longer run and keep providing us insights on that front. And we hope to do some movement ecology studies to tag birds in the future. There are some other things that we think are important with respect to raptors in the desert. This includes looking at vulture restaurants and whether construction of these vulture restaurants is useful in this landscape. We want to do diet analysis to understand what exactly are these birds eating and whether there is NSA prevalence in some of this diet. And finally, do a bit of genetic analysis to resolve some of the problems involving subspecies level classification of some of these species. There are a lot of people to thank here. The data that I presented was collected by multiple people over multiple projects and multiple years. And I would like to thank all of them. And I thank all of you for a patient here. Thank you.
Thank you for being perfectly on time. I request the other four presenters to please come on the desk. We have received the questions and we have received a lot of interesting questions I do not wish to miss. So for the sake of brevity, I might rephrase the questions and I request researchers to please answer in brief to the point. So first we have a series of questions for Rajdeep Mitra. So Dr. K. Ramesh asks that population trends, it's natural for it to fluctuate. So instead of focusing on a specific island, shouldn't we be looking at regional scale?
Thank you for the question, sir. The case here is the population assessment which we did primarily for the sooty tern and brown noddy. They are known to nest in PT Island. The other island where nesting reports have been documented way past in 2018 was in Cheriavani Island. There were very few number of birds that were last documented in 2018. After that, there has not been any documentation of this because of the sea level rise and other cases. That sand flat is actually receding. That land is receding actually. So PT Island was the only island where the nesting population was observed, and that's why I chose that island. We chose that island for our population estimation study.
Next question is from Dr. Raman. Dr. Raman asks, what's the estimated degree of submergence threat due to the predicted rise in ocean level?
See, as I mentioned in my slide, that 0.4 to 0.9 millimeter per year, the sea level is rising according to the Government of India's data. And I believe not currently, but definitely within years, as in Dry Tortuga National Park in Florida, it has seen that within 8 decades, the population from 3 lakh receded to around 3 lakh, I mean, from population of 3 million receded to 3,50,000, something like that. So definitely there is a threat, and we cannot say for sure for, I mean, after how many years it will happen, but definitely the other islands would definitely lose. Maybe PT Island, which is only a 1.5 meter, it's its elevation from the sea level. So with the rising sea level, I think there will be an issue, but we need to assess it with systematic studies going forward.
Ms. Swapnali asks, 6 visits, is that enough for making robust population estimations or trend?
Thank you so much for the question. As I mentioned, if you look historically, these islands have always been extremely difficult to access. I mean, landing on the island has been so difficult that the 5 or 6 studies that I mentioned, only 2 of them managed to land on that island. I actually tried to make landing on multiple occasions, but due to the rough oceanic conditions, from Kavarati to reach the nearby waters of PT Island was a challenge. And then to again reach there and land on the island, it took around 100 to 200 meters of swimming. And as you know, the sea conditions can get even rougher. So you have to swim and get into the island is another challenge. So I know this is the case, but our study also is one of the very first systematic observation of 3 months. The previous studies have either been for a single visit or 2 visit surveys. So, and we will actually try to replicate that as soon as the monsoon season ends and the islands reopen for inter-island communications in September 15. And probably with that data, we will try to give you a much better understanding of the population assessment in the next presentation, probably.
Thank you, Rajdeep. So next we have Ms. Khushi, who has asked, what's so special about PT Island?
Fantastic question, Khushi. Actually, as my presentation name actually indicates, Pakshipiti. This island is probably one of the only known nesting seabird locations from of sooty tern and brown noddy, which are as a least concern species, globally distributed across. But as my first slide indicate, that 80% of the least concern species are actually showing population decline. And me, as a budding ecologist or a biologist, as you can say, I want to focus more on the least concern species that are actually losing population, rather than directly focusing on the critically endangered or near threatened or endangered species. So for me, focusing on a species like that, which is losing its population and might lose the entire population of PT Island from 20,000 to around 1,400 something from the last data, and from my data it shows. So it definitely has a special place in my heart, and definitely would want to focus on those species. That's why Pakshi PT Island.
Last question for you, lastly, from Dr. Manjari. So among all the species, most of the species are declining, but that's not the case for sooty tern. Why so?
Thank you, ma'am, for the question. See, the sooty tern population, if you look at Matthew Ambedkar's data in 1963, they reported around 20,000 birds that would nest in that island, way back in 1963. So, and from there, the numbers have come down to, from my study, I observed 1,000 birds and around 425 nests, which we believe with the monsoonal season, the nest count would rise. As it is inaccessible, we do not know what is the true scale of their nesting. But I think the numbers have definitely gone down because of the egg harvesting, which again, I went around asking people, doing a questionnaire survey, because of the shift in the economical viability. Like, you don't want to be a fisherman spending that much money for diesel fuel going into that island, and you only find 100 nests. And you come back and sell those 100 nests with 2 rupees, getting around like 1,000 rupees or 800 rupees, and then you have to spend like 5,000, 10,000 rupees for the fuel cost. So it's not viable anymore. Sir, definitely, it is a special island, and I think, sir, speciality definitely depends on people. It is special. I think, sir, special is a perspective that depends on different people, and for me, I can say it's definitely special. And as I mentioned, one of those places with few nesting sites in the country, and for me, we'll be able to go there and assess the island. Definitely, it has a special place.
Yeah, maybe we can discuss it tomorrow about chai. We'll have the next set of questions for Ms. Aarti.
Thank you.
So Aarti, Ms. Swapnali asks that during your questionnaire survey, did you also do questions on threat assessments?
Can you repeat?
Yeah, the questions are there. So during the questionnaire surveys, was there also a component of threat assessment? Like, did you ask people about the threats?
Yeah, I have asked them, but people are really reluctant to tell a lot of information because I think there is a suspicion of nest collection throughout the islands. So they were not so open to most of the questions.
And the next question is from Dr. Johnson. He asks that there are a lot of swiftlet nests around APWD guest house in Great Nicobar. Are these same or different from edible nest swiftlets?
There are plume-toed swiftlets nesting in APWD guest house. I did survey them. There is no edible nest swiftlets found there.
Okay. Thank you, Arti. Next, we have questions for Mr. Harshad. So, Mr. Rajesh asks that you have mentioned about the increase and decrease in number of the nesting sites for the bird species. Have you also considered re-nesting or nest shifting to another location?
So basically the data was about the nesting site locations which were earlier present from the areas and how they are shifted now. So my surveys were for the nesting surveys, nesting site surveys. So I think that's how it answers the questions.
Thank you.
Yeah.
Next question is from me to George. There are two questions. I'll take the second question. These vultures seem to be very species specific when it comes to selecting the tree, nesting tree. Is that true?
From the data which I have collected, there are evidences that they are species specific, but if we take a bigger look, the species which they are selecting are the tallest trees in that ecosystem or that present areas. So that's how they are preferring, because vulture being a big bird, it requires a certain height to fly off, and I guess they are nesting on the tallest trees possibly in that ecosystem, and that's how they are
Thank you, Harshit. The next question is from Dr. Khurshid. He also has some comments and queries on your study. Maybe question comment part you can discuss with him. The question is, what are the sites in Jammu and Kashmir that you have surveyed for Himalayan griffin plus bearded vulture?
Okay. So bearded vulture was found near Doda in Jammu, and Kishtwar National Park for the Himalayan vultures. So these are the areas where the vultures have been found to be nesting during my surveys.
Thank you. Next question we have is from Dr. Sudeep. There are two questions. Again, I'll take the first question. What are your thoughts on how to use nest count as a population monitoring metric for these vultures?
So basically, what we can use, what we have done is we have given a minimum number of the vultures. Obviously, there will be a lot of more vultures and lot of nests which are undetected, but at least we are trying to give a conservative number of minimum individuals which are there, and there will be a lot many more individuals apart from this. So I think that should be the case if we are trying to estimate a population, and it's again an estimate, it's not the exact number which we are giving of.
Thank you, Harshit. The last question for you is from Dr. K. Ramesh. He asks, why do you think the nest sites are increasing in Madhya Pradesh?
So this is sorry, sir. Can for what?
I think I was just going to ask because why do you think that the population trend, some other places as a state showing a problem, even the states which are supposed to be protected, certain states showing.
So for the for the long-billed vultures, so for the Indian vultures, there are majorly the Madhya Pradesh state has a lot of cliff and lot of protected areas which are present in that state, and I guess because of that there might be a shift in that area.
But I think it's more to do with the gauchalas. I think one of the important aspects that we intend to miss with the vultures. So it's not only protection. I think the kind of footage that is available. I think that the aspects which are not generally built into the protection, I think we should actually.
So actually, during my surveys, I was when I was around Madhya Pradesh, I happened to look around the goshalas, but then there are majority of the goshalas which are not allowed to put out their carcasses out in the wild. I mean, out in the open for the vultures or any other species. They are just burying it, and there are certain areas where they are doing that, but then majority of them are just burying the carcass across India, and I think that is also a problem for vultures or any other species where we are just burying out their food. That's what I feel.
Thank you, Harshit.
Thank you.
Next, we have questions for Ms. Neha Pandey. So the first question for you is, so you mentioned about the temperature. So was it air temperature or soil temperature that you did?
So we have used two set of temperature data set. One was land surface temperature, which was derived through Landsat data. It is the surface temperature. And one was the temperature through bioclimatic variables.
Okay. Next question is from Ms. Swapnali. During the model building, did you also take into consideration soil parameters, given that they make mounds?
So soil parameters was not taken, but we have used a surrogate for it, which was one of the class in our land use and land cover map.
Do you have plans for ground truthing of the model that you have obtained?
We have, but given that getting the field permission for Nicobar Islands are very difficult, we don't have a specific month or time period in which we'll be going.
Final question for you is from Dr. Nandri. She asks, what are the possible recommendations you can make for conservation of this species?
So actually, we are still working on improving the accuracy of a model, and as of now, we have not concluded anything solid.
Sorry, that was not the final question. There's two more questions. Yes. We have a question from Johnson sir, who asks, Zelicide in the Port Blair, did you also consult them or took their data for making your prediction models?
So these data sets, like these locations, they are already there in one of the report which were included for the species occurrence point. So those points have been taken.
Okay. Can I add?
There are two points. One is that so much of data is available. If you use their data, your prediction will be more accurate. Second thing, the question related to Dr. Sopnali. So the soil quality is very important, and they are making keep on managing the farm building. So the terrain and the soil quality is very important. That also should be taken care of when you are building. Thank you.
Thank you for your feedback, sir. We are still trying to make a model more accurate, and we will try to include one or two parameters related to soil.
Finally, we have two questions from Dr. Kiran regarding the methodology that you will use. The first is that Maxent is a presence only data model. So how do you account, and it is prone to biases from pseudo absences. So did you consider that? Or how do you account for the biases in the presence parameter?
I have actually not understood the question.
See, basically, the thing is that when you do all these present points, also for most people, the maximum model. We picked up the points, the islands are disturbed. So, species may be that because of conservation is not there. So, when you collect secondary data like this, you tend to pick up points which are actually a presence point, but they are not there. Or, sometimes you collect vice versa. So I think, so even in the previous presentation that we saw about, I think, probably for so it actually tends to bias towards the sample point that we have. So therefore, I think for such a species like output, I would say in ideal precision, I won't do a model. I will basically ask a questionnaire and get real real-time data. I simply map it out. I don't think you should do it. But the second question I also want to add, see, megapods as a species much more microclimate oriented. When you do a model with the large scale data, you're mixing more local scale and large scale. So without optimizing that. So I think instead of looking at some very ambitious model, instead you actually collect the primary data and map. It might be much more useful for the kind of project that makes you wants to do. Thank you. You don't need to answer.
Thank you.
Thank you, Sneha.
Thank you.
So next, the final questions we have for Mr. Dehradun. So we have Sunny. He asks that legged falcons show contrasting or opposite densities in Bikaner and Jaisalmer. Why so?
Good question. I also want to find out. Hopeful in this piece.
And the second question for you is from Dr. K. Ramesh.
Sir, the occupancy estimates that I showed, those were posterior predictions from a Bayesian model, which were 95 CI. Getting it back to a frequentist model, you'll have those standard errors to be much smaller. Yeah.
My question was more about looking at this occupancy rate, which has got a wider value. Whereas when you look at densities and abundances, you have high, very low standard error. I'm curious, is it more to do with you have large sample size with the raw people? Because to me, since I don't see the confidence interval, the estimate, it's very difficult to understand what is actually the population.
Right, sir. One is about sample size, like you mentioned. We have done a lot of fractal surveys to get the density estimates, whereas some of the occupancy estimates are from a smaller data. But broadly, I think this is just a difference between how we are putting in occupancy here. The box plot was what a probable value could be, and we were more interested in whether restoration is having an effect on occupancy or not, more than the actual estimate.
No, I was actually expecting you to present the confidence level associated with density level rather than just a standard error.
Fair enough, sir. Right. Point taken.
Okay, that was the last question.
Thank you.
So we have come to the end of the session. I suppose we are just about in time. There's always so many questions to ask. But first, I would like to congratulate all of the researchers for. I see quite a few people walking away. Please sit down. It's not over. Please sit down. This is not a cinema. Please sit down. Another 5 minutes. I mean, I've been given the opportunity to speak for 2 minutes. So I will first request my colleague Dr. Ashish to make a few remarks on the session.
As a bird enthusiast and a bird researcher, it's always fun to hear about the work that people have done on birds. And we have had presentations from the coasts of Tamil Nadu to coasts of our island to the deserts and to the Himalayan forests and central India forests. It was a fantastic study. Though I felt that it was just one study, we had some positive news. The edibleness swifted. It was fantastic to know that the populations are rising and they have adapted to urban landscape and they're nesting in buildings. But it wasn't so positive news with the other species. So I hope next IARS will get to hear. And even in general award NCS, I would love to hear a lot of positive research news from about our bird diversity.
Okay, so I'll try and make it sweet and quick and fast. Like I said, fantastic research done by our researchers from WA as well as from SACOM. It's heartening to see a lot of interesting stories. One thing that is very, very evident and clear is that footwork, getting down on the field, getting down to going collecting data, swimming into those islands or getting to each of those caves in these remote corners in Nicobar or Andaman is simply amazing. So I personally like to congratulate those researchers, trying to get us this information. Of course, that there is a lot more to be done, but this is somewhere at the start. And what is truly important in many of these studies is the baseline data. For example, the vulture data in which I'm currently involved. We all know that the vulture populations have declined almost 99%, the three species that was discussed primarily. And this we've been hearing about since the 2000. And there are quite a lot of people on the ground collecting data on these birds. There is conservation focus, so there's a lot of people involved. But what we definitely notice is that all of this information is scattered. So thanks to this IDWH program, where we've been able to go about collecting data, meticulously. Of course, there will be a few sites missed out. But this is all that is. And these are the sites that most people know. And it is not that there are still some nesting sites where people are not going there and things like that. So this is all that is there. And when we will try and when we bring all of this data together into what we would like to know as the National Vulture Database, then from now on, it's going to be I wouldn't say easy, a very good way of monitoring what is happening to each nest. So what Mr. Harshal talked about is those nesting sites. But in fact, we have GPS locations of each nest. Of course, the Indian vulture nesting on cliff faces, we wouldn't have each nest, but we would generally know this is where there are 40 or 50 birds that are nesting there, 50 nests that are there. So this information is extremely critical for us to go back in the future when we would go back and see whether these particular populations have done well or not. And likewise, the other presentation coming from the western part of the country, where Warren talked about the Ladakh, such an uncommon bird, but generally is thought to occur all across the arid parts of India. But here is a study where we are getting some baselines of what is happening with these populations. Of course, there's lots of issues possibly with the way the statistical estimates have arrived. But I would just generally look at it as a very important baseline data on which we can. So to finally conclude, I would like to thank the seminar committee for giving us the opportunity to be on the stage and speak to these presentations. And I would like to thank Ashish as well as Dr. Frank for supporting me on this session. Thank you so much. Thanks to Dr. Frank.
All the research scholars for sticking to time. It has been wonderful. Otherwise, it goes on 96, 37, 8 times. So I hope we'll maintain the same tempo tomorrow. Stick to the time and sit and have a productive day like this. I was personally very happy and impressed with the quality of work that was presented today. So a lot of exciting things happening tonight. Go back and see the iPhone launch, iPhone 16. And, tomorrow night also a lot of interesting events coming up. So Have a nice time. See you tomorrow at 9:30. So please be seated by 9:20 tomorrow.